Boy, this is a compliant group. So the other request I have is if this is being webcast, so if you could remember, I feel like a movie theater announcement, please turn off your cell phones or put them on vibrate so that they don't interrupt the proceedings. So my name is Carolyn Deal, and on behalf of the National Institute of Allergy and Infectious Diseases, I want to welcome you and thank you all for coming today to this workshop that we're holding jointly with the Food and Drug Administration, the Center for Biologics, on live biotherapeutic products. I think all of us, and certainly by the amount of interest there was in this workshop, we realize that this is a rapidly moving, evolving, and important area. NIAID has supported research in this area for quite a while, mainly in the basic area, and it's exciting to see it evolve from the basic research area into translational work leading to product development. However, we all know this does pose new challenges, questions, but I would say also opportunities. And we hope that these opportunities can lead to new products that will improve public health. For those reasons, NIAID partnered with the FDA to start a discussion with the scientific community and our manufacturing partners as to how best to approach the need for rigorous clinical studies to evaluate these products. For this, we know there are two requirements. One is well-characterized products, and the other is well-designed clinical studies with defined endpoints. These are some of the topics that we hope that we can discuss today and get your input and thoughts and see how we can all collectively move forward. We really look forward to this discussion and hope that everyone at the end of the day will come away with some new ideas. And now it's my great pleasure to introduce Dr. Peter Marks, who's the director of CBER, who will go into more detail about today's program. Thank you, Peter, for coming.
So good morning. I want to welcome all of you in the room and on the webinar to this workshop on the science and regulation of live microbiome-based products used to prevent, treat, or cure disease in humans. Before I go further, I want to thank colleagues at the National Institute of Allergy and Infectious Diseases and at the Food and Drug Administration for putting together such a stimulating program. We really have a group of presenters assembled today that's highly qualified to discuss the relevant issues. And I hope you'll find all the presentations, panels, and interactive dialogue informative and engaging. Just to orient you to the day, we'll start off with a keynote address by Dr. Vince Young of the University of Michigan, and this will be followed by two presentations on the regulatory framework for probiotics and live microbiome-based products. After the morning break, we'll first hear part of presentations on the safety and effectiveness of live microbiome-based products used to treat, prevent, or cure disease in humans. And these presentations and the discussions will continue after lunch. And then following the afternoon break, we'll hear presentations and a discussion of strain selection for live microbiome-based products to prevent, treat, or cure disease in humans. Now, it's certainly true that over the past 2 decades, the relevance of the human microbiome to maintain health and to prevent the occurrence of disease has ever been more greatly appreciated. And I think the following, which is quoted from the science journalist Michael Spector, summarizes this all quite nicely. I think his words are much better than mine could be. We inherit every one of our genes, but we leave the womb without a single microbe. As we pass through our mother's birth canal, we begin to attract entire colonies of bacteria. By the time a child can crawl, she or he has been blanketed by an enormous, unseen cloud of microorganisms, 100 trillion or more. They're bacteria mostly, but also viruses and fungi, including a variety of yeasts. And they come to us from all directions: other people, food, furniture, clothing, cars, buildings, trees, pets, and even the air we breathe. They congregate in our digestive systems and our mouths, fill the space between our teeth, cover our skin, and line our throats. We're inhabited by as many as 10,000 bacterial species, and those cells outnumber those which we consider our own by 10 to 1 and weigh, all told, about 3 pounds, the same as our brain. Together they're referred to as our microbiome, and they play such a critical role in our lives that scientists have begun to reconsider what it means to be human. So it's my sincere hope today that you'll find the presentation stimulating and the dialogue will provoke questions that will help define where additional work is needed to fully realize the potential of microbiome-based products to prevent, treat, or cure disease in humans. And with that, I'll wish you all a wonderful day engaging on this topic, and I think we're actually just about on time. So thanks very much.
Thanks, Peter. So with that, I'll introduce our first speaker. Our keynote address today is by Dr. Vince Young. Vince got his bachelor's degree from MIT and then went on to Stanford for his MD and PhD before starting his first faculty position at Michigan State University. 2007, Vince moved to the University of Michigan, which is where I met him, and we've interacted quite a bit since then. He is currently the William Henry Fitzbutler Professor in the Department of Internal Medicine and Infectious Diseases, and he has a joint appointment in the Department of Microbiology and Immunology. And I think most of you probably know Vince. Vince has been on the cutting edge of the microbiome field and also C. difficile, both in the context of the microbiome and beyond. So with that, I will turn it over to Vince, who's going to give us an overview of the microbiome from his perspective as a clinician scientist.
Thanks to Paul, thanks to the FDA and NIAID for giving me the opportunity to speak today. I want to tell you a bit about the microbiome, and I know people have varying expertise, varying things, so I apologize for those people who've heard me talk before, and I'm going over things again, but I wanted to kind of set the stage for the day. We're going to have a lot of discussions about the microbiome, and I think it might be useful, since I am a clinician and I've had the opportunity to kind of think about how we might use this in clinical medicine, to kind of set a framework for this. And first of all, my disclosures. I have done some consultantships, but I won't be talking about any of that work, and I won't have any discussions of off-label use of any FDA-approved therapies, and I've retired from football. Microbiome, right? We all hear about it. This was from Saturday on the airplane back from San Diego. Like, what's the latest count when you put microbiome into PubMed? So we're up to 45,937 papers as of Saturday. And finally, we actually have more primary literature than reviews. There was a time where we kind of were the other way around. There was like 3 times as many reviews on the word microbiome than there was data. But, you know, and you can kind of see some of the ones that come up with best matches there. It's kind of interesting. So I published this a year ago in BMJ because I think clinicians are very interested in the microbiome. And I was actually at the American College of Physicians in May at their national meeting. And I was speaking to a group, there were probably about 1,000 practicing internal medicine physicians at the ACP. And I asked them, who's heard of the microbiome? Everyone laughs, every hand went up. And I said, who has had patients that have asked them questions about the microbiome? And about 70% of the hands in the room went up. And then I asked, who has had patients bring in microbiome surveys that they've gotten through various commercial, I won't name any of those entities right now, but places that you can get a microbiome survey done? About 30% of the practicing internal medicine clinicians in the room raised their hand. And then the final question was, okay, who knew what to do with these? And there were no hands up. And I said, yeah, you notice my hand isn't up either, because I'm not sure what to do either. Because, you know, this is something that we encounter all the time. On Saturday, I also love doing the Google News search. You know, look at what we're talking about with the microbiome. It's the usual things. Is your microbiome making you sick? This one, with regards to today, they took a couple of the papers that were published from the group out of Israel a couple weeks ago, and kind of saying that, oh yeah, the probiotics don't do anything. I made sure to read those Cell papers. They didn't come out and say that, but that's how it was interpreted in the news. So it's out there. There are a lot of people interested in the microbiome. So for the purposes of my talk, and I know other people use different definitions, but when I refer to the microbiome, I am talking about the microbes, but I'm also talking about the environment they inhabit. In other words, the soil, the human body. And this is important for me because when I refer to the gut microbiome, this is the organisms, these are the compounds that are being produced in there. And what's very important with regards to the latter, that is actually due to the metabolism of the host and the microbe. So it's actually the biome. That's the root of the word, that it's this environment there. And then when I use the word microbiota, I'm going to just be referring to the microbes. So, we've all seen various pictures like this. This idea that it's a forgotten organ. We have a lot of different species in there as we go through the GI tract, as we go through the lung, as we go through the skin, as we go to the GU tract. They're microbes in and on us. Okay? And they can be very important in terms of what we're doing. And they can be important for two ways. They can be important both in terms of anatomy. When we're talking about the microbiota, we can just be wanting to know who's there. What's the anatomy? Taking census, doing 16S surveys to say what are the microbes that are there, doing fungal surveys, doing sequencing so that we can look at the viruses that are there. But the physiology, as a physician, it's important not to know just the anatomy, but we also want to know what they're doing. In other words, what can they do, but actually what are they doing at any given time? And this is from a review this is kind of modified from a review where we kind of looked at the kind of plethora of different techniques that people use to study the indigenous microbiota and the microbiome itself as we look at proteomics, metabolomics, cultivation is still important. We do a lot of 16S surveys, and if we're going to try to come up with a biotherapeutic, I can't imagine that we're going to ever treat someone with a 200 base pair snippet of their 16S gene, but we might treat them with an organism that contains that 16S gene. What do we mean by anatomy? Well, if we look at the human anatomy, we do note that there are different organisms that are on different parts of the body. And they're fairly characteristic, but there's a lot of individual variation. We knew this from the Human Microbiome Project, that everyone's sort of individual. And so the anatomy can vary, but what we are finding a little bit more is that the physiology, the functions of these communities seem to be relatively stable in individuals that we would consider quote-unquote healthy. And they carry out a lot of different conversions. They can break down compounds. We hear a lot about how fermentation of resistant starch can give rise to short-chain fatty acids, which may influence how obese we are or how much inflammation we have in our gut. We can actually take xenobiotics, drugs and toxins. We can convert them in multiple ways. I'll discuss that a little bit later with some examples. The microbes themselves can just synthesize things that are useful to us. And there is a lot of signaling back and forth between the host and the microbes through the epithelium, through the immune system. And so this microbiome here, as you can see it, this is all related to the host and the microbes. It has a pretty dramatic and very complicated physiology. And what can we learn about it? Well, I'm going to pick a couple examples. I'm going to start out with a little bit of pharmacology. FDA is sponsoring, so I'll talk about drugs and microbes. But I am an infectious disease physician, and we are here at NIAID, so we'll end up on that. And I know that there are a number of people who are giving talks on C. diff, and they have shorter talks, so feel free to just kind of skip over some of your intro slides as you need to, because I'm going to kind of cover C. difficile in some detail here. But drug metabolism, I was saying that the microbes can do all sorts of things, and they can metabolize xenobiotics, which includes drugs. And digoxin is a classic example. In medical school, I was taught, digoxin is a great cardiac aminoglycoside, it's good for arrhythmias, et cetera, except for the fact that it has this narrow therapeutic index. The amount of digoxin that you give to a person between helping them and becoming toxic is very, very narrow. And even more tricky is the fact that some patients you can give the tiniest whiff of digoxin and they go to toxic levels. Other patients you can keep on upping and upping the dose before you get therapy and they don't seem to have toxicity. Well, a while ago, it was reported that this particular bacteria, Agarthella lenta, could metabolize digoxin. And that kind of stayed there for about 20 years until Peter Turnbull decided to revisit it and actually figure out exactly how did E. lenta inactivate cardiac glycosides, and could that actually be used to predict the ability of a person to actually get toxic or actually have a good therapeutic effect. And when he found out, as a good microbiologist, he kind of got different strains of E. lenta and found that not all of them had the ability to reduce the drug. So it actually varied. And that's one thing, that's very important. That's actually why it's important not to just get a snippet of 16S and say, oh, you have Elenta there. Depending on which strain you have, it might be able to reduce, the type strain is able to reduce cardiac glycosides like digoxin, but there's others that don't. And then he looked very carefully to see what was happening. He took patients that were these high reducers versus not, and he showed that, yeah, okay, fine, they could reduce. The microbiota itself can convert, and Elenta itself could convert digoxin. But there seemed to be some sort of interaction between this organism, Elenta, that has this particular gene cluster that he found out that was very important for this bioconversion. And there was interactions with the microbiota. Okay, so here are the two strains of Elenta, this one that is very good at reducing digoxin, this one that can't. And he took a patient who did not have the ability in their microbiota to reduce digoxin, and when he added the type strain, sure, he actually got good reduction, and in fact, even more reduction based on the number of organisms than Elenta alone, where the gut microbiota did not enhance the ability of the organism that didn't have this ability. So there's not just the bug, it's the bug and all the other microbes that are there. And so sometimes we try to reduce things too much. Oh, does the person have this organism? Or does this person have a microbiota? But it's more complicated than that. When you actually put human genetics on here, now you're really building up this idea that this is a very complex system. How about something a little bit more modern in terms of therapy? Immunotherapy, cancer and immunotherapy. It's being advertised on TV now, right? This so-and-so's place that does all of this anti-tumor therapy based on the host immune system. There are a number of drugs that have come out. And about 3 years ago, I was being invited by some of my old residency and med school classmates who are all in hem-onc. And I said, you guys like these papers that came out in Science, didn't you? You want someone to start? Finally, after wondering, what are you doing studying this microbiome thing, Vince? They all wanted me to come and talk. Because there were 2 papers that came out on animal studies where they showed that the efficacy of cancer immunotherapy was modified by the microbiota. I'm going to go over these 2 papers briefly, not so much that I want you to have the details, but I want you to understand how we can actually look preclinically to study the effects of the microbiota. So in this first paper where they were looking at ipilimumab, and they showed that the microbiota was necessary for anti-CTLA-4 therapy. And what they did is, okay, so here's the therapy. They put tumors into mice, and if they use basically an isotype control antibody, these tumors get bigger and bigger and bigger. But if they give one that's related to ipilimumab, anti-CTLA-4, the tumors shrink. Okay? So you have a good, or don't grow as fast. They don't necessarily shrink, but they don't grow as fast when they're transplanted into these mice. Now, they did something interesting. You can raise mice without any microbes. And if you take these germ-free mice and inject the tumor, and it doesn't matter now if you give the anti-CTLA-4A antibody, the tumors grow just the same as if they got an isotype control antibody. And they could also kind of replicate this by taking animals that do have an intact microbiota, but kind of suppressing it somewhat by giving an antibiotic cocktail. And once again, instead of seeing the anti-tumor effect, they've eliminated the anti-tumor effect by changing the microbiota. And they did some other studies we won't go into here. It's not all antibiotics. It depends what the spectrum of activity is. So there are certain elements of the microbiota that are responsible for mediating this anti-CTLA-4 response. So they did the same thing with anti-PD-L1 therapy, and they did a different kind of study. Again, don't worry about the details of what the message is, but here's another way to study it. Okay, once again, they were taking mice, and people say, oh yeah, get a black 6 mouse, wild-type mouse. Doesn't matter where you get it from, as long as they're genetically identical, you should have the same results. Not true. If you buy your mice from Taconic or Jackson Labs, 2 of the major vendors, you had different responses in genetically identical mice. So, again, you didn't have as good a response to the anti-PD-L1 antibodies if you buy your mice from Taconic as opposed to if you buy your mice from Jackson. So the differences that people might see in their studies, it depends where they buy their quote-unquote genetically identical mice. Okay? They did some immunology here. We'll kind of skip that a little bit. What they did show, though, if you house the mice together before you start treating them, And mice are very convenient. They like to give each other fecal transplants. They'll pick up their neighbor's feces and they'll eat it. And so you kind of, quote unquote, normalize or at least kind of neutralize the effects that you might have of a different microbiota. At that point, if you house Jackson and Taconic mice, now you have the same response in both. Okay? And they kind of worked a little bit further on this to kind of figure out that, yeah, there are certain elements of the microbiota that might be important. Well, that was all kind of fun. It was in mice. That was 2015. This is an example of how fast things can move. Just in January of this year, three papers came out in Science. And these are studies now in humans that are showing that the microbiota actually has some sort of influence on anti-PD-L1 therapy. Again, for epithelial tumors, melanoma. Now, the interesting thing about these papers is that they got the same results as far as, you know, the microbiota being able to help or influence a response, but there were some differences as to what they found as the microbes that are important, quote unquote, or at least associated with these kinds of effects. Showing again, not everything's the same. It's not just an individual organism that you need to find. Okay, let's find this organism. If you have it, you don't. It's a little bit more complicated than that. And I think that's why there's some frustration in the field. And we'll be hearing some talks about people who are using single-strain probiotics, looking for communities, looking for combinations, and perhaps trying to tailor the therapy based on the patient. Let's move to my favorite topic, infectious diseases. Okay, so for over 100 years, we've been associating microbes with disease using things like Koch's postulates of finding an organism, giving it to a medical student or a mouse, recreating the disease, pulling it out again, and, you know, and saying, okay, this is how we can get pathogens. But here's a classic case that we would find. So this was the case that was first presented to me about 30 years ago when I was a med student. Says, okay, you have a patient who has chronic lung problems. He comes in, he has an exacerbation of his chronic bronchitis. He's given, quote unquote, broad spectrum antibiotics. This is more of a more modern kind of therapy as opposed to we might have given when I was a med student. And he gets better from the pulmonary standpoint. But 3 days into hospitalization, he develops abdominal pain, diarrhea, hypotension, actually has to get transferred to the intensive care unit. You know, what happened? You're trying to treat a person's pulmonary infection with antibiotics, and now he gets GI distress. You know, he didn't, hopefully our food's clean. He didn't develop a gastroenteritis in the hospital. What's going on? Well, this is C. difficile. A lot of people know about C. diff. And it was sort of even said at that time by one of my PhD advisors, Stan Falco. He's saying, well, we disrupted the normal, he referred to it as flora at the time. You know, the normal gut flora was disrupted by the antibiotics, and somehow this allows C. difficile to come in. And sort of the paradigm is that, you know, people have a normal microbiota. It has this magical property of colonization resistance, able to keep away certain pathogens from growing in. But when you alter the community with antibiotics, you create a more susceptible microbiota, whatever that means. And C. difficile is a spore former. And interestingly enough, the spores are unfortunately all over the hospital, and you see the alcohol dispensers in the hospital. They don't get rid of the spores. They just help you spread them around, perhaps a little bit more. But when the spores encounter the right environment, we'll talk about that a little bit, of this susceptible microbiota, the spores germinate. You have the vegetative form that produces a very potent toxin that causes all the damage in the intestinal tract. That's when you get disease. And depending on who you are, what the strain is, perhaps what the microbiota are, you might have mild disease, even asymptomatic colonization, or you might actually have more severe or fulminant disease. And we don't know all of the aspects of the microbiota, the pathogen, and the host that determine all of that. But there are a number of us who are studying that quite intently. But as an infectious disease doc, even if you got in trouble with antibiotics, hopefully more antibiotics will get you out of trouble. So you treat the C. difficile. Hopefully when you stop all the antibiotics, the microbiota goes back to normal, everything's back to normal, and you don't have disease. But a lot of patients, unfortunately, when you stop the antibiotics, about 20%, depending on the series, after the initial treatment, will develop recurrent disease. You stop antibiotics, even though they got better when you were treating their C. difficile, they have disease, they're toxin positive again, and you have C. diff infection going around, and you can treat them with more antibiotics, and you can go through this recurrent cycle. And we're going to hear about some of the approaches that people have for breaking this, but one of them that's caused, has a lot of interest is this idea of fecal transplant. My younger son is a freshman at the University of Michigan. He's taking freshman biology, and in the second lecture, they're talking about fecal transplants for C. difficile. He actually texted me with the slide of the professor, and kind of giving me the thumbs up. And interesting enough, he happened to be sitting next to a friend of his from high school, who's the son of a friend of mine who's the gastroenterologist who started the fecal transplant program at Michigan. So I actually kind of wrote a quick email to the professor. He had at least 2 people who were pretty amused by that. So it goes back a long ways. You know, they're talking about Pliny the Elder. You know, we can go to ancient Greece about using fermented milk products and perhaps fecal transplants. And in China, there was this talk of having yellow soup, which is basically you take feces, you mix it up, you let the thick parts settle, you take the kind of liquor from the top, and that can be used to treat a variety of illnesses. And it's kind of fun, I don't know. If you ever see yellow soup on the menu, I don't know. You can decide what you want to do with that. Really, the modern age of FMT came from our surgical colleagues in 1958. So after people started using antibiotics, they noticed that there was this pseudomembranous enterocolitis that could arise. And actually, it's interesting to read this article because a lot of the stuff that's said here, we're 60 years on, we're still sort of saying the same things. We assume that it has something to do with antibiotics, adjusting the microbiota, and so they had a case series of giving basically fecal enemas to rescue these patients that would normally have had to have their colon taken out. And of course, a lot of people are very familiar when this paper came out. We're going to hear updates to this. Our colleagues are going to talk about, really much more. This is based on a total of 16 patients that everyone, if you just take this paper at its face value, that's the reason to use fecal transplant. But we have a lot of other data that we'll hear about using feces to treat recurrent C. difficile. But how does this work? What's going on? So I'm going to take a somewhat older paper from my lab. This is Anna Seekatz, and as of January, she'll be an assistant professor at Clemson, continuing to work on the role of the microbiota in C. difficile infection. But when she started as a postdoc, we actually had some fecal specimens that we'd gotten from a number of investigators in Minnesota who actually had been treating patients with fecal transplantation for a number of years for recurrent C. difficile. But they saw me at a meeting and they wanted to say, well, what does this do to the microbiota? And so Johann Bakken, the former president of the Infectious Disease Society of America, Charles Gessert, who's now retired but has done a lot of fecal transplantation, asked us, what do you need? I said, well, I want the fecal specimens before you transplant the patients and after you transplant, and I also wanted the donors. Okay? And these were patients who had a lot of C. diff. This is the time that they got their fecal transplantation, and the circles with the positive, these are times they had positive tests for C. difficile, and then the colors are the various treatments that they had had with regards to antibiotics to try to treat C. diff. These patients had a lot of recurrence up to the time they had their fecal transplantation. And interestingly enough, they did what we're not supposed to do. They tested for cure, so some people were still positive. But of these patients, all but 2 responded to the initial FMT. Some of them had an additional FMT, and they subsequently responded there. But I'm not telling you this because FMT works for C. difficile, but this is what we did. This is kind of to show a little bit of the example of one of the many, many techniques we have to look at the microbiota. And this is sequencing amplicons of the 16S gene that encodes for the small subunit of the RNA, of the ribosomal RNA. Because it's conserved in life, you can have kind of near universal or basically group-specific broad-range PCR. And because of these stem loop structures, there's variability. We use these sort of, people refer to them as barcodes for specific bacteria. This is how we can kind of get an idea of who might be present. Not what they're doing, not what their functional capacity, but what organisms might be there. And you read microbiome papers and you see all of these different kinds of analyses that people are doing either for this or metagenomic sequencing. You hear about all these diversity indices and your eyes kind of glaze over and you go, what do you do with all these data? But I want to take you through some of these just to show that it's not rocket science. One of the simple things you can do is you can just try to classify what organisms might be present. So all the patients here are organized in that you have their pre-FMT sample, the post-FMT sample, and when we got it, a couple of them we missed it, what does the donor look like? Okay? And who cares what the organisms are being classified? Because sometimes you can actually get fooled. For example, C. difficile gets classified as Clostridium group 11. you know, and if you're not familiar that it might be in there, who cares? But then you kind of go, oh, wait, that's C. difficile itself. But if you just look at the communities, let's look at patient number 1. You see that pre and post, doesn't matter what they are, the composition is quite different. And interestingly enough, the post looks more like the donor. Okay? And this is 2 weeks after transplantation. And you can see this over and over again throughout here. So this is one way to look at things. This is kind of simple. You only have a handful of patients. This is okay. But what if you have a study with 1,000 patients? Seeing all these stacked bar charts might, you know, it's hard to make sense of it. What are you going to do with it? Well, one of the things you can do is you can let the data speak for themselves. Now, these are all the different types of bacteria based on the 16S arranged here, the kind of clustered taxonomically. But now we're looking at the communities, and we're using one of these various clustering techniques to see, okay, all of the samples, how do they cluster? Which samples are more similar to the other? And what you see, there's 2 main groups here. And even if you just look from afar, and you can notice that, hey, there seem to be fewer bacteria in this left-hand cluster than there is in the right-hand cluster. Okay? This is more diverse, this is less diverse. And you can even look that this has a lot of things related to E. coli over here. Not C. difficile, related to E. coli. That's something that we see over and over again. And then if you look to see what the samples were, we find out that the pre-FMT samples are in this low diversity group. And then all the donors, and most, but not all, of the post in green FMT samples, they're also over here in this diversity group. I told you that 2 patients didn't respond. When I saw this, I said, oh, Anna, please tell me that these 2 samples were from the 2 patients who didn't respond. And she goes, no, that's not true. So you can't use, as much as you can get broad generalities from looking at groups of patients, perhaps we don't have enough resolution, and maybe it's this technique, to be able to look at an individual fecal specimen from an individual patient and make any sort of predictions at this time, okay? That's the lesson there. One last thing is we kind of showed that this idea of lower diversity. This is actually the first paper I published on C. diff back in 2008. And again, we were just kind of looking. I was just learning how to use these techniques. And yeah, patients with recurrent disease had lower diversity than patients who had an initial episode that responded or healthy controls, okay? And that can be seen, again, when you treat these patients, you go from pre-FMT, and it doesn't matter what kind of diversity index, you don't have to worry about the details here, but the pre and the post, you basically increase it. You don't get quite to where the donors are, but you, in general, you increase the diversity. So diversity in and of itself doesn't predict things, but it's sort of it's associated with a more healthy microbiota. But I think we have to go down to the details of really who's there and what are they doing. But how do we study what's going on? Okay, so you're making these observations. How do we get at mechanism? Because if we're going to come up with drugs, we need to know what mechanism. So we do have model systems. The hamster is one model system. Mice are the other. And the mouse work actually around the time, whoops, sorry, this is blurry, don't worry about it. In 2008, Karen Kelly and his group in Boston revisited the mouse model and showed, and that's actually nice that it's blurry. It doesn't matter what antibiotics you're giving. You can give a whole set of antibiotics here. And then if you infect with C. difficile, you can take a mouse that has a normal colon and you can create C. difficile. You get a lot of edema, a lot of destruction of the epithelium. And we've played with this for about the past 10 years in a lot of different ways. We can model recurrence. We can model varying severity, depending on the microbiota, varying severity, based on host factors, varying severity, based on the C. difficile strain. And we actually have a systems biology grant where we're trying to look at all these. So look at, can we get an idea of what the host and the microbiota are doing specifically to try to interfere with C. difficile? And there are a lot of potential mechanisms here. And let's go over one of them. And this came from Casey Terrio from when she was a postdoc in my lab. She's now an assistant professor at NC State. She's now in Atlanta, hiding from the storm. She was actually wanting to look at, how do we look at functions? Well, let's look at the metabolites. I mentioned the microbiome. Let's look at the metabolites. What could be going on in C. difficile infection? And she used one of the models where you can take mice with a normal microbiota. She used a single drug at this time, cefoperazone. The animals become susceptible to C. diff. They develop very bad disease. And from work from another postdoc showed that after giving this antibiotic, if you take them off the antibiotics, keep them on sterile food and water, 6 weeks later, their microbiota goes back to a different state. Not the original state, but Casey showed that this secondary state is still resistant to C. difficile infection. So what Casey did is first she looked at who's there, and she showed that when you become susceptible, again, these are the microbes along here, clustering based on the types of organisms that are present in the community. The susceptible state is quite different than the animals here and here that never saw antibiotics, either at right away or 8 weeks later. Their microbiota is pretty stable. But this altered community had a different community structure. The population of organisms, the community was different, even though it had the same function, that is resistance to C. difficile. And when she looked at a lot of metabolites, she looked at thousands of metabolites, she kind of focused in on bile acids, and she saw that regardless of what the community looked like, the panel of bile acids seemed to be very similar in all the animals that were resistant versus those that were susceptible. And what might this have to do? I told you that C. difficile comes in as spores. Certain of the bile acids, in particular the conjugated bile acids, the ones that are secreted in our liver, are very good at triggering sporulation of C. difficile, where other forms of bile acids, such as deoxycholate, were actually very toxic to vegetative C. difficile. And what's important here, this is the idea of co-metabolism. Sure, our liver has these glycine and taurine conjugated bile acids, but there are microbes that will take off through hydrolysis, take off those amino acids, and there's still other microbes that will do these conversions like 7-dehydroxylation that can produce these toxic, at least toxic to C. difficile, toxic bile acid specimens. So that you assume that if you disrupt the microbiota-mediated metabolism of bile acids, you might change your susceptibility to C. diff. And Joe Sorg actually posited this about 8 years ago when he was looking at this particular organism, C. scindens, that was able to take bile acids and convert it to deoxycholate. And actually, Eric Paymer, through a separate set of experiments, came across the same thing a number of years later and showed that this particular organism, Clostridium scindens, because of its 7-dehydroxylase assay, in an experimental model, could restore bile acid-mediated resistance to C. difficile in a mouse model. So again, this idea that it's the host and the microbes working together. Final story I want to tell you is just, it's not all about bile acids. So my friend and colleague at Michigan, Pat Schloss, two of our grad students were working together, Matthew Genier in Pat's lab and John C. Leslie in my lab. We're trying to look a little bit more at how altering the structure and metabolism function of the microbiome could actually promote sustained colonization by C. diff, or actually just make C. difficile change its physiology. Well, what do we mean by that? Well, what Matt did is he took 3 different antibiotic regimens. He used cefoperazone, which he used before, streptomycin, and clindamycin. He took genetically identical, and in this case, mice with the exact same microbiota. I actually have a breeding colony of wild-type mice, and people ask me, why do I have that? You can always buy them. I said, but you can't always buy the same microbiota, which is why I've been breeding these animals for almost 20 years now. What he did is he created 3 different environments for C. difficile by giving 3 different antibiotic treatments to these mice. And then to look to see what C. difficile was doing, they did RNA-seq, basically purified the C. difficile right from the community and basically looked at the transcriptional response of the pathogen to see how is it behaving in mice treated with cefoperazone versus mice treated with streptomycin versus clindamycin. Then he also constructed some metabolic networks based on, computationally based on the genome of the infecting strain and the response that he saw. And so here's the transcriptional response. So basically what he did is he looked at all of the different things that C. difficile was doing in terms of transcription under the various conditions, clindamycin treatment, cefoperazone treatment, streptomycin treatment. And he found that C. difficile actually had a different transcriptional response depending on which kind of environment it was. It wasn't behaving the same. Certain genes were turned up in cefoperazone-treated mice versus clindamycin-treated mice versus streptomycin. And he focused on the fact that a lot of them had to do with core metabolism of the pathogen. Sugars they're using, monosaccharides, disaccharides, proteins they could do, transporters for nutrients. So when he did this, he was able to kind of predict, modeling the metabolic network, what kinds of sugars would C. difficile utilize under the different conditions. Sort of he used the shared under all conditions, but then he looked specifically for strep, cephalosporin, or clinda. And he saw that different sugars were preferentially going to be used by the pathogen under these settings. He tested to make sure that in vitro that C. diff could utilize all these as a so-called carbon source, and he did, and that was true. But then he also used untargeted metabolomics, and he showed that, yeah, under the different situations, different of the sugars were being not only generated in the untreated uninfected animals, when he infected with C. difficile, those sugars would drop, suggesting, not directly testing, but indirectly suggesting that the C. difficile was utilizing those sugars. So this is how we can get an idea at how changing the metabolic landscape present in the gut can influence not only the host, not only the indigenous microbes, but a potential pathogen. Where do we go? You know, there's a lot of things here. There's a lot of things that are going on. We have to consider not just host, not just pathogen, but now we have to really consider these hundreds, thousands of different species of microbes present in the gut in all sorts of setting of health and disease. So, you know, I hope that some of the work that I'm showing you is we're just trying to go away from association. Oh, this microbiota is different in patients with disease versus patients without. Can we begin to get a causation? Can we begin to understand how this altered, and people sometimes use the word dysbiotic, microbiota, what's different in terms of the function of that community? And therefore, could we then try to intentionally manipulate the microbiota to, quote unquote, improve health, prevent disease, treat disease? What's the FDA statement? Yeah, we all know it. Actually, all the things I saw this morning when I had CNN on, the FDA has not evaluated these statements. These are not intended to do all these things. It's kind of funny. So what could the future look like? You know, this is something that I would like, perhaps. You know, we talk about precision medicine. You know, there is this, all of us that NIH has started, that they're going to try to get, what is, I think it's 1 million? I think it's 1 million individuals. They're going to look at their genomes to try to predict from their genomes, how is the host going to respond to drugs? How susceptible are they to developing certain types of diseases? What happens if they're in different environments? You know, can we predict adverse reactions to drugs, like I was saying with digoxin? But I would like to say that maybe there should also be a microbe-censored microbiota, focused precision medicine. Interestingly enough, I hope no one's here from the NIH who's responsible for this. They're probably listening. Microbes are not a part of this. And I think that was a conscious decision for whatever reason, and that's fine. But I would like to think that perhaps we also need to consider what the microbiota would do. Because if we assess the microbiota, there might be deleterious organisms, there might be beneficial organisms. The things that we predict from the host genome might be influenced by the microbes that are there. We already saw that with a couple examples I gave with immunotherapy, with response to relatively simple small molecule drugs. And perhaps if we can do all of this data analysis of both the microbe and the host, then we can come up with customized therapy that's based on genetics and predisposition. So I hope I gave you the proper overview of this idea that this indigenous microbiota is part of a balanced ecosystem, that health reflects the balance between us and the microbes that live in and on us. We have evidence from the past almost 20 years now that disturbances in this balance can lead to the pathogenesis of multiple conditions. We haven't talked about autism. We haven't talked about inflammatory bowel disease. We haven't talked about depression. We haven't talked about alopecia areata. There's a number of things where there are associations between the microbiota. But I would really like to stress that it's going to take teams of people working together to understand the dynamics of the system, what is the function of the system, and most importantly for the clinicians in the room, how are we going to able to manipulate this complex system to prevent or treat diseases. So, did I stay on time, Paul? Okay. I'd like to thank a lot of my collaborators. Again, this is team science. This is just a small handful of the people I've worked with at a number of institutions. So, and we come from all sorts of backgrounds: bacterial pathogenesis, immunology, clinical microbiology, machine learning, computer engineering, etc., microfluidics, of course, all the people in the lab who actually do work. And I'd like to thank NIAID, also NIDDK from previous money for previous awards to study the microbiome in health and disease. I'd be happy to take any questions at this point. Thanks.
We have plenty of time for questions. So I just want to ask that if you have a question, you come to the microphone and give your name and affiliation prior to your question.
Have time. I know a lot of the questions will probably, yeah, if you can come to the microphone. I know a lot of these questions might be best addressed by some of the subsequent speakers. So if a question comes up and one of the speakers says, I'll get to that, the speaker could raise their hand and say, I'll get to that, and I won't try to flail around and answer. Go ahead.
Hi, I'm Christian Lewis from the Peggy Lewis Foundation. In addition to antibiotics and stuff that we put into our systems, have you guys looked at anything in terms of how the environment itself might be impacting us, like the overuse of antimicrobial soaps and different things that we're kind of putting in and on our bodies?
Yeah.
Because I've always wondered about that.
Yeah, so the question is about, how does the environment outside of drugs that we use, in particular antibiotics, antibiotic residues in food, there have been a number of papers that have tried to associate that. Triclosan, there's a lot of studies on triclosan and what that may do to the microbiota. And you also raise the idea of there is a whole field of so-called the microbiome of the built environment. There are people who are looking at how microbes that we could get exposed to in our cars, in our houses, in restaurants, in healthcare systems can influence us as well, and trying to suss that out. So the long and short is almost any time you do kind of a study to compare two groups, you'll find that there are differences in the microbiota, but a lot of times we don't know if there's a significance there, because many times we're not necessarily looking at function and we're not looking at how it impacts health directly. So.
Thank you very much for your talk, Dr. Young.
Very well.
Jessica Eunice, the IPA, Probiotic Association. My question is about sex dimorphism. I really appreciate the work you guys are doing with the mouse models, but can you maybe make a comment on that, especially in light of the topic of the workshop?
Right. So sexual dimorphism in microbiota responses is something that should be looked at. I know my program officer is here. We write a section on that right now. And we make sure that we always look, and in our studies, we do stratify by sex. And in our studies, and probably because we're giving antibiotics, which really overwhelm the microbiota, we haven't seen any sex differences in responses. But I know there are papers when they're using more subtle perturbations in the microbiota where there are distinct, in terms of the response of the microbiota and also the response of the whole system. In other words, whatever the health outcomes are based on sex. And so that's something we have to keep in mind. But in the Human Microbiome Project, we did not see, other than the obvious that the vaginal microbiota is only seen in women, for example, we didn't see anything, say, with skin and gut and other things with sexual differences. Again, looking at a very crude, high level by 16S in metagenomics. We haven't seen it. But it's something that needs to be kept in mind. Yeah, thanks.
You talk about the...
Could you give your name quickly and your association?
Human way. You talk about the cardiac glycoside, the Jackson metabolism microbiome.
Yep.
The metabolite is still effective. It's the more broad the spectrum of action.
I have to remember the 83 paper. I believe that actually some of the metabolites were no longer active, but were still toxic in some ways. They had less antiarrhythmic, and I can't remember of which ones were inactivating versus the primary response or not. But that's something that's seen over and over. In some cases, you create new compounds that have differing activities. In some cases, you create compounds that are just different structurally, but maintain the parent activity. And that's something that we've seen over, like, for example, with the bile acids, you can shift function with some of these. But specifically with the digoxin, I'd have to go back to that paper to figure out which of the metabolites still had toxicity versus therapeutic effect on them. But it was looked at in both of those studies, so it's in there. Other questions? Comments? Have we solved it all? Are we ready to go out and treat patients, send their fecal specimens or whatever specimens to whatever diagnostic lab? I think there's something very important. I mentioned the two papers in my introduction that came out from Aaron Elanoff's group. They looked at some things that were very important, things that we'd looked at, but finally they published. We use feces a lot to kind of survey the microbiota. They also did colonoscopy, both prepped and unprepped. And there are differences between the microbiota at the mucosal surface, the microbiota in the lumen, and those are quite different from feces. So we have to also figure out how are we going to most properly assess a patient's microbiota. I think some of the things in feces, that most of the things in the GI tract eventually end up in feces, but the relative abundance that you find is going to be altered in feces as opposed to what it might be more proximal in the GI tract. And I had gotten into a number of, shall we say, heated comments about people who talked about the relative efficacy of stool, because I said, yeah, it may or may not. But I think now that these have been published, I think people will be a little bit more circumspect that stool is not the only analyte that we can look at. Or if we look at it, there's certain caveats that we have to have. We look at feces as a marker for what's going on as far as the microbiota. Yes?
I'm Yuli First Allen Capital. I wonder if what thoughts you've given and what research you might be aware of, just generally speaking, going on identifying the impact from maybe changes in soil, given that affects our food supplies, right. And one would expect that maybe there's some changes going on there.
Yeah, so it's interesting. I think his name was on there. Tom Schmidt is the person who taught me how to do microbiota when I was up at MSU, and he actually, his research at the time was looking at different agricultural practices: till, no-till, amended, whether or not they had fertilizer in the soil, on the effect of the microbiota of the soil itself, and how actually that could change the flux of greenhouse gases through those soils. And this was done at the Kellogg Biological Station up in Michigan, where they had different plots all in the same area where they had these different treatments. And he saw that it was quite dramatic effects on the microbes that are present in the soil and their ability to take and fix nitrogen, and basically to take CO2 from the atmosphere and fix it back into plant material. And so that's where I actually interested him. I said, hey, would you want to look at another community? I actually have a different community of microbes. I'm very interested in their function. And that's how we started looking at that. So no one's tied together those changes that you see clearly in the soil with what happens to people who eat crops grown under those different regimens. That actually hasn't been done. I don't know how much I would expect that to change, but really this idea of an integrated earth microbiome, that it's not just the ones in us, for example, the microbiota in cows and how much methane they can produce. You can actually have cows that produce more or less methane, speaking of greenhouse gases. So everything is interrelated in this world, and the microbes may be actually the link between a lot of them. So these people have been looking at soil microbes for like 30 years, and there was an article published in the New York Times about 5 years ago that I showed to Tom, where it said, it was talking about people who were looking at crop microbiome. And it says, taking clues from people who are looking at the human microbiome, soil scientists are now, and I said, hey, here's a little bit of revisionist history. I said, I learned from you, but supposedly you're learning from me now. So, that's kind of, you have to kind of take that broad view. I think I only bring up that anecdote, that it's important that and that's why I was trained, doing reductionist, mechanistic-based science. But some of these questions are so complex that you sometimes have to back out a little bit, try to look at the big picture, move back down, zoom in on mechanism. So that's the approach. Yep.
Sam Forey, NIST. I wanted to ask about sampling recommendations in the context of human samples. I know with mouse models where they produce fecal material more often, we can track and see diurnal cycles. In humans, where people often have a single bowel movement a day, it's much harder to pull that out. And I'm wondering what you do in the context of, as a clinician, in your research, how you go about acquiring fecal samples and what the best practices are to try to mask that or control for it.
Yeah, that's actually a very important question when you're sampling. And I already brought up that stool only gives you one kind of view. So invasive sampling gives you a different view. We've actually sampled the upper GI tract using an FDA approved device that actually has 4 lumens, 2 meters long, goes down, and we're looking at drug dissolution studies, and we've looked at the microbiota through there. And over time, in these individuals who are fasted and fed, you see all of these changes. We didn't monitor them long enough to look at diurnal variation in a fasting subject, but these are all these things that you have to take into account. You want to sample as much as possible, For example, one of the areas that a number of investigators, and we'll hear more about necrotizing enterocolitis, is you collect all the feces that comes out of an infant if you can and see how that changes. But know that it dwells there for a while. You're right. Some people only have one bowel movement a day. I mentioned Stanley Falco. He had this saying that one man's constipation is another man's diarrhea, right? Is one sample a day enough on a person who has 4 bowel movements a day or only has a bowel movement every 3 days? These are the considerations that we have, and we don't have best practices. And I know other people from NIST have been at some of these conversations, and it's kind of daunting to figure out, how do you standardize this? And I think a couple more questions.
Earl and Deal Nyad. So, Vince, I was struck by the fact you were saying the low diversity was more susceptible and the higher diversity was more resistant.
Mm-hmm.
But for other body sites, say, for example, in the human microbiome, you see a project, you saw the opposite. Like in the vagina, less diversity tends to be more resistant, and more diversity seems to be more susceptible to bacterial vaginosis. I was wondering how you think about, when you think about different body sites.
Right. And that's why, even though I've been guilty of putting the word diversity in my early papers there, is I realized that diversity is just a marker for how many different kinds of organisms that you might have there. And if you have the wrong organism there, or you're missing the right organism, it doesn't matter how diverse you are or not. And it does vary from site to site. As you said, in bacterial vaginosis, you actually have much higher diversity. In the healthy vaginal tract, which is generally dominated with lactobacillus, but not in all healthy individuals, they tend to have that, and that's lower diversity. I published a paper with John LaPuma on the cystic fibrosis lung, and actually increased diversity was quote-unquote protective early on, and when you just have nothing but Pseudomonas or Burkholderia or staph later on, and maybe that's because you've had a lot of antibiotics, people say that was bad. Again, we had to be careful what we said, because we're not going to stop treating our cystic fibrosis patients with antibiotics, because that's what's extended their lifespan by decades. But maybe we do have unintended consequences that over time they're going to be more susceptible. We have about till 10, right? We have time for one more question. Yeah.
Thank you for your talk. Noah Voriatis from GenBiome Consulting. Mm-hmm. Going back to the question about stool collection, I think there's still a lot of open questions in regards to what is the appropriate way to collect the stool, meaning is a swab from a tissue paper sufficient? Can you take an aliquot with a scoop, or do you need to essentially take the whole stool, homogenize, and then sample from there? I wasn't sure if you have any best practices, or within the community that you're in, if you can provide any insights.
Sure. As far as sampling, those are important studies to do and boring papers to convince your graduate student to write. But, for example, we looked at that, and we did a study where we compared feces to swab in a number of patients, and we showed that the rectal swab taken matched the fecal specimen pretty closely for all intents and purposes what we're doing. And I think what happens is then you say, oh, what's the best storage technique? What's the best extraction technique? What's the best way to do your amplification? Which is the right Taq polymerase? Which is, And I think what we have to come up with is that we're always going to have biases. And what we need to do, and I kind of tell my students, in spite of all the biases we have technically, appreciate that they might be there, do things the same way, and design your experiments very carefully so that you can get an answer despite what the biases might be. And then try to test it in another way. In other words, go to a germ-free mouse after you've sampled. Go and try to actually do interventional studies. Go into bioreactors. Go into organoids to try to figure out that the answer isn't just from sequencing. I hope no one's from NHGRA, but the answer is not just from sequencing. All right, thank you very much, folks.
And the next two speakers are from FDA. And the first speaker I'd like to introduce is Bob Durkin. And Bob is the Deputy Director, Office of Dietary Supplement Programs at FDA's Center for Food Safety and Applied Nutrition, or CFSAN. And his office is the agency lead for the regulation of dietary supplements. And today, Bob will speak about how the agency approaches the regulation of products labeled as dietary supplements.
Good morning. I very much appreciate being asked to speak here today. As I was introduced, I am the Deputy Director of the Office of Dietary Supplement Programs at FDA Center for Food Safety and Applied Nutrition. I think an obvious question at the forefront is if we're here at a workshop to talk about the use of microbiome products as drugs, why do they invite a person from foods to speak? I think that's a good question, and I think it should be addressed at the front of my presentation. Probiotics, live microbiobules, you know what I'm trying to say, are a very quickly growing segment of the products labeled as dietary supplements. They're approaching coming into our marketplace very rapidly. They're taking a lot of market share, and they have FDA's attention when they're regulated as a dietary supplement. Products that are regulated as dietary supplements or dietary ingredients are really regulated like nothing else at the Food and Drug Administration. There are a lot of similarities, a lot of crossovers, but even when you think you hear something that sounds the same, I can almost assure you, I don't want to speak in absolutes, but it's very likely that there's some nuance or tweak, a difference between things that sound the same, the way they're handled in a drug or a biologic or a conventional food compared to a dietary supplement. That said, I thought the best thing for me to, nope, I didn't do that right. How do I get to the next slide? Okay, there we go. Did it. So that said, I thought I would start off. I considered the folks that would be in the room today, the folks that would be interested in this. And on one hand, I think there's some people that are very well versed in the regulation of dietary supplements. I see some familiar faces in the room. On the other hand, I thought there were some folks that might be in the room that are a little uneducated or uninformed about how dietary supplements are regulated. And a basic 101 about that might be a good place for us to start our conversation. This slide here shows some very different ways that dietary supplements can be presented on the market. We'll get into some of that a little bit later. Am I supposed to find out something in particular? Okay. So we'll start with the definition of a dietary supplement as found in the DSHEA, the Dietary Supplement Health Education Act. The DSHEA states that a dietary supplement is a product that is simply intended to supplement the diet. That can be translated to mean that it cannot be a conventional food or intended to be the entire substance of an entire meal. A dietary supplement must be intended for ingestion. It cannot be sublingual, topical, injected. Those products fall out of the definition of a dietary supplement. Dietary supplement must also contain a dietary ingredient. There's a list found in 201FF1, a list of ingredients that can qualify as dietary ingredients: vitamin, mineral, herb, other botanical, amino acid, dietary substance for use by man to supplement the diet by increasing the total dietary intake, or a concentrate, metabolite, constituent, extract, or combination of any of the above. Oh, you're going to come here and show me. So happy he was unsuccessful. You have no idea what a relief that was. Oof. One more. Thank you very much. Again, the definition of a dietary supplement. It's an article. This is an exclusion from the definition of a dietary supplement. It's found in 201FF3B. Essentially, it says that an article that was the subject of an IND, an approved IND, or an ANDA, for which there was a significant clinical investigations that were made public, is excluded from the definition of a dietary supplement. Basically, you cannot do research on an ingredient and then someone in the dietary supplement industry come in and take it out from underneath you. This was meant to preserve the incentive for development under the rubric of drugs. Old versus new dietary ingredients. The new dietary ingredient or NDI notification requirement is for those products that contain a new dietary ingredient. New dietary ingredient is an ingredient that was not marketed prior to October 5th, 1994. This NDIN process is basically the only pre-market opportunity that FDA has to look at a dietary ingredient before it comes on the market. Again, it's a notification process, not an approval process. The manufacturer or distributor of the dietary ingredient to be contained in a product labeled as a dietary supplement has to notify FDA of their intent to go to market 75 days prior to going to market. During the 75-day period, FDA will evaluate the firm's basis for thinking that their ingredient is reasonably expected to be safe to go to market under the conditions of use, the labeled conditions of use. FDA responds to new dietary ingredient notification with essentially two types of responses. A response without objections and a response with objections. A response without objections is known as a good day letter. It means go to market. We don't have a problem with your product. A response with objections can come in a few different flavors. We can disagree with your basis for thinking your product is reasonably expected to be safe. We can disagree with how you identified your product. You didn't tell us what it was. We can find some shortcomings in your manufacturing process. Maybe you didn't show us that it was going to not be contaminated or have some other follow-on constituents or components that would be dangerous. We can also send you a letter saying that your ingredient is not a dietary ingredient or that you didn't even follow the directions for filing a complete notification. New dietary ingredient notifications. A proper notification must include the name and address of the manufacturer or distributor that is introducing the NDI into commerce, identity information on the ingredients so we know what we're talking about, information on the dietary supplement that contains the ingredient, conditions of use, and safety information. The safety information can be based on a history of use or other studies that demonstrate the ingredient will reasonably be expected to be safe, or a combination. In other words, you can show historically where your ingredient's been on the market and it's been used safely, or you can show us scientific literature, preclinical, clinical studies, or you can show us the combination of all the above. New dietary ingredient notifications. While the requirement's been in place for about 20 years, we've received about less than 1,100 independent NDI notifications, representing about 720, 750 individual ingredients. This shows anecdotally maybe that there's an underreporting going on in the industry, and that might be something you want to address before you get a visit from FDA. Current good manufacturing processes. This is important. I thought it was something that should be mentioned here today in regards to dietary supplements. A large part of FDA's post-market regulation of dietary supplements is based on the good manufacturing process regulations. FDA published the final rule for good manufacturing processes in June of 2007. This rule is found in 21 CFR Part 111. That's different than drugs. Drugs are found in 211. That's one of our differences there. The GMP, not just where they're at, but also the substance of what the GMPs represent. GMP regulations are an important tool to ensure that dietary supplements are produced consistently and at high quality. Maybe not as high of a quality as someone who's familiar with drug GMPs would think, but certainly more than conventional foods. It's sort of in between. The regulation has an emphasis on production and process controls, building quality into the product, as well as requirements for the testing at the raw material and finished product stage. This is an extensive regulation, maybe relative to conventional food, but not so much to drugs or biologics. An extensive regulation that covers all aspects of manufacturing, from setting up a facility and establishing personnel through product design, production, and testing to records and record keeping. A little more on GMPs. They're applicable to all firms to various degrees who are involved in the manufacturing, packaging, labeling, or holding of dietary supplements, both domestic and foreign. FDA investigators confirm GMP compliance through a series of investigations. We conduct hundreds per year, somewhere between 500 and 700, split between domestic and international. Non-compliance with the regulations can result in FDA action. Another interesting aspect of products labeled as dietary supplements are their labeling requirements. In addition to the previously mentioned manufacturing requirements, dietary supplements also have labeling requirements. As dietary supplements are a category of food, they must follow the food regulations found in 21 CFR 101. A few requirements that are specific to products labeled as dietary supplements relative to other foods would be that they must be labeled as dietary supplements. They must actually use a statement that describes them as a supplement, as a dietary supplement. It might say dietary supplement. It might say probiotic supplement. It might say calcium supplement. It has to have something on it to describe it as a supplement, to show the intent of the person putting it in commerce. As with foods, dietary supplements must list all ingredients, but the ingredients must be formatted instead of a nutrient facts label, they must be in a supplement facts label. Additionally, dietary supplement labels must contain the name and location of the manufacturer or distributor and have contact information, a U.S. phone number or address to which consumers or healthcare providers can notify the firm of adverse events. A little bit more about supplement labeling. In addition to the required aspects of the label, dietary supplements are afforded 3 types of claims they can make regarding their products. The first of these claims would be nutrient content claims. An example of this would be a product that is high in calcium or low in sodium. Dietary supplements can make structure function claims regarding the effect of the product on the structure or function of a body. An example might be calcium helps build strong bones. I'll talk more about these in a minute. Finally, a dietary supplement can make some authorized health claims or qualified health claims. These are actually spelled out in regulations. They can be found on our website. An example might be regarding calcium or vitamin D, reducing the risk of something like osteoporosis. A little more on structure function claims. Structure function claim is intended to describe the role of a nutrient or dietary ingredient on the structure or function of the human body. DeShay created an exception to the drug definition that authorized dietary supplements to bear these structure function claims without being regulated as a drug. Dietary supplements, with maybe just 1 or 2 other exceptions, are the only type of food that can make a structure function claim. Any other food that makes a structure function claim has now put itself in the unapproved new drug box. This is a unique exception for dietary supplements that is separate from most foods, cosmetics, and such. The exception from the drug definition applies only if the claims are made in accordance with the information found at Section 403(r)(6) of the Act. Firms tend to get in trouble when they make a claim that is intended to be a disease claim. A slide here about disease claims. Disease claims are hard. They can be in a gray area. Context is critical. You have to take the label and the labeling, the totality of the circumstances. A good example might be an EKG symbol on a label may in itself not be a drug claim. But if they then make a statement about cardiac health, the two together may be considered a drug claim. Again, some guidance for industry can be found online, structure function claims, small entity compliance guide. No claim is ever likely to be absolutely violative or absolutely okay. Again, it's all a matter of the circumstances and an evaluation. A little bit about adverse event reporting. FDA's post-market surveillance of products labeled as dietary supplements includes adverse event reporting. This is a result of the SEER adverse event law, which took place in 2006. Dietary supplements must submit serious adverse events to FDA for review. The reporting system works through FDA's MedWatch program, and submissions can be received through an electronic portal, email, phone, call, letters, email. While manufacturers are required to submit these reports, consumers and healthcare providers can do it on a voluntary basis. If a manufacturer receives an adverse event and determines it to be serious, they have to report it to the FDA within 15 days and follow up on that specific event for a year. A little more on adverse events. Once the reports are entered in the MedWatch, the dietary supplement specific adverse event reports are entered into our CARE system, which stands for the CFSAN Adverse Event Reporting System. ODSP has medical doctors that review every single adverse event. They'll make a determination if there's association or causation. They'll look at the trees, they'll look at the forest from all different perspectives. They'll see if we have a problem with a product or maybe even an ingredient. If there's signal of a risk to the public health, we do what's appropriate. We do other investigations. We'll call up the person that provided us with the adverse event. We'll investigate, and if required, we'll do what we need to do to protect the public health through enforcement actions. Just a little bit here about what we deal with when we talk about the dietary supplement market. When DeShay was passed on October 15, or enacted on October 15th, 1994, there were about 600 manufacturers with about 4,000 products worth approximately $4 billion. Today, there are over 7,000 registered facilities. There are over 75,000 independent SKUs for products labeled as dietary supplements. And external sources estimate that the industry is worth upwards of $40 billion. How does FDA approach this large, diverse, fractured industry? We try to regulate it. This is a basic pictogram or organizational chart for the FDA as a larger entity. You can see the Office of the Commissioner up top. Down bottom, you see what we call product centers. And you can see the Center for Food Safety and Applied Nutrition is the second from your left. You see the Center for Biologic Evaluations and Research is the fourth one in. We don't work with the folks directly that work for CBER. We're in different product centers. We know each other, we have relations, we have good communications, but we don't actually work together in the same building or even the same office structure. Just some other offices on here that impact the regulation of dietary supplements. You have the Office of Operations, that would basically be ORA, the inspectors, the boots on the ground. CBER, we work with CBER, we work with CDER, we work with the Office of Chief Counsel. This is the organization at CIFSAN. As I mentioned, CIFSAN is one of the product centers. You can see there are then offices within CIFSAN. Office of Dietary Supplement Programs is highlighted. We're one of about 12 product offices within the product center. So you say, what does FDA have to regulate this industry worth $40 billion? We have 26 people. Yeah. Now that's directly in ODSP. That's not including folks that we try to leverage in other offices such as ORA and OCC. ODSP wasn't always an office. We were once a division, part of ONLID's Office of Nutrition, Labeling, and Dietary Supplements. Back in December of 15, I think it was, or 16, we became an office. That elevation brought us up to the table. We're now a product office within a product center. It was meant to give us a higher profile and put us in a better position to ask for resources and to work with regulated industry. ODSP's program priorities are first and foremost to protect consumers, ensure product integrity, and help to promote informed decision making.
We've heard from the Center for Food Safety and Applied Nutrition, and now it's time to hear from the Center for Biological Evaluation and Research. And our next speaker is Sheila Strayer-Lesnick. And Sheila is a regulatory coordinator in the Division of Bacterial, Parasitic, and Allergenic Products, or DIBAPAP, as we say, in the Office of Vaccines Research and Review in the Center for Biologics Evaluation and Research. And DIBAPAP is the product review division, which is responsible for reviewing product information for regulatory submissions for a wide range of products, including bacterial and parasitic vaccines, allergenic products, live biotherapeutic products, or LBPs, FMT, or fecal microbiota for transplantation, and also the phage therapy products. And Sheila's presentation today will discuss the regulatory oversight and considerations for live microbiome products when they're used as drugs. A little different from the previous talk.
Thank you for that introduction. Let's see if I can manage this. So today, my talk will broadly cover, the first part of my talk will broadly cover the regulatory oversight for development of live microbiome-based biological products. And I'll be just briefly touching on the IND regulations and definitions and broadly cover the stages of review. And then the second part of my talk, what I hope to do is point out some additional considerations for clinical studies using live microbiome-based products. And I'll point out a few chemistry, manufacturing, and control information points, some CMC points for live biotherapeutic products, and a few points to consider for fecal microbiota for transplantation. So what is an IND? An IND is an investigational new drug application that, when in effect, exempts an investigational new drug from pre-marketing approval requirements. It also allows an investigational new drug to be lawfully shipped across state lines for the purpose of conducting a clinical study of that investigational new drug. The IND regulations require that human research studies be conducted under IND if the following conditions exist. The research involves a drug as defined in Section 201 of the Federal Food, Drug, and Cosmetic Act, The research is the clinical investigation as defined in the IND regulations, and the clinical investigation is not otherwise exempt from the IND requirements. And pertinent to our discussions today, I just want to point out that a biological product subject to licensure under Section 351 of the Public Health Service Act fits within the drug definition under the FD&C Act. And a few clarifying points about exemptions. What this means is that clinical investigations of drugs lawfully marketed in the United States are exempt from the IND requirement if certain criteria are met as listed in 21 CFR 312(2)(b)(1). And drugs are lawfully marketed if they have been approved under the following pathways: a new drug application, a biologics license application, an abbreviated new drug application, or an over-the-counter monograph. And just to point out here that conventional foods and dietary supplements are not lawfully marketed drugs, and therefore do not qualify for an exemption of the requirement of an IND as described above when they're studied for a drug use. So if not exempt, when is an IND needed? In general, the FDA regulations require the evaluation of a drug or biologic product in humans be conducted under IND. And a drug is defined in part as articles intended for use in the diagnosis, cure, mitigation, treatment, or prevention of disease, and articles other than food intended to affect the structure or function of the body. So the intended use then determines whether a product is a drug. And the question becomes, is the product in the study being investigated for a drug intended use? If the answer is no, then no IND is required. And if the answer is yes, then an IND is required. And this is true whether it's for commercial development or for research-only studies. For additional details, I'll refer you to our guidance from 2013 determining whether human research studies can be conducted without an IND. Who sponsors INDs? Big companies do, small companies, individual bench researchers, individual clinical investigators, and other government agencies. And FDA's primary objectives in reviewing an IND is to, 1, assure the safety and rights of subjects in all phases of the investigation, and in phases 2 and 3, to help assure that the quality of the scientific evaluation of drugs is adequate to permit an evaluation of the drug's effectiveness and safety. And this slide is just to give you, or to remind you really, of the typical phases of development for biological products under IND. They typically start as small phase 1 studies and then progress to larger phase 2 studies. Data generated from these phase 2 studies are then used to inform the design of the larger phase 3 efficacy studies. And then data from the phase 3 efficacy studies are then used to help support a biologics license application. And to obtain licensure, the applicant must demonstrate the following: that a particular product is safe, pure, and potent, and that the facility in which the biological product is manufactured, processed, packed, or held meets standard designs to assure that the biological product continues to be safe, pure, and potent. And a point I'd like to make here is that potency has long been interpreted to include effectiveness. And only those biologics that are demonstrated to be safe, pure, and potent, and that can be manufactured in a consistent manner will be licensed by FDA. And to date, the FDA has not approved a live microbiome-based product to prevent, treat, or cure a disease or condition in humans. So that covers the first part of my talk, and now I'd like to get into some additional considerations for clinical studies using live microbiome-based products, and I'll start with live biotherapeutics. In 2012, FDA published a guidance document discussing chemistry, manufacturing, and control information, or CMC information, to include in an IND application for early clinical trials with live biotherapeutic products. And an LBP is defined as a biological product that contains live organisms and is applicable to the prevention, treatment, or cure of a disease or condition of human beings. And a commercially available probiotic may fit the definition of an LBP depending on the intended use. And while commercially available probiotics are generally considered safe in healthy adults, safety issues may be critical in clinical trial populations compromised by specific health concerns or conditions. In recognizing the difficulty that sponsors had providing the CMC information required under 21 CFR 312.23, FDA revised the LBP guidance in 2016 for proposed trials in generally healthy subjects. And the updated guidance describes how for IND studies using commercially available LBPs such as probiotics, a waiver of the requirement for CMC may be granted if all of the four following conditions are met. 1, the LBP proposed for investigational use is lawfully marketed as a conventional food or dietary supplement. 2, the investigation does not involve a route of administration, dose, patient population, or other factor that significantly increases the risk or decreases the acceptability of risk associated with the use of the food or dietary supplement. 3, the investigation is not intended to support a marketing application of the LBP as a drug for human use or a biological product for human use. And 4, the investigation is otherwise conducted in compliance with the requirements for INDs. If the investigation meets all of these conditions, we ask that sponsors submit a waiver request documenting the above, a copy of the label, and a commitment to record the lot numbers and date of expiry. So therefore, INDs using commercially available LBPs, if the request for a waiver of the requirement for CMC is granted, then the label on a commercially available LBP will generally be sufficient to satisfy the CMC requirements for the IND application. If the waiver is not applicable or granted, then the sponsor needs to submit CMC information in their IND application. And we do recognize that specifically for commercially available LBPs, that the IND sponsor may not be the manufacturer. And in this case, the manufacturer and the IND sponsor can use the master file mechanism to provide confidential manufacturing information directly to FDA. And what we're looking for when reviewing CMC for INDs with live biotherapeutic products is sufficient information to assure the proper identification, quality, purity, and strength of the investigational new drug. And as product development proceeds, we ask that the sponsors submit amendments to the IND to supplement this initial CMC information. What does the CMC information look like? So the guidance then goes on to describe what to include. And that would be strain information as available, such as the name, the source, the strain and passage history, relevant genotype and phenotype, or full genomic sequence. We also ask to include an antibiotic resistance profile for clinically relevant antibiotics. Information on cell banking system, a description of drug substance and drug product manufacturing process, and stability data, and specifically to demonstrate that the product is stable for the duration of the treatment phase of the study. CMC information should also include manufacturing information about manufacturing controls and release testing, including potency testing, which is typically a measure of viable cells expressed in CFU. And for multi-strain products, we ask that as product development proceeds, the sponsor work on enumerating all strains in the final product. Potency testing can also include additional biochemical or physicochemical measurements thought to predict potency. Manufacturing controls and release testing should also include bioburden testing. And there, we want to see that the sponsor can demonstrate the absence of extraneous undesirable bacteria. And we've typically asked sponsors to perform bioburden testing per USP 61 and 62. But I do want to point out here that additional testing may be required depending on the intended population and other organisms manipulated in the same facility. And just a slide here on CGMP again. Current good manufacturing practices for drugs and biologics follow 21 CFR 210 and 211. And basically what it states here is that it assures that a drug is safe and has the identity and strength and meets the quality and purity characteristics that it purports or is represented to possess. And as described in our guidance here for CGMP for phase 1 investigational drugs, FDA recognizes that the extent of manufacturing controls differs not only between investigational and commercial manufacture, but also among the various phases of clinical trial. And now on to a few points about fecal microbiota for transplantation. This slide is a summary of the history of FMT guidance from the FDA. And it starts in May 2013, where FDA and NIH held a joint public workshop. This was attended by clinicians, bench researchers, members of the public, and government employees. And at that workshop, FDA noted that the use of FMT and clinical studies to evaluate its safety and effectiveness are subject to regulation by FDA. Recognizing concerns from healthcare providers at the time that applying IND requirements would make FMT unavailable for individuals with C. diff infections unresponsive to standard therapies, FDA published a guidance document for immediate implementation in July 2013. And this guidance explains that FDA intends to exercise enforcement discretion regarding the IND requirements for use of FMT to treat C. difficile infection not responding to standard therapies. Enforcement discretion does not extend to other uses of FMT. Since then, FDA has published 2 draft guidance documents, one in March 2014, where FDA clarified that they expect to exercise enforcement discretion only if the donor is known to the doctor or the patient. We received many comments, and they were all considered. And in response, we then published a revised draft guidance in 2016. And in this draft guidance, FDA clarified that they intend to exercise enforcement discretion only if stool for FMT is not obtained from stool banks. And I know some here in the audience are really hoping for an update to this guidance, but I don't have new information to share with you today. But what I can say is that we are considering all the comments that we've received to date. And as we move forward, I'd like to point out a few safety considerations for FMT. We can address safety by adequate donor screening and establishing appropriate donor screening protocols for the intended population. We can also test stool, but we have continued questions about the sensitivity of available stool tests and their ability to detect pathogens present in low numbers. Questions are also arise in terms of longer-term safety. What are the potential longer-term effects of the transferred microbiota on the recipient? With regards to purity and potency, questions remain about appropriate measures of potency for FMT and our current understanding about whether there are specific organisms or consortia of organisms that mediate effectiveness. So as we move toward licensure of live microbiome-based products, I just want to reiterate here that only those biologics that are demonstrated to be safe, pure, and potent, and that can be manufactured in a consistent manner will be licensed by the FDA. And what this means is that we need clinical data to demonstrate safety and efficacy. But we need to remember that this is linked to product quality and consistency of manufacture. And all three are needed for licensure. And I want to end with some final thoughts here. Interest in microbiome-based biological products has increased greatly in recent years. And CBER's regulatory approach is science-based. And this does allow for novel approaches to be safely tested in the clinic. And also, we are committed to working with our sponsors to find a path forward. Thank you for your attention.
So I think we have time for a few questions. Bob, did you want to come up? Sorry.
My name is Mary Ellen Sanders, and I'm with ISAP. And I had a question regarding the intent of research and research endpoints, considering the fact that there's an overlap between the definition of drugs and the definition of foods. And both drugs and foods can affect the structure and function of the human body, and both can reduce the risk of disease, as well as provide nutritional support for other disease conditions. And the situation exists in the United States today, where human research on probiotics is viewed, even when there's no intent to develop a drug, it's being viewed as research that needs to be conducted under an IND. And my question is there a role that CIFSAN can play whereby oversight of human research on probiotics that fits under legitimate legal intentions of use of foods and dietary supplements to affect the structure and function of the human body or reduce the risk of disease can be overseen by CIFSAN rather than CBER? The CBER does a great job overseeing drug research. They don't really oversee food research, yet these are legal uses of foods.
Thank you very much. So the way I understand your question is if someone is using a product, say licensed as a dietary supplement, and if they use that product licensed as a dietary supplement for an investigation for a structure function outcome, that would not be diseased, and that would not, I don't believe, require an IND. But if you were to use that product labeled as a dietary supplement for a disease outcome, something more than a structure function claim, now you're in essence using it as a drug, and it would require an IND.
Okay, but just to clarify, my point is impacting the structure function of the human body legally is both foods and drugs. And so CBER,
You're right.
CBER very much could look at a structure function endpoint of a study and say this is a drug endpoint. And they'd be correct if the intent was to market a drug. If the intent is not to develop a drug, and in addition to structure function, you also have reduction of risk of disease, which are appropriate for foods and supplements. If you have research endpoints that are focused on that,
right,
is there a way for CIFSAN to oversee that research rather than CBER?
We wouldn't oversee the research, but we could certainly partner with our other product centers or our product offices within the agency to make sure we're all working off the same definition of what a proper structure function claim is for a product labeled as a dietary supplement. And I think we do that, actually. We do communicate when IND requests come in and someone's making a claim, and it's sort of in that gray area that I discussed between a disease claim and a structure function claim. We do actually communicate and try to flesh out which side of the line it comes down on. If it comes down on the structure function side and your label is a dietary supplement product, I don't believe you would require an IND. I mean, the devil's always in the details, but based on the high-level description.
This could also be investigational, so it might not even be marketed as a product as yet. It's a question of developing the research on it.
Yeah, it would have to be a dietary ingredient that's legally on the market, I believe.
It seems that the default, though, is to but to choose on the side of considering that to be drug research, not food research.
I actually have to say that this is a complicated area, and as Bob has just alluded to, sometimes what at a high level might appear to be a dietary supplement drug claim it actually, and sometimes it isn't when you get into the protocols. And I'd also like to say we do have that guidance, and as you probably know, the dietary supplement, part of that, in that guidance, some of the requirements for an IND have actually been stayed for certain studies of dietary supplements.
I think it could be summed up with the statement, there are no CIFSAN regulated INDs.
A question for Sheila. Is there a definition for the term live microbiome-based product?
No.
Is it different from LBP?
No. And what we meant to do with that is sort of to broaden the scope a little bit. The definition for live biotherapeutic products really, back in 2012, didn't take into consideration FMT, for example. And so I think what we hope to do with this is to really show that this workshop encompasses more than just live biotherapeutic products in that sense.
And FMT is not an LBP.
Well, yes. So you could say that FMT does fit the definition of a live biotherapeutic product, and that is actually discussed a lot internally. We did not have that in mind when we wrote the guidance. It's time to reconsider.
We do recognize that there are aspects of that guidance that may not be applicable to FMT, such as the requirements for cell banks and things like that, but we're not unreasonable. We do recognize that.
Catherine Duff. Just would like to say, when you had the slide up about the original workshop in 2013, that should not have read members of the public. There was just one of us, and it was me. And everywhere I go, I'm the only member of the public talking about fecal transplant. And as the touchstone for literally hundreds of thousands of patients around the world that contact us every day, I think I have 27,000 unread emails right now. Our concern has always been that as engineered microbiome-based products come through the pipeline, that condition of live microbiome-based products, the phrase about being able to prove the potency, purity, standardization of manufacturing will be used to exclude natural fecal transplant, which I have to say, as the voice of the public, would be a huge wrongdoing and disservice to millions of people, and there would be a public outcry like you cannot imagine. So we hope that that is not the intent and that that will not be the outcome. And of course, whenever these draft guidances are published, we rally the troops and we comment vociferously. And I know that those comments are seen and heard, and I appreciate that. But, we are watching closely and we are very concerned.
Yes, and thank you for that. And we're working hard, and really, it's a complicated and difficult place. And I think where we are now is we're really trying to think of the best way forward here. And we do listen, and we are taking everything into consideration.
As the only member of the public in the NIH-funded microbiome transplantation working group, we noted that there is also no way to ensure the consistency of other products, biologics, such as blood or bone grafts or tissue. And we all felt that natural stool-based microbiota transplantation falls more into that category than a traditional drug. So we just hope that you will keep the findings of that working group in mind. Thank you.
We have a question from our overflow room. LD20, you can ask your question.
This is Deborah Demuth from Bayer. I have a question, clarifying question actually. The FDA has gone on record that prevention of antibiotic-associated diarrhea is a disease claim. And there's several products, dietary supplement products on the market with substantiating evidence that make the claim they help prevent antibiotic-associated diarrhea. Can you help clarify whether or not FDA still considers that a disease claim, or has something changed? Thank you.
In the abstract, I mean, I can't see the claim in front of me. As I mentioned, it's a totality of the circumstances, the label and the labeling. It's never just one statement. Although a statement, treats or prevents that, antibiotic-associated diarrhea, that would be a disease claim. There are folks in this room that have had this discussion with us. I'm looking right at Amy right now because she comes in a lot. We're open to conversation on this. Right now it is a disease claim, and based on our resources and priorities, we may enforce it as a disease claim. We could send you a letter, or we could seize your property if it was drastic enough. I don't know that this rises to that level because, as you said, you've seen those products on the market. It is a disease claim. It is a violation. Whether they have substantiation or proof for it or not is right now irrelevant. But for the here and now, something that direct would be a disease claim.
Pinaki Panigrahi, University of Nebraska Medical Center. When I listened to individually to you, it was very clear and I understand 100%. But when I put them together, I'm totally confused. And my concern is why can't CIFSAN have an IND? And the reason I'm asking this question is we need more research and more studies and more work done in this field, not regulate and not reduce it. And you also mentioned that if somebody does something develops it as a drug, others cannot come back and sell it as a supplement. But how can you stop them? They don't say they will say it may help, or they will cite something, or even the physicians can use it off-label, or all different things. You don't have to have regulated products to be prescribed even. So how do you, and the question to CBER is, okay, I understand, I am fully with you. I want to develop probiotics as drugs. At the same time, it cannot be always developed as drugs, because if you do that, and even after it is approved, if you believe anything that Vince Young said, that's not the particular organism which is doing the final change in the physiology or in the pathology. There are other things happening. How can you say that, okay, that's the one which did it, and that's why it's a drug?
I appreciate your question because it gives me a chance to tighten up my language or provide some context. When I said there is no such thing as a CIFSAN regulated IND, that's not to say that a product marketed as a dietary supplement couldn't also be studied as a drug. It could be the same exact ingredient, the same exact product that's looking for a disease endpoint or surrogate endpoint could be the subject of an IND and be studied as a drug. So I didn't mean to say that you couldn't take a dietary supplement and never study it for those purposes. You can. It's just then you would be regulated through the IND mechanism. As far as a 201FF3B violation, where someone tries to market an ingredient as a dietary ingredient or dietary supplement while there's an IND or NDA with significant clinical studies to support it, that's something that we do take seriously and we do enforce. But because we're stuck with a largely post-marketing paradigm for enforcement of dietary supplements, we don't get the opportunity to enforce it until we find out for some reason. And that may be somebody who has a proprietary interest in that ingredient letting us know. We might get involved because there's a signal that it's hurting someone. We don't actively look for those types of violations, but when we become aware of it, we do enforce those.
Boriana Tsarovsky, Johnson & Johnson. What is FDA's stance on prebiotics? And what would be the best reference where we could find this?
Glad I came. So prebiotics, as far as I know, now we don't really have a hard and fast definition even for a probiotic. But a prebiotic, not that we have a hard and fast definition for that, but I believe it's something that supports probiotics, supports the environment to allow probiotics to develop. If they're a dietary ingredient, if they're an old dietary ingredient or a new dietary ingredient for which a notification is required and one is made, it could legally be on the market. It would just have to follow the paradigm. Is it a dietary ingredient? Is it not meant to supplement a meal? Is it not derived from tobacco? Is there not a 201FF3B exclusion from the definition of a dietary supplement? It could legally be on the market as a dietary supplement if it did the right things.
If it's simply being incorporated in a product as an ingredient that has a product that has a different purpose, then what would you expect?
Well, it would have to be a legal ingredient. It would have to be an ingredient that's on the market legally as either a conventional food, a food additive, or a dietary ingredient and a dietary supplement. If it's in a dietary supplement, it would have to be on the proper part of the label. If it's not there for a technical effect, it would have to be listed as an ingredient. If it's listed as an ingredient, it would have to be a legal ingredient, which means it would have to fit the definition of 201FF. I don't know if that made any sense. I'm really sorry.
Where can we find this on the FDA website?
How about if we chat?
Is anything been public? Okay.
Okay.
Thank you.
Okay, and I'm looking at the guidance, actually, Mary Ellen, and if a clinical investigation of a dietary supplement is intended only to evaluate the dietary supplement's effect on structure and function, an IND is not required. And there is a stay on the studies to support a health claim. So, and with that, I think we should break for a quick break and be back by 11.
My first speaker. Oh, try. Perfect. Yes. Thank
that would come out. Okay. So we talked to Burnett a month ago or something. Yeah. Okay. Very good. Just want to put a name to a face. Yeah. Yeah. Parents and friends. Eric Mertz. Eric Mertz under section. Pat Carolyn. Pat. That's kind of second in our. Yeah. Yeah, Vince is, yeah. Hang on. I would have efficient. And it's been very helpful down. You find a lot of you can have grants. But it's not the rule. Most of the time, yeah. Okay. Oh, sorry. That'll work. That was like, it was like many pictures there, and then I was very different with all things.
John and you hook up, and we're excited, that Lactin B study is fully enrolled. I have it up on my whiteboard. Last patient, first vision, last patient, last visit. And now I'm waiting to see. Exactly. Bet.
Yeah, hey, I just see it. Yeah, I'm gonna need your help. It's a 30-day package, so please get up and help me. Well, I mean, you don't hold it. But I think that I don't know, I got it. But I think it's telling you about a little bit. You want? I brought some. Brought some of my multi-million-dollar drug. You taste it. I got a little cup. You can have it. Got even worse. Get a new brand for the top. Last month. You know, you were quick. Clearly stuck with water. Ain't fatty acids. We're not trying to hide anything. Our outcome is short chain fatty acids. I don't know. Now, whenever it makes sense. Yeah.
Oops, sorry.
Yeah. An outcome. We're not even looking for claims. Yeah. What? Not even a step. Biomarker, it's clearly not we're not looking at negative. Yeah. I get my grade. I look forward to your talk. So yeah, my original grades were through USDA and Foundation and Advance. And I get to read
No problem.
Yeah,
Get in
so
there.
before they said you can go ahead and do it. Yeah. And they're like, you're not in this room?
No, I'm.
Here, find those?
Okay, people, back to your seats.
Okay.
Like clinking glasses. Yeah, they did. Oh, now? Now?
Yeah, I know.
Want me to just get started when people sit down?
I'm going to run out. I can leave early.
Oh, he was right outside. I was just talking to him. That was what was holding me up.
You're Convening ESSA for this talk.
Hello. If everyone would mind sitting down, and we're a little behind. I think it's just forward, yep. All right. Thank you very much. I appreciate everybody coming back in. A little bit of a brief break, and I'm not going to hold us up, just so you all know who I am. I'm Susie McCune. I'm the director of the Office of Pediatric Therapeutics in the Office of the Commissioner at the FDA. And the folks kindly invited me to be part of this conversation today because I'm a pediatrician and a neonatologist and particularly interested in this area. So just to give you an overview of what we're going to do now, we have session 2, part 1 and part 2. So the session 2 is entitled Safety and Effectiveness of Live Microbiome-Based Products Used to Prevent, Treat, or Cure Diseases in Humans. Part 1, which is what I am moderating, will be before lunch. Part 2, which Paul Carlson will be moderating this afternoon. And then we'll have all of our speakers come together for both Part 1 and Part 2 to have a panel discussion. And I will say that after all of our 3 speakers this morning, we'll hold questions, and then the 3 speakers will be able to answer questions, clarifying questions before lunch. So with that, I'd like to introduce our first speaker, who is Dr. Joseph New, who is Professor of Pediatrics and Director of the Neonatology Fellowship Training Program in the Department of Pediatrics at the University of Florida College of Medicine. Dr. New is going to talk to us today about the use of commercially available products to prevent NEC.
Thank you and good morning. Here's my disclosure slide. And over the next 15 minutes, I'm going to quickly cover historical perspectives and difficulty defining necrotizing enterocolitis. This is a big conundrum that I think we're just beginning to recognize more and more, that we do not even have a good definition for this particular disease. Then I'll talk a little bit about the pathophysiology of the most classic form of necrotizing enterocolitis, and then get into probiotics and necrotizing enterocolitis. Well, let's begin. This is a typical neonatal patient cared for in the neonatal intensive care unit. And these babies now, over the last 50 or so years, we've been caring for more and more of these babies. At one point in time, when I first started my residency program, we would take babies who were 26, 27 weeks gestation and put them at the side of the neonatal intensive care unit and allow them to die. Now we are taking 22, 23 weekers and being very aggressive in trying to save these babies. And along with this, we're starting to see more and more this particular disease process that we call necrotizing enterocolitis. And here's a picture of a baby with this problem. This is not a typical inflammatory bowel disease. This is very different than what we see typically. This is a disease that once it affects the baby, within 24 hours, that baby can be dead. And so this is a problem that is very difficult to treat, and I think we need to aim at prevention of this particular disease. So over the last 50 or so years, since we've been starting to really work on these, saving these very small preterm babies, we really haven't made very much progress in this disease. And there's several reasons for this. One is that we've been lumping several disease processes into or underneath the umbrella of, quote, necrotizing enterocolitis. And I'll talk about this very briefly. We have some animal models. For example, there's this rodent model that you asphyxiate and as babies, and you put them into a refrigerator and you treat them with antibiotics, and they develop some necrosis of the bowel, and that is called necrotizing enterocolitis. That is not the same disease that we see in preterm babies. There are over 100 published papers using that particular model. And then there's been a narrow focus on individual inflammatory pathways or oxidative pathways rather than a whole systems approaches for this disease. So I think we need to consider looking at whole systems rather than just individual pathway components. In the late 1970s, a surgeon by the name of Martin Bell developed these criteria called staging criteria for necrotizing enterocolitis, stages 1, 2, and 3. We're beginning to recognize that stage 1, if you take some of the babies that we care for today that are born less than 750 grams, about 70–80% of those babies could be diagnosed as having stage 1 necrotizing enterocolitis. Stage 2 relies on radiographic criteria, and sometimes we make mistakes with those radiographic criteria. Stool in the bowel actually can look like pneumatosis intestinalis, which is one of the major criteria that we use for diagnosing that disease. Stage 3 relies on free air in the peritoneal cavity. Well, we have another disease entity called spontaneous intestinal perforation, which occurs fairly early in very preterm babies, and some of our surgeons don't operate on those babies. And this is not necrotizing enterocolitis, but these babies get recorded as having necrotizing enterocolitis. So the criteria that we are using for this disease are not very good. We don't have a very good definition. So we have in the middle here, this circle, intestinal injury that we are calling necrotizing enterocolitis, but we can have some babies who have cardiac problems. For example, hypoplastic left ventricle or interrupted aortic arch. Those babies don't get enough blood to their gastrointestinal tract, and they develop necrosis of the intestine. They get charted as having necrotizing enterocolitis, but that's a misnomer. They have ischemic bowel disease, but not true necrotizing enterocolitis. And we have these spontaneous intestinal perforations. Then we also have some diseases that are associated more with what we are feeding the babies. So really this is more than one disease, and we are struggling with really trying to define a classic form of this disease process. Now, we do think that microbes are associated with this disease, and our group at the University of Florida was among the first to see differences in the microbiota in stool samples of preterm babies prior to the development of the disease. And what we were able to do, working with Dr. Mohan Pami at Baylor University, we were able to take sequences from several different neonatal intensive care units that did the same types of studies. So we had stool samples from several different neonatal intensive care units that looked at necrotizing enterocolitis versus control babies, and we were able to find, as we see on this particular slide here, differences in the microbiota prior to the development of the disease. So here we have control babies. Each one of these colors represents a different phylum of bacteria. And in the controls, you don't see a lot of differences. But in the babies who subsequently develop necrotizing enterocolitis, over time, we see an increase in these light blue, which are the proteobacteria, and a decrease in the firmicutes, okay, also a major phylum of bacteria. We also saw that there were very few Bacteroidetes in the necrotizing enterocolitis babies. But again, these are studies that were done at the phylum level. But they do suggest a difference prior to the development of the disease in the microbiota. I don't have much time to talk about any of the other agents, but I do want to talk about probiotics. Okay. The question here is, are we there yet? And I think there's a lot of debate right now, very heated debate about the use of probiotics in preterm babies. And in fact, I've seen several review articles that say the only disease entity where we have definitely proven that we can prevent a disease is in necrotizing enterocolitis using probiotics. Okay, this is in review articles. And so there's this belief out there that we're there with the use of probiotics. Let's talk about this a little bit and where this story came from. In 2010, a meta-analysis came out in pediatrics looking at 11 different centers where they used 10 different probiotic preparations. And here we see that in terms of prevention of necrotizing enterocolitis, favored treatment. In fact, death was lower in those babies who received the probiotics. Okay, that's probiotics. So in this meta-analysis, 11 studies were evaluated. 10 different probiotic preparations were used. 10 different preparations were used. That's like saying, I'm going to prevent ear infections using chloramphenicol, amoxicillin, clindamycin. That's sort of a similar analogy, okay? they found that risk for neck and death was significantly lower in the probiotic group. Sepsis did not differ. And, in quote from that paper, the overall incident evidence indicates that additional placebo-controlled trials are unnecessary if a suitable probiotic product is available. Okay, so, you don't see this very often after a meta-analysis. You usually see more studies are needed. Okay, here, done. Okay, it's all over with. And there was a commentary along with this saying, is it ethical to not use probiotics in preterm infants? So the Journal of Pediatrics, the editors asked me to look at this very closely, and so I came out with this commentary in the Journal of Pediatrics, Routine Probiotics for Preterm Infants, Let's Be Careful. And I outlined some of the reasons why we do need to be careful and move slowly in this area. And I'm going to go through some of these rationales as we go on. First of all, I want to start with systematic reviews and these meta-analyses. If you put garbage in, you'll get garbage out. This is one of the problems. In many meta-analyses, after a few years, about 50% are proven to be not very good, untrue. One of the big mistakes is pooling data across trials as if they belong to a single large trial. And over the years, just about every single year since 2010, there's been another meta-analysis, or at least one meta-analysis, including a Cochrane review. This is the Bible for neonatologists. Cochrane review says that we should be using a certain agent, that we should go ahead and use it. Well, Cochrane review recommended that we should be using probiotics. But which one? There's hundreds of them out there. So here's a study that came from Europe, and they looked at one particular probiotic. They did this with a couple of other probiotic preparations, and they found no real difference if they just looked at one probiotic preparation by itself, rather than putting them all together. One of the biggest studies in that first meta-analysis that I showed you came out of Taiwan. And one point that was not very well discussed in that paper is seen here. See the red arrow pointing to sepsis? The study patients were those that received the probiotic. The control patients were those that were in the control group not receiving probiotics. So we had 12 babies in that study who developed sepsis, and one baby in the control group that did not develop sepsis. Okay, so large association with the development of sepsis in these really small babies. And if you look closely at the meta-analyses, babies less than 1,000 grams were not benefited by the use of probiotics. They were all babies that were greater than 1,000 grams. So more than 2 pounds, it seemed to have some benefit. Less than 1,000, less than 2 pounds, no real benefit. There was another fairly big study in Australia which was not powered to look at necrotizing enterocolitis. It was powered to look at sepsis, and they had 1,099 very low birth weight infants, and they found no difference in sepsis or all-cause mortality. But on secondary analysis, looking back, they saw that there was a difference in necrotizing enterocolitis. NEC went from 4.4 to 2.0% on the secondary analysis with a p-value of 0.03. The number needed to treat was 43, with 95% confidence interval, 23 to 233. There was no effect in babies less than 1,000 grams birth weight. Another study, this is the largest study and the only study done thus far that I'm aware of. This was done in UK by Dr. Costello and colleagues. It was a double-blinded, randomized prospective study, adequately powered to look at necrotizing enterocolitis, using a Bifidobacterium breve probiotic, and it studied babies at 23 to 31 weeks gestational age, and they found no difference in NEC, late-onset sepsis, or death. So the question that was raised this morning, is this a food supplement or drug? It depends. Well, maybe it doesn't depend after what we heard this morning. If we have a medical claim, prevention of necrotizing enterocolitis usually should be considered a drug. Drugs that are sold by prescription are subjected to rigorous testing. Foods can be sold by anyone and not subjected to rigorous standards for the most part. Here's one study, and this is one of several case report that shows certain bacterial species that caused some bacteremia in babies receiving this particular probiotic. We see several of these in the literature. A few years ago at Yale University, a preterm baby died, was taken to the autopsy suite, found to have mucormycosis. The mucormycosis was traced back to the product. Was this a product that was tainted? Was this a product that was not well controlled in terms of its development? This is what we are trying to avoid, and this is why I'm saying we need to be careful. In the United States, about 15% of neonatal intensive care units are already using probiotics, but the types of probiotics that are being used tend to be those that happen to be available in the hospital. The most commonly used is Lactobacillus rhamnosus. Lactobacillus rhamnosus, the studies that have been done have not been shown to decrease necrotizing enterocolitis. So here we see the states, and we have no real evidence for safety or efficacy in some of these probiotic preparations that are being used right now in the United States. There's also no current standards for quality control of this reconstituted product and good manufacturing practices for the use of probiotics, as drugs are not available. The quality of some of the products are questionable. People have looked at the probiotics that are actually out there, and some of them are not really what is being sold in terms of the product that they say that this particular strain, this particular genus and species is in a sample. They are finding different genuses and species using PCR technologies. You have to be careful. This is a study that was done by a group at Emory. Ravi Patel is also here, and this is an interesting study. Neonatologists are sick of necrotizing enterocolitis. We hate this disease. This is a disease that kills babies very quickly. 5 to 7% of these babies are babies from 500 to 1,500 grams are affected by this disease, and when these babies develop the disease, it's very hard to treat, as I mentioned before. If it goes on to surgery, 20 to 30% of those babies die of necrotizing enterocolitis. If they survive 5 years of age, and if they have a short gut, it costs $5 million to care for that baby with that short gut. This is not a trivial disease. And these babies who have necrotizing enterocolitis also have neurodevelopmental delays. So this is a terrible disease, and we are looking for something that will prevent this disease. But the problem is that we are sometimes maybe a little bit too aggressive in moving forward. This is a interesting study that if you look at the necrotizing enterocolitis prior to implementation of probiotic, here we see necrotizing enterocolitis. And after implementation, we see an increase in necrotizing enterocolitis. Study in Europe. And I'm just going to show you the title here. This just came out very recently. Increased incidence of necrotizing enterocolitis associated with routine administration of probiotic in extremely preterm infants. Again, this was not a prospective randomized trial. This was a retrospective observational type of study, as was the study at Emory, which is limiting, but this is something that should be. So, in summary, neck pathogenesis is multifactorial. Even if we invoke a classic form of necrotizing enterocolitis, we need to have better definitions going forward in our future studies. Treatment of NET, once it's developed, is extremely difficult. We need to prevent. The intestinal microbial environment, along with developmental aspects of the GI tract, are key in understanding the pathogenesis of NET. We need more studies. We need to have better systems, enteroids, animal models, to evaluate mechanisms that fulfill criteria for causality derived from strong associations found in humans. And lastly, once we have a clear understanding of the causes of the different forms of NEC, we will be best able to target preventative strategies. And again, let's be careful.
Thank you so much, Dr. New. Our next speaker is Dr. Daniel Merenstein, who is a professor of family medicine at Georgetown University, where he also directs the family medicine research. He also has a secondary appointment in the undergraduate department of human science in the School of Nursing and Health Studies. And today he's going to talk to us about the evidence is in for probiotics to prevent antibiotic-associated diarrhea. What is holding up evidence-based use in the United States? And I just will say that we're shifting to the diarrhea topic for the next two talks. Dr. Merenstein.
Thank you very much. I really appreciate this opportunity. I'm excited that so many people are interested in this. I'm going to be speaking about what I study, antibiotic-associated diarrhea, or as I refer to as AAD. But I was also asked to speak about why it hasn't taken off in an evidence-based manner in the United States, and I'm going to give some opinions about that. These are my conflicts. I won't be speaking about any of these today. I put this up, in God we trust, and all others must bring data, because I am going to give some opinions today. And you might not agree with my opinion, and that's fine. That's reasonable. And we should discuss it later at the panel or at lunch or whenever. But I am going to present the data. And just because you disagree with my opinions, I hope you don't ignore the data, because the data are really robust and really tell a story. So if you remember Dr. Young's graph, it just kept going up, the microbiome research. But this is probiotic research, and it's been going down, down, down. I assume this will go up a little bit because the year's not over, but it's not going to get to up anywhere near there. We're going to talk about why I think that's happening, why it's obviously a serious problem. So I'm going to discuss the evidence behind AAD for probiotic use. And just to make it a little more robust, I'm going to show you what other people say about it, so you don't think it's just my opinion. And then I'm going to give you some opinions of why I think we're having a hard time implementing this in the United States. So this is the Cochrane pediatric AAD, and I agree with Dr. New that evidence in is only as evidence out is only as good as the evidence in. But really, in medicine, this is considered the highest level of evidence. There's over 23 studies, almost 4,000 patients, 11 of which use a single strain. In AAD group, in the probiotic group, excuse me, it's 8% AAD versus 19% in the control group. If you work your way down, the relative risk reduction is 58%. I don't have time today to talk about other interventions, but next time you read an article, think about 58% and where that falls in. The absolute risk reduction is 11%. The number needed to treat is 9. And again, when you read articles and you see number needed to treat, think about when you see such a low number needed to treat. The initial thing, and this is what Dr. New already said, is, okay, what product do I use? Well, there's multiple products to use, but let's just take one product. This is a meta-analysis of one single strain. 12 RCTs, almost 1,500 participants. Almost the same exact data, 22% versus 12%, relative risk, 49, number needed to treat, 9. Adverse events, as we've seen and many people also have seen and shown, and the RAND study showed this too, are nearly the same in the experimental and the control groups. In fact, a lot of RCTs show they're lower in the experimental group than the control group. I know we're going to talk about C. diff a little bit later, but I think there's no way to talk about AAD without talking about C. diff, because C. diff is really what we're mainly worried about when we're talking about AAD. This is another Cochrane review, 8,600 participants, 8,672. 27 studies, and I also want to go back. Not all these studies are perfect. I don't mean to say every study was low risk of bias. They have some problems with some of the studies. Incidence in probiotic group was 1.5, control group was 4%, relative risk 62%, absolute 2.5, number needed to treat 40. Interestingly, when they looked at this, it really mainly is a benefit when your infection rate is greater than 5%. So if you know your hospital rate is greater than 5%, the data is much more impressive. Physicians use the medical letter, pharmacists use the pharmacist letter. This is well-respected evidence-based review. They conclude treating 12 patients with a probiotic prevents 1 case of AAD, treating 29 prevents C. diff. They go on to say probiotics reduce the duration of acute diarrhea in infants and children by about 1 day. And for those who might say it's just 1 day, that's exact treatment we get when we give influenza drugs, when we give antibiotics for strep, we give antibiotics for otitis media. We'd be happy with 1 day. Usually it's actually less than 1 day. 2 years ago, JAMA had 3 articles about probiotics. The first one was just a survey. It showed 156 increase. People have already talked about that. I think this editorial, though, was even more powerful. They said, not all supplements, of course, lack evidence of efficacy. Many supplements, including vitamins, minerals, and probiotics, are important components of modern healthcare. I don't think we would have seen that 10, 15 years ago, but it's well accepted in the mainstream medical journals, the evidence of probiotic. And they concluded with a third article in that, where they talked again about the evidence of the AD, which I've already shown you. 3 articles in JAMA talking about probiotic usage. So how are people using probiotics? This is one survey, found 87% of academic hospital formularies carry a probiotic. If you're in this area now, there's 3 major hospital systems. Hopkins, I'm going to talk about Hopkins in a few slides. There's MedStar and Inova. MedStar and Inova, so there's 10 million people in this area. Not one, if they're hospitalized, has a chance to have a probiotic that has efficacy. I'm not going to call out products, but there's products on these, and just like Dr. New just showed, that don't have efficacy that hospitals use mainly for cost reasons. And it's embarrassing. And if you get hospitalized now, even though I've shown you the data for AAD and prevention C. diff, you can, in the local area, you will not get a probiotic unless you bring it to yourself that will prevent AAD. And there's a good chance they're going to put you on antibiotic if you're hospitalized. The CDC did a review in 145 hospitals with about 2 million discharges. They found 96% of hospitals use the probiotic. You're 9 times more likely to get antimicrobial and 20 times more likely to be diagnosed with C. diff if you were on a probiotic. They concluded in the sample of U.S. hospitals, a sizable and growing number of inpatients receive probiotics as part of their care, despite inadequate evidence to support their use in this population. I would just add editorial, just because you don't know the evidence, you shouldn't conclude with inadequate evidence. The evidence was there already. The evidence was clear from Cochrane reviews, the highest level evidence we have, that the number needed to treat is 9 or 40. For AAD, 9, and for C. diff, 40. I'm going to talk a little bit about FMT data. I am a big proponent of FMT. I have a son with ulcerative colitis. I think the FMT data is very promising. I think it not just teaches us how we can do with FMT, but we can do with drugs. But I'm going to present the data. So before you attack me with FMT, look at the data. Then in 2016, there was a review, about 7,500 original articles, not studies, articles, again, mainly reviews. This is well accepted. This review found about 28%. You'll see about 30% AEs, mostly mild, but some serious infections. And again, as someone already pointed out, we have no idea about the long-term implications. These are all I could find. And I wrote every author and asked them if there's other studies. There's probably ones in other languages. But there are 5 RCTs. This is what the FDA changed their discretion in IND about. 5 RCTs with FMT. 2 were done with enema, 2 with colonoscopy, 1 with nasoduodenal tube. 187 patients. Of these 5, 2 were blinded. 2 blinded studies. The one, the best highest level that was placebo-controlled blinded, found efficacy of 61% versus placebo 45%. So that's the data. Look at the data. Now, IDSA, which we can debate is if it's evidence-based review or not, looked at this data just this year. And they concluded there's insufficient data at this time to recommend administration of probiotics for primary prevention of CDI. 27 clinical trials, 8,600 participants. They said for fecal microbiota transplant is recommended for patients with multiple occurrence of CDI who have failed appropriate antibiotic treatments. And again, one is prevention, one is treatment. But just think about it if we flip those numbers. If we flip those numbers, I would never have gotten a grant for AAD. I would never have even thought of applying. If you told me you have a product, St. John's wort, that has 5 clinical trials, 2 of which are blinded, and I wanted to apply for a grant, my chair would be like, can you find a better product to apply for a grant? Because you're not going to get funded. So part of the reason I think it hasn't taken off is lack of understanding evidence, maybe bias, but there's no question, and it's unfortunate, I appreciate CBER inviting me and they asked me to speak about this, that CBER has part of the blame. There was a horrible death that Dr. New talked about. Horrible death. And this is what CBER did after that. They said, the FDA encourages healthcare providers who use dietary supplements containing live bacteria yeast, probiotics, to submit an IND for FDA's review. FDA's primary goal in reviewing IND are to ensure the safety and rights of subjects and help ensure the quality of the scientific study of drugs is adequate to permit an evaluation of drugs' effectiveness and safety. This is what they sent out. This is what happened. There's headlines that you get, tons of headlines, but one on Forbes, infant death triggers FDA health providers warning of probiotic risk. But this is what happened at Hopkins, one of our top institutions in the country. They outlawed all probiotics. This is what they wrote. Due to the documented risks associated with probiotic use in the hospital, probiotics are not available for use at any Johns Hopkins Health Service hospital. Not purchased, stored, administered, or dispensed. I'm going to read that again. Not purchased, stored, administered, or dispensed. The utilization of a patient's own supply of probiotics from the hospital could put patients and healthcare workers at risk for possible infection and is therefore prohibited. I did my fellowship at Hopkins. They let me give Benadryl. No study was ever shown. It was one study ever in 1976 to infants, 6 months to 9 months, to see if it helped them sleep through the night. Benadryl. We know as physicians there's major side effects of Benadryl. That was fine. The IRB approved it. This is what they wrote about products. You are not allowed to bring in your own probiotic into Johns Hopkins Hospital. It's the danger of other people. So clearly, we know, because there's bright people at Hopkins, this was written by the lawyers. And the lawyers looked and they said what FDA wrote in the letter. FDA encourages healthcare providers to use dietary supplements to submit an IND. It's pretty clear, actually. You can't blame them. If you're going to use a probiotic, you need to submit an IND. But doctors can't do that. They're not going to do that for NEC. They're not going to do that for AD. They're not going to do that for colic. They're not going to submit an IND every time they use a probiotic. Just a few months ago, we had a horrible transfusion problem with platelets causing infections with ACB. This is what happened. The Centers for Disease Control and Prevention, working with 2 states to investigate the potential ACB complex transmission through platelet transfusion, has issued a nationwide call for cases. Please report any patients who developed or developed sepsis due to ACB complex within 24 hours of receiving platelets. Imagine if the CBER did that. This was a horrible death, and it was an infection with a contaminated product that should have been called to as it was. But instead of saying we should figure out the products are safe, or if you see anything in your hospital, they sent a letter that says you need to give INDs when you get probiotics. That's greatly impacted probiotic research in the U.S. The second and the final thing that's greatly impacted is the definition, and you heard it today, and I think you heard it really clearly with the 2 speakers going back and forth in the confusion. So I study a yogurt. It is a yogurt. I brought enough for everyone to taste to prove it's a yogurt. In fact, I study the same exact strain that's in every one of these products, including infant formula. Same exact strain at the same exact dose, or they have a higher dose than I have. So I have a lower dose of the same strain. I'm on my 4th IND. The first 2, you could argue, were reasonable because it's antibiotic-associated diarrhea. And we already heard today that's considered disease by the FDA. Now, I already had 15,000 days because I'd done a prevention of preschool absences study with the same yogurt here. So they could have said, you can go to a phase 3 trial, because that's what NIH funded me to do. But they didn't. They said, you do a phase 1. So I did a phase 1 safety in adults, then in kids. Now I'm doing a phase 2. But even more surprising is about 5 months ago, I got funded to do a mechanism study of AAD. My outcome is short-chain fatty acid changes. That's it. Short-chain fatty acid changes. My secondary outcome is microbiome changes. There's no question, and it was already explained this morning, that's a structure function claim. There's no debate. There's no clinical outcomes. Healthy people, not hiding anything out, I'll send you the protocol. Healthy people, 60 people, the FDA required an IND for that. And this is slowing down research in the United States, and I'll show you that. And just quickly, I think I have time. This doesn't have to be. Well, there's lawyers here who can explain it too, but I also applied to do a chamomile tea study to see if it helped kids sleep through the night. I do lots of crazy studies. So I wrote to CDER because my IRB said, it's never been studied. You need to ask them if you need an ID. And they said, what we need is your CV to make sure you're a legit person and your protocol. 2 days later, they sent back and they said, you can go ahead with your procedure. That's what they could do. It took for the structure function claim, it took the FDA about 3 to 4 months for me to go ahead with my study. And because of that, I have to wait till the next budget season because we missed the budget season. So those are all the products. They have the same exact as the one you can taste if you want. Okay, FMT versus probiotics. Most hospitals, not all because of Hopkins, and I'm afraid some are going to follow because they're going to follow a place like Hopkins, are using them. But let's talk about it. What we always say about why you don't use it, Dr. New actually said a lot of this. Why don't we use it? We don't know the strain data. We do know the strain data. There's multiple products that are well proven for AAD. I showed you one, there's other ones. We don't know how to give. FMT, in the 5 studies we had, was given 3 different ways. What's the dose? Well, tell me the dose of FMT. We know the dose of probiotics. What are the adverse events? The adverse events are minimal. There are horrible cases of contamination, and there are some evidence of some sepsis very infrequently, but it's unbelievably low, unbelievably low. And what's the long-term data? Well, we don't have long-term data really for most of the drugs I use in clinical practice. It's not an excuse, but we just don't, and we clearly don't have it for FMT. We have, again, better for probiotics than we do for FMT. But CBER, rather quickly, I was impressed. I didn't realize it was as quick as Sheila showed. Within 2 months, changed their rule and let people go ahead with that. Why did they let this happen in 2 months? Interesting question. These are all studies throughout the world. So if you see in the U.S., and this is what happens in the U.S., about 40% of clinical trials are done in the U.S. We can argue about what I used again. I thought these were reasonable comparisons. Omega-3 is about 37%, vitamin D about the same. Probiotic trials are about 17. From my anecdotal evidence of people calling me and asking me how to do trials, I think that's on its way down. So the U.S. is falling behind in probiotic trials. In the age of the microbiome, the U.S. is not doing probiotic trials. So we need more AD studies. I'm a little biased, that's what I do for a living, but I think we do need more studies, okay? We need to know the time, the dose, how long, when you take, we need to do that. But physicians and patients are using these. I would argue often incorrectly. FDA, and specifically CBER's action via the letter and lack of waiving INDs has slowed research down. I think to conclude, CBER needs to remember their mission. Is responsible for advancing the public health by helping to speed innovations. And I think they've done the opposite with probiotics. Thank you for your time.
Thank you, Dr. Merenstein. We'll do questions for the group after Dr. Friedman's talk. So Dr. Stephen Friedman is a member of the sections of Pediatric Emergency Medicine and Gastroenterology at the Alberta Children's Hospital in Calgary, Alberta. In 2016, he assumed the role of Chair of Pediatric Emergency Research Canada and was appointed the Alberta Children's Hospital Foundation Professor in Child Health and Wellness. Today, Dr. Friedman is going to talk to us about use of probiotics in acute pediatric gastroenteritis, 2 large North American clinical trials. Dr. Friedman.
Thank you very much, and it's a pleasure to be here today. And I think this is a nice segue from the 2 earlier discussions. And I do have several disclosures, so I do actually hold an IND, or actually I'm not the holder, it's actually David Schnatter is the holder of an IND related to funding from NICHD for the conduct of one of the trials that was conducted in the US. And also similarly, Health Canada approval was obtained by NHPD for the CIHR funded trial in Canada. Study drug and placebo were provided by the manufacturers of LGG, as well as Loudmouth Solutions for the Lactobacillus rhamnosus helveticus. So I'm going to segue from antibiotic-associated diarrhea to acute infectious gastroenteritis, which is one of the most common diseases of childhood. Second most common cause of death globally in children under 5 years of age. It is different than NEC, where children in the U.S. don't usually die from this, but it's the global burden of it in kids and on the economy and on healthcare providers and on schools. 1.7 million ED visits per year in the United States, nearly 100,000 hospitalizations. And there are a few options to modify the disease course. So probiotics are being touted and advertised as actually changing the disease course in kids. We do currently have other options for symptomatic short-term relief and treatment of dehydration should it occur. So I'm going to, we've been hearing about Cochrane reviews and the pros and cons. So the biggest Cochrane review of this topic was done, and latest was in 2010 by Allen and al. And as you can see, there was a decreased duration of diarrhea. They concluded about 25 hours till the timing to the last diarrheal stool. Several challenges, though, that can come up from this. Number one, it's mostly inpatients, primarily in an era of rotavirus, which has been dramatically reduced to the introduction of the rotavirus vaccine in North America. Most of these studies were single center, very small sample sizes generally, although there were many studies, as you can see. But unfortunately, this led to significant heterogeneity. So there's 97% heterogeneity between studies in this Cochrane review. They employed varying probiotics and varying doses. Nonetheless, based on this data, several organizations issued strong recommendations, but they then go on to say, based on low quality evidence, that support the use of probiotics. The most notable being ESPGHAN, which is a large European group. There was no position statement really on this. The last one from the CDC was in 2003 and didn't really address this issue very much. So this raised one question that 2 networks decided to try to answer. So I'm the chair of Pediatric Emergency Research Canada on the right, and then we work closely with our sister network, PECARN, Pediatric Emergency Care Applied Research Network in the US, to conduct one question across 2 networks using 2 different probiotics. They shared a common hypothesis, however, that probiotic administration would result in a significantly lower proportion of children with moderate to severe disease in subsequent 14 days compared with placebo. And we didn't just look, and I'll come back to one isolated symptom, we looked at the global burden of disease as our outcome. They were conducted as randomized, double-blind, placebo-controlled trials. Eligible children were aged 3 months to 48 months. They both, in both studies, had to be clinically diagnosed with an acute intestinal infectious process defined as greater than or equal to 3 episodes of diarrhea in a 24-hour period, which is the working definition for gastroenteritis accepted by all organizations. We used a web-based random number generating software, randomize.net, employing random block sizes. We stratified by sites, and we used a 1-to-1 treatment allocation ratio. Several differences between the studies, which I'll try to highlight as I go through. I decided to present them kind of in parallel because they are so similar as opposed to going back and forth between the two. The U.S. study included 10 emergency departments, all pediatric centers. Kids were able to have symptoms up to 7 days, so up to full 7 days of symptoms. And this was based on the only one real prior study in the U.S., which was conducted by Nixon at Albert Einstein, which found actually that they did not see a difference in the group administered probiotics, but they did maybe see a trend amongst those who had a longer duration of symptoms at baseline. So they focused on that group of kids. They studied LGG, a dose of 1 × 10 to the 10th CFU BID for 5 days compared to placebo. And then randomization was also stratified by the duration of diarrhea, given the importance of that as an a priori hypothesis. In the Canadian study, we included 6 emergency departments, focused on children with shorter duration of diarrhea, because most of the other studies in the literature had shown greater benefit in shorter duration events, so up to 72 hours. And we studied Lacidofil, which is a combination of a Lactobacillus rhamnosus and a Helveticus product, in 4 × 10 to the 9th CFU twice daily for 5 days. Both of these dosage ranges were what was supported by the existing literature. In Canada, they actually held an indication for that dose and use of the product, and in the U.S., it's a commonly recommended dose of LGG. So we excluded children who are at risk for invasive disease and infection. And I didn't go into it, but there actually are numerous case reports in the literature of individuals with central lines who developed bacteremia with the probiotic strain, particularly in ICU settings. So we excluded all children with indwelling vascular access lines, congenital heart disease because of the risk of reports of endocarditis, immunodeficiency, immunosuppression, chronic GI problems such as IBD, in particular pancreatitis because of a large European study that showed increased mortality in that group, and then kids who may not have gastroenteritis, so bilious emesis or hematochezia, bloody diarrhea, so not traditional, at least North American viral gastroenteritis. The studies also had some specific peculiarities, kind of at the pushing of some of the local federal agencies. So premature infants and those less than 6 months corrected age were excluded, those on supplemental probiotics, or an allergy to LGG or the antibiotics that would be used to treat a bacteremic episode. In Canada, we excluded those who had had recent oral or gastrointestinal surgery preceding probiotics in the 2 weeks prior to enrollment, and soy allergy because the soy-based culture medium was used to grow the probiotic. We conducted follow-up surveys every 24 hours until symptoms had resolved for at least 24 hours, as well as day 5 and 14 post-randomization. And then actually in the U.S. at the FDA's urging, we conducted follow-up for safety at 1, 3, 6, 9, and 12 months following conclusion of this very short study. Stool specimens were also collected, and actually we use rectal swabs, and we can discuss that if people are interested. We actually collected specimens on all individuals who participated. We analyzed them for infectious agents, including 15 pathogens in both sites using a multi-analyte pathogen panel. And then we used an in-house viral panel for 5 viruses in Canada, along with bacterial culture. And then we also did independent testing of the batches of the probiotics in both studies to ensure that they were delivering the CFU counts that we had intended to deliver. Our primary outcome was moderate severe disease, defined by a modified Besicari scale score greater than or equal to 9, and I'll discuss that on the next slide, which ranges in score severity from 0 to 20. We secondarily looked at duration of diarrhea, duration of vomiting, future healthcare provider visits, as well as adverse events. So this measure is a composite score, and we chose to use a composite score as opposed to individual measures, because what if you reduce the duration of diarrhea, but they actually have more diarrhea for 2 days, but they have it for 2 days instead of 3? What's better? I don't actually know. I don't think caregivers really have an answer to that. There is an easy one. So this is a score that's actually emerged from the rotavirus vaccine trials and been adapted for use in the outpatient setting. So it actually looks at duration of diarrhea, duration of vomiting, maximal frequencies of diarrhea, maximal frequency of vomiting, fever, which is very concerning to caregivers when their child is ill. And then we also looked at interventions. So healthcare provider interventions, either as an outpatient or in the emergency department, and need for IV fluids or hospitalization. Our sample sizes had 90% power to evaluate a 25% rate in the placebo group, aiming for a number needed to treat of 10, as you were hearing, but number needed to treat, which would be based on a minimally clinically important difference of 10%. We conducted two-sided analyses with 5% significance and adjusted for follow-up drop-ins and drop-outs, so meaning people who take probiotics over the counter even though they're not randomized to it. And then we did interim analyses, so we adjusted for that as well. In the US, the calculated sample size had to be increased because on one of our analyses of the probiotic product, it was found to have too low of a CFU content, lower than what we had intended to deliver. So we worked with our DSMB to increase our sample size appropriately to 971 participants in the end. The Canadian trial, there were no concerns in that regard and ended up enrolling 886 participants. Our analyses were by intention to treat principles, multiple mutation, and we employed logistic regression stratified by site with secondary analyses looking at other covariates, and then we conducted subgroup analyses looking for interaction. I'm going to present the Canadian data first after this slide, which essentially shows the groups were similar in both studies, around just over about 16, 17 months of age. The only difference being the duration of diarrhea slightly higher in the US cohort. As you can see over here, 57 hours based on the eligibility criteria, and hence their baseline modified Vesicari scale score was slightly higher at 12 compared to 10 in the Canadian cohort. So in the Canadian study, as you can see here, if we look at all participants, the proportion who actually had the outcome of interest, the primary outcome in the probiotic group was 26.1% compared to 24.7% in the placebo group. We look at some of our a priori identified subgroups: kids less than 1 year of age, exclusively breastfed, antibiotic usage in the preceding 14 days, or greater than 70% compliance. As you can see, there was no difference between groups. Look at some of the secondary endpoints. These are particularly important because of the meta-analyses that had shown reduced duration of diarrhea. When we look at that, there's no difference in diarrhea duration, no difference in vomiting duration, no difference in follow-up healthcare visits. There additionally were no differences in adverse events between groups. When we looked and dove a little bit deeper into this issue of duration of diarrhea, because that's been the greatest claim for usage in acute infectious gastro, we looked at daily episodes of diarrhea from our diaries. And as you can see, they're actually essentially identical between groups with an incident rate ratio of 0.98. Only difference we did find was in vomiting. The incident rate ratio was slightly higher, and that was primarily due to a difference on the first day of treatment. However, the magnitude is actually relatively small, and probably the clinical significance of this is minimal at 0.83 versus 0.55 episodes on the first day after randomization. Now we're going to move on to the US study, the PCARN study, and the results are actually remarkably similar. The proportion having a modified Vesicari score greater than or equal to 9, which was our primary outcome of moderate to severe disease, 11.8% in the LGG group compared to 12.6% in the placebo group. The p-value was 0.83. When they looked at mean episodes of diarrhea per 24-hour period, or the mean episodes of vomiting per 24-hour period, a very similar graph is displayed. There were no significant differences on any of the days or on either of these parameters. This is a busy figure, and what I'm just going to try to highlight is what we looked at here to show you how we tried to stratify and look at different things. So the top five, the 5-column headers are the different a priori stratifications. So age, less than 1 year, greater than 1 year, duration less than 48 hours, greater than 48 hours, antibiotics versus no antibiotics in the preceding 14 days. And then some of the early analyses that we've done related to the etiologic agent. We looked at no pathogen identified, a bacterial pathogen identified, or a virus identified. It gets more complicated than that, and I'm not going to go into it too much right now. And then on the left here are the 7 different outcomes of interest. So moderate severe disease, repeat healthcare visits, health cold members becoming sick, time to last watery stool, time to last vomit episode, hours of work missed by parents, because that's a huge impact on your GDP, and a big reason, and the economic reason for giving probiotics is lost work and wages, and days of missed daycare. On all of these 7 outcomes across the 4 different subgroups identified in the columns, there were no significant differences between groups. So both of these studies are subject to several limitations. One is based on recall bias. So there's no hardcore evidence. We don't have biologics that we've analyzed yet. We do have data that will be going into later on. But basically, it's based on symptomatic recall of parents. We did contact them every 24 hours and very robustly, I think, did the best we could to accurately report that. We use composite outcome measures as our primary, which can be criticized because they're composite. However, I would argue that they're much more meaningful than individual outcome measures. But when we broke it down by looking at all the individual symptoms in these outcome measure scores, none of them were significant. And we ultimately only studied 2 products, 1 dose of each, and that's all that we studied. That's really where I restrict my conclusions to at this point in time. But based on the data that we have presented and analyzed so far in children presenting to an emergency department with acute gastroenteritis, probiotic administration does not prevent development of moderate severe disease within 14 days. And a huge thank you to Dr. David Schnatter, who really led the PCARN study, and then to all of our coordinators, site managers, program managers, our laboratory partners, and our funding agencies, so CIHR, as well as the NIH and ICHD. Thank you.
Okay, we're standing between you and lunch, and we're going to go a couple of minutes over into the lunch break for questions, but I want to ask our 3 speakers to come up, and I'm going to open up the session for clarifying questions, recognizing that we are going to have a panel opportunity this afternoon to hear from all of them again. So if you want to, are the microphones working at the table? Just push. You just need to push down when you're talking. So.
Mary Ellen Sanders from ISAP. Joseph, thank you. That was a very nice talk. And I wanted just some clarification. You mentioned some case studies about bacteremia and adverse effects from probiotic administration to premature infants. What is the overall number needed to harm for probiotic administration?
I could not tell you the answer to that. I don't know.
Is it fair to say that when you're considering an intervention that number needed to treat compared to the number needed to harm is a relevant comparison versus just, well, here's flaws in the particular data, and therefore because there's flaws, it's not perfect data, we're not going to act without considering the number needed to harm?
Yeah, I think that that these harms are very likely, and this is, again, opinion, are probably highly underrepresented, because so many of these babies do have problems. They develop sepsis. Much of the sepsis that we see in our preterm babies is gut-related, translocation of bacteria. So I think that a lot of the problems that we see are actually underrepresented with the use of probiotics. And again, that's my opinion.
So, Christian Lillis from the Peggy Lillis Foundation. My mother died of a community-acquired C. diff infection in April of 2010. And so listening to Dr. Merenstein in particular talk about this mishigas with the FDA and letters and such, I find that really troubling, and I would like to know what patients and caregivers can do in this space, because I often feel like Catherine Duff, my partner in crime, earlier said that she was the only person at the last one of these, and I think, the other person who represents the public and patients. And so these events happen. They happen in the Beltway. It's really, I didn't even learn about this until like 2 weeks ago. So how can we become more involved? Because I think that's the missing ingredient. You have patients fighting over cancer treatments. You have patients fighting for heart disease. When it comes to infectious disease, there's just no patients in these rooms ever. And I don't buy the whole I'm also a patient nonsense. So leave that at the door if you represent an industry. That's just crap. So I would like to know more about the probiotic stuff. I mean, if we can prevent these diseases, I think it's very important. And it's something that I personally find very frustrating, because we get asked about it all the time, and we know that there's evidence, but we don't know exactly where to direct somebody. So if you have any ideas, I welcome them.
Maybe asking the exact question, but I mean, as you can see, you had some high quality, hopefully, evidence presented and emerging, and I think the patients and the advocates need to advocate for independently funded studies, federal funding to look at it. I mean, I think Dan was highlighting the lack of, investigation and the barriers to conducting probiotic research. And so if it's left in the arms, I truthfully think it shouldn't be being led by industry and setting their own outcomes, the own measures, et cetera. I think these need to be federally funded studies, big, large, answering questions important to patients, caregivers, healthcare providers. To me, that's really where it needs to move. The problem I can comment more on the acute gastro world, is almost all those studies were funded by industry. We know there's a lot of negative studies that never got published. And so I'm going to get all the industry people very upset with me right now. But that's okay. I'm running for lunch somewhere. But I think truthfully, we need large studies, several thousand people to answer some of these questions. Rare outcomes such as C. diff and bad outcomes need very large studies to answer them. Numbers of 33 patients aren't going to tell you whether everybody with C. diff should get treated. Obviously, very sorry to hear about your loss. But to me, I think that is where the advocacy needs to come in.
Yeah, I guess I would add, I'm also sorry for your loss, and this is, in family medicine, there are often talks about how can we get people interested in these we call them sexy diseases, and it's difficult. And I have a question from your question to Dr. New. So if I have a preemie, is it ethical not to mention that there's these things called probiotics to them? You talked about all these issues, and you showed all the harms of the studies, but the Cochrane Review, and if I'm not mistaken, I think it was started by neonatologists, is pretty high standard. And if you have a preemie and you don't offer probiotics at your Hopkins or something, is it ethical not to even mention it to the family?
So tell me, what probiotic am I going to mention to the family?
I'm not a neonatologist.
Which one probiotic has been proven to be safe and effective against necrotizing?
So your answer is it's ethical not to mention that.
Yes.
Your answer is? Okay.
I think we'll get into a little more discussion of some of these issues this afternoon. I did want to thank, I'm missing where you went, but thank you so much for your comment, and I'm sorry for your loss. And I really do want to say that from an FDA perspective, we are very interested in the patient perspective. I would say that there are a couple of venues to be able to be involved in these issues. One of them, I think we heard this morning about providing feedback to the guidance documents, I think is always welcome. I think there's another outlet right now are the patient-focused drug development meetings that are being arranged through the agency, and certainly something that can be talked about, especially externally derived meetings where FDA members come to listen about these issues from a patient-focused drug development perspective. And then the third one that I know folks in the neonatology space are aware of, but consortia efforts like the International Neonatal Consortium, where stakeholders from all of the various groups, including industry, academia, patient advocacy groups, as well as regulators, all come together to talk about these issues and how to do the best studies moving forward in a pre-competitive space. So I think that there are opportunities out there. We really want to hear the patient voice. I wasn't supposed to inject myself here, but sorry, I did. Please.
Pinaki Panigrahi again from University of Nebraska Medical Center. I just have a comment. I asked this question in the previous session. I think I have the answer after I'm hearing to three of you. My concern now is how much, again, it is directed to Dr. New, like if we want to ask number needed to harm, we don't know that. Yes, we don't. Again, I agree fully with you that do I have a probiotic I will use for neck? Maybe we don't know because all of them are using different ones. So how do we get to a point where we can really answer these questions? And probably 20 years ago, there was a similar meeting in College Park, and it was told that the companies are self-regulating themselves. They want to put out good products. If the one that you cited, one case, that was an accident. I mean, that was a bad. It was manufactured in a sloppy way. I mean, that can happen anywhere, anytime. But to take that as a reason not to do probiotic research will not be fair. And only as long as we do more and more research, we will find out more and more adverse events. And if they are there. And if we don't do it, then we will never learn about it.
So I can't agree with you more that we have to do the right kind of research. And we have to have, safety The problem is with these probiotic agents that if we treat all preterm babies, and this is what we are talking about, all preterm babies with between 500 to 1,500 grams with a certain agent, and it is a tainted agent, we're going to have a major problem on our hands, not just one baby. We're going to have hundreds of babies that die at one time. And so I think we need to be very careful about the product that is available.
Pinaki, who just asked a question, is being very modest. I think he had a study published last year where 3,000 infants, the number needed to treat prevent death. Death, that's the best outcome you can get. I think it was 27. So I think he has a product that he has used.
Well, I didn't want to elaborate on that. I mean, that was a huge, very large study done overseas, all NIH funded, and the prior studies were also all NIH funded. If NIH had asked me to do it under IND, which is the case now, if I go to them and say that I want to study sepsis or NEC, they will ask for an IND. NIH will. But at that time, they didn't. And had it been the case, then I wouldn't have done a study in 4,600 babies. Which not only shows efficacy, it also shows at the least that the side effects are none. Literally, that it's an extremely safe probiotic to use. So that could have never been done.
So I think that we need to compare apples to apples and oranges to oranges here. And you were evaluating babies, infants. We are talking about preterm babies. And a preterm baby that is less than 1,000 grams is a different human than a baby that is 1,000 to 2,000 grams. And so we are talking about a very different individual than those babies or those infants you studied.
I fully agree with that. I mean, with a vulnerable population, with the kind of disease, of course, yes. But with the same token, if there is a blanket regulation made by the FDA that any time you do and a probiotic study in infants instead of a preemie, just a 1-year-old infant, or in adults, or in AD, or in any other disease for that matter. If it requires an IND, it's going to stop. I am a proponent of IND because I want to study one disease, one bacteria, very precise outcome. Yes, I will go for an IND myself. But if it is at some point, but to take that as a standard and demand that every study needs an IND, I think I differ in that.
So I think maybe we'll talk about a little bit some of these issues this afternoon. We're in the lunch session, so I'm going to say the last 2 questions, and if you might be able to make them brief and the responses brief, otherwise I'll be in trouble for not letting you go to lunch.
Good morning, and thank you for your presentations. My name is Deborah Topham with Knowledge Bank. I'm a regulatory consultant, and I also educate graduate students in food science and nutrition. And my question, comment, clarifying point is, how come the studies are poorly designed as far as how they characterize the background dietary intakes and the placebos? Because in any good drug study, you have, of course, the phases of work, but in a phase 1, you're maybe even controlling the diet, the environment. When you start throwing all of these kinds of organisms at the public at large, you bring in other intermediary effects. And I often find that even in the case of the Canadian study to the U.S. study, that was not necessarily a fair comparison between 2 treatment groups, let alone the ignorance of using any kind of background characterization of the delivery system with or without milk, with or without oligosaccharides. So comment, question, clarifying point, if you will.
I can try to tackle a little bit of that. So several things. So in terms of breastfeeding, we actually did look at exclusive breastfeeding. We actually had specific predefined mechanisms of delivery that were the ones recommended by the manufacturers of each of them. So they were in solutions that were deemed to be most compatible for viability of the organisms. I didn't go into all of that. The other element, for those people who do clinical trials, such as myself, trying to get in touch with caregivers on a daily basis, nearly impossible. Trying to get them to even tell you what they've done for the last day or how many stools, very hard. Try to get them to tell you what they fed their 18-month-old, impossible. They're at daycare. So getting that data is nearly impossible. The other is actually, and I would argue on the other hand, these are, and actually I think where research needs to move, pragmatic clinical trials, real world. So in the real world, people are eating whatever they're going to eat. They're going to feed their kids. We could try to tell them if there was a specific thing, but you actually need to look at effects of interventions in the real world on real patients and what's going on. And so that's really why, yes, it could have been interesting where there are subsets, but truthfully, trying to get that data is nearly impossible in these types of trials. And so it was not the focus of our effort post-randomization. Getting antibiotics was a big one, obviously. Getting breastfeeding status, daycare status, we focused on a bunch of those. But dietary history intakes, good luck.
All right, last question, please.
Oriana Tsarovsky with Johnson & Johnson. You actually answered along the lines of my question, and you mostly answered it, 90% of it. I was just a little piece I'm missing. How did you go about selecting the probiotics that you would study for the indication that you were studying and the organisms? I think you said lactobacillus. What was the process? If you could share.
How long do I have? So really, if you go for so in short, actually, it was a several-step pragmatic process. So initially started working in the Canadian side with a product made by Lauma Health Solutions, or Lauma Health Inc., which is a Canadian company, also subsidiary from France, where one of my longtime collaborators had been working and studying this product in vitro. I'm working then in animal models and demonstrating efficacy and immunomodulatory properties and cytokine mediation and improved benefits in the animal models. They had some human data as well showing evidence of benefit. And so we went to that. We then did a dose finding study that we've published in Clinical Pediatrics several years back, where we actually looked at the higher dose. So they had their recommended dose. We then said, because I didn't want to have too small a dose, we then doubled the dose and actually did a study looking at safety of that, and we found that the higher dose was safe, no adverse events. So we used the higher dose because I didn't want to be criticized as having too high a dose in that study. And so that's how we moved forward on the Canadian side. We then went to and if you want to get into INDs, we then submitted a study in the US funded by NICHD. We unfortunately could not get an IND for the product I studied in Canada. From the US, we couldn't get the purity piece. Oh, well, I don't make it, the manufacturer. They had too much contamination, couldn't get an IND. We then decided, okay, can we just tweak this and look at who else? And we looked at the number one player in the market, most commonly used, and with a fair amount of evidence of benefit, which was LGG. And then they also held an IND on their master file, and we were able to use their master file to obtain an IND to do the study that we then conducted. And so that's how we ended up using LGG. Truthfully, we didn't start out that way. I wanted to study the other product, both countries. Actually, probably to a certain degree, I'm actually happier at the end that we studied two different products, two different countries. And so I actually think it was a good resolution of the problem. But there are issues of, we couldn't get the IND to study it.
So I want to thank all three of our speakers from this morning. It's 12 minutes after. We would love to have you back by 1 o'clock, please, so that we stay on time.
As Susie pointed out earlier, this is the second half of this session, looking at live microbiome-based products for disease indications. This part of the session is really going to focus on C. diff, both from use of commercial products to discussion of FMT and then CMC considerations for this type of product in the context of C. diff. So we'll go ahead and get started. I'll introduce the first speaker is Krishna Rao from the University of Michigan, who's going to talk to us about commercially available products for prevention of C. diff.
Thanks, Paul. I get to be the guy to talk about diarrhea right after lunch, but I'll do my best to keep this civil. So I only have one disclosure. I'm a co-I on a grant from Merck, not related to this particular topic. So I want to talk a little bit briefly about C. difficile infection. Vince Young gave a nice overview this morning, so I won't touch on that too much. I do want to talk about some of the basic science and mechanisms behind probiotic development for C. difficile specifically, and I think it'll be clear by the end of the talk why I wanted to spend some time on that. And then we'll delve into the clinical literature and talk about future directions that I think may be helpful. So briefly on C. difficile infection, so as we know, it's a gram-positive spore-forming bacillus. I won't go through all these numbers, but I will point out that it's striking. These numbers here that you see are very high, and they're for the United States alone. So clearly this is a major problem. If we're having nearly 500,000 people a year getting this every single year, what can we do to actually prevent this? And again, Vince showed a similar slide to this, so I won't go into too much detail. I will point out the important part that we need for our discussion, which is that C. difficile is a spore-forming organism, and so you do need germination of the spores to a vegetative state, and then production of toxin from those vegetative cells in order to actually initiate disease. Notably, though, this person who is currently symptomatic in red doesn't go to green once they get treated immediately. It takes some time for that microbiome to recover, and if they get re-exposed or they have another hit or an insult during the susceptible period, they can end up in this cycle that sometimes only fecal transplant or other measures can fix, and we'll hear more about that in the next talk. So how do these even work? And I think I get this question all the time from my patients, so it's only fair that we ask ourselves this question too, which is it even feasible to think that these drugs might work? A lot of them will say, Doc, I'm taking antibiotics right now at the same time you're giving me this probiotic. Isn't it just going to kill the probiotic you're giving me? And it does reassure them a little bit that we've thought about this and we've thought through this, and that many probiotics can easily make it to the lower gut. Lactobacillus famously is a lover of acid, tolerates low pH just fine. Lactobacillus and bifidobacterium in the setting of C. difficile literature can colonize the gut, and we see it persisting even after it's been its administration has been withdrawn. These are actually occasionally a little bit too invasive, and we sometimes see them in extraintestinal sites. And they're often selected to be resistant to certain antibiotics. We give lactobacillus, for example, with vancomycin because it's resistant to that, even though it's another antibiotic. And what's important is also that these are very strain and person dependent. And half of patients don't colonize at all. Yeah. Like Vince, I actually read this study in Cell that just came out, unlike the press reports, apparently. And they don't say that probiotics are useless, but they do make some interesting claims that there are very strain and person-dependent findings that need to be accounted for. Again, this is just kind of a general overview. We can look at and think about these mechanisms from the more widespread ones, such as colonization resistance and production of short-chain fatty acids and secondary bile acid metabolism, and the more rare and very strain-specific ones of specific immunologic claims or neurologic claims. But for C. difficile infection, broadly speaking, we have a few areas that we can target. So one is this bile acid hypothesis that we've talked a little bit about. Clostridium scindens is one that we'll hear a little bit about that can target that pathway by promoting the conjugation of primary to secondary bile acids, which are inhibitory to vegetative C. difficile and sporulation. You can actually have these probiotics producing antibacterial compounds that actually can be cidal or static. In this case, this is an example of Lactobacillus reuteri, which is able to do this. Or they can make compounds that inhibit the toxin activity, in particular proteases such as Saccharomyces or kefir. And they can have very nonspecific general effects too. So some of these will increase mucin production, they'll alter local pH, inflammation, increase production of IgA. And just to pick out a couple of mechanisms to spotlight, so one is bile acids. There's a lot of data suggesting that bile acids are important in C. difficile infection pathogenesis. Vince showed one study earlier today. Here's another one where we looked at specifically fecal transplant patients who were successfully treated, had a cure of their C. difficile infection. And whether you look at short-chain fatty acids or secondary bile acids, you see an increase in these patient populations. Sometimes that's a transient bump. In many cases, it's a more persistent bump. But the question remains, what is the direction of causality here? There are some nice mechanistic studies looking at probiotics and C. difficile that are coming out and have come out actually in just the last few years. I'll draw your attention to this date because this is going to be another theme that's important. This is in 2015, just 3 years ago. Eric Paymer's study, Vince alluded to this briefly, but they looked at mice who were treated with antibiotics and those who weren't, assessed the microbiome, found that there were a couple of taxa that were highly differentiating those two populations of mice. And in particular, Clostridium scindens and 3 other taxa seemed to confer resistance to C. difficile. And either alone, or if you gave the C. scindens with other bacteria as well that they identified, looking at both the colonization levels of C. difficile as well as the survival of the mice, they saw a significant effect. And in fact, the consortia actually completely abrogated the effect of C. difficile in these mice. Going on another spotlighted pathway, let's look at bacteriocins. So L. reuteri makes Reuterin, and that's one pathway that has also very recently, again, look at this date, 2017, been shown to be efficacious in C. difficile mouse models. Here they administered L. reuteri along with its substrate glycerin in a bioreactor model, and they found that that actually inhibited C. difficile almost several logs, and that the populations differed when you assess their microbiome. And again, this is only in the presence of the substrate that Lactobacillus reuteri actually needs to make Reuterin. Now, what about the clinical literature? And I would say that broadly speaking, you can look at individual studies or you can look at meta-analyses. And when you look at individual studies, you see a lot of really very poor quality evidence to a large degree. So many of these are uncontrolled studies. There's not one large definitive RCT yet that's been done for this topic. And there's a lot of heterogeneity. There's heterogeneity at the strain level, at the doses level, at the regimens, and also in the patients, because not all patients come into C. difficile with the same level of risk. And it's important to define that population specifically and strategically when you're designing your studies. So what do the individual data show? And so there's one study that has been mentioned, this Placide trial, that was a couple of years back in 2013. And that was a large negative study for the use of probiotics in C. difficile. Now, it was negative, but it's important to mention that this was a study of antibiotic-associated diarrhea, not C. difficile, so C. diff wasn't their primary outcome. Secondly, only a percent of patients in that study actually had this outcome of C. difficile infection. So even if they had an effect size of 50%, it would be really hard to power a study to detect something like that. So again, it wasn't focused on a high-risk population, and many of these studies were not. Many of these studies have very ill-defined inclusion-exclusion criteria. In fact, many of them allow people to eat yogurt at the same time that they're on their quote-unquote assigned study probiotic. There's other confounders. There's one study that actually noticed a pretty decent effect size for C. difficile, but when you read the study, they literally moved to a new hospital during the study, and so of course their rates went down. So the preponderance of this evidence, I think I agree with one of our earlier speakers that it has cooled the interest in probiotics specific for this indication. In particular, this Allen study really did cool the interest in studying probiotics for C. difficile, and the current guidelines don't recommend them, as has been pointed out. Now, what about meta-analyses? And there have been several conducted over the years. This is by far not an exhaustive list. I don't think I even include the Cochrane study on here, the recent one from last year. But these are some of the 3 that are commonly recommended. Now, look at the dates for these. 2012, 2015, 2016. That's actually flipped from the way you would normally think about it. A lot of the mechanistic studies that I highlighted were more recent ones. And these studies looking at efficacy in the clinical literature are older ones. And this has been pointed out about meta-analyses before, so maybe I'll point it out a different way and use a different analogy. My meta-analysis instructor in school liked to use a different analogy to say that a lot of times when you combine meta-analyses, we think that we're mixing a whole bunch of turds and out comes a pot of gold. But really what sometimes happens is you mix a bunch of turds and you get turd soup. And I fully acknowledge that. However, I think there is a signal here that we should not ignore and that needs some elucidating. So one of the problems with a lot of these prior meta-analyses is that, again, these individual studies, there's a lot of them, many of them are poor quality evidence. So which ones you include matters. How you include them and how you extract the data matters a lot. And a lot of these didn't actually follow PRISMA best practice guidelines, which is why there's heterogeneity even in the meta-analyses themselves. sometimes they study different populations. So broadly speaking, what I've seen is that if you have meta-analyses that have these broad criteria that take a lot of studies, they have weaker effect sizes. They don't tend to demonstrate statistical significance. There's more heterogeneity in those studies. And when you have narrower criteria, not surprisingly, you actually get better results. And when you focus on a high-risk population and only include, for example, RCTs with placebo-controlled, you actually get a little bit of a signal that you're able to tease apart. This, I think, not to pick out just one meta-analysis, but I think this is one that's really clarified what's going on here, for me a little bit at least. So this was done last year in gastroenterology, and they point out that there's a lot of heterogeneous data in the past. Here they focused on RCTs, so controlled studies, in hospitalized patients on antibiotics, so among the highest risk population that we have. They rigorously adhered to the PRISMA guidelines. In fact, they even went so far as a lot of these studies have attrition bias. Some of the prior meta-analyses didn't assess for attrition bias. This one did. They went so far in their sensitivity analysis, however, to actually assume that in the patients that were lost to follow-up in these studies, the rate of C. diff was 5 times higher than in the other population. And even when they made those very conservative assumptions, and actually they had very low heterogeneity. Their calculated I-squared was 0% for this meta-analysis. But they did a meta-regression anyway and used mixed methods anyway. And even with very conservative methods, with over 6,000 patients included among 19 different trials, you can just look at this forest plot. And if you would guess that the heterogeneity is low, you'd be right. There's a significant effect here. And it's, you know, it's about 40% relative risk, or a number needed to treat of about 43. And even with the very conservative 5-fold increased incidence in the untreated group with the missing data, they still see an effect size of a 40% reduction, a relative risk of 0.6, and a number needed to treat of 63. And, you know, as Dr. Merenstein mentioned, you know, this is definitely within the acceptable range of what we do clinically. Just to give you some thing to anchor your thoughts a little bit, we routinely prophylax against venous thromboembolic disease among inpatients who are admitted who meet certain risk criteria. And the number needed to treat there is in the 250s. And the number needed to harm is quite a bit more than with probiotics. But we're not using probiotics. And the question is why? Before we get to that, real quickly, the other place you can look at probiotics for C. difficile is not in preventing the primary C. diff, but preventing a secondary C. diff episode, or that recurrence cycle that we talked about. And the punchline is here is actually this is much less robust in terms of literature, and it's not clear that there is much of a signal here, at least with single agent probiotics. We'll hear about FMT shortly. So why aren't we using probiotics for C. diff? I just told you that there does seem to be a little bit of a signal, even in a really well-conducted meta-analysis with very rigorous adherence to the best practice guidelines. So maybe they aren't safe. Maybe that's one concern. They're officially generally regarded as safe. There are some symptoms people report, IBS-like symptoms that can occur that are actually fairly common, and this recent paper in Cell that we had talked about did highlight some of those. However, there are some major concerning things that we have to be aware of. So there are patients who are treated with Saccharomyces formulations, lactobacillus formulations that have had bacteremia and endocarditis respectively reported with those formulations. And yes, they went back to actually verify that the strain in the probiotic was the one that was isolated clinically. There's this other famous study in inpatients in the ICU that were given probiotics for their pancreatitis, where there was actually increased mortality in the probiotic arm. Not sure what that's about, but that definitely has cooled a little bit of the interest in this. And also notable is that many of these trials that I just talked about in these meta-analyses, they excluded immunocompromised patients, IBD patients, ICU patients, those patients who are among the most highest risk for getting C. diff in the first place. And so it's hard to generalize to some of these other populations where we'd actually want to use these agents. Maybe we're not using them because there's lots of major evidence gaps that need to be filled. And I think that's part of it. And there's some examples here, things like what are the interaction between specific classes of antibiotics and probiotics on C. diff risk? To what degree do dietary probiotic use impact the results of prior RCTs? And ways that we can maybe navigate some of those discrepancies and gaps. Maybe there's too much heterogeneity, and we've talked a little bit about this, but in the past, it seems like we've almost been moving bedside to bench. And only now, in the last few years, are we finally kind of developing, I think, the rigorous preclinical research to say, hey, these are the strains that actually are showing efficacy in these really nice models. Now, the challenge, of course, of that is translating these models to actual humans, and that can be challenging. Bioreactors are a little bit easier to do than with mice because you can actually use the human microbiome in a bioreactor. And there are humanized mouse models, but there's all kinds of issues with that. But those are challenges that I will acknowledge. However, what's really encouraging is that the strains that we're seeing in these newer preclinical studies aren't largely a lot of the same strains that we've seen in these trials before. So I think it's encouraging that there's a signal here and that we may be able to kind of make some progress. However, that's going to be challenging for this other reason, another recent study looking and really showing that there's not only strain specificity, but disease specificity. So whether you're talking about a specific strain and you look at different disease processes, you're all over the place in terms of the efficacy. Or if you look at a specific disease process, C. difficile, as we've seen, you're all over the place in terms of what strain is actually going to make a difference here. So what are the future directions? Where do we go from here? What advice could I have? And one thing that I think is happening and that needs to happen more is we really need to go back to the bench and I'm making an argument here that we need to have a rational mechanistic approach to how we actually design these probiotics. And these are examples of all the mechanisms that are currently being studied and have been studied in the last couple of years and have yet to make it into the clinical literature. And I also think that on the clinical literature side, we need to have very strict, well-defined inclusion-exclusion criteria. Don't let your subjects eat yogurt. I think that clouds the picture. Actually have good randomized placebo-controlled trials. We need to power it appropriately for C. diff. And one of the other recent developments, not to plug my own research too much, is we're actually starting to have electronic health records being able to be used in machine learning algorithms to risk stratify people. So this is a study we published last year where we can actually risk stratify people and actually predict an episode of C. diff 4 to 5 days before it happens in the inpatient setting. Using a risk model like that and randomizing those patients to an intervention may be a lot more fruitful than prior approaches. So conclusions, I think no argument here that C. diff prevention remains a major need. The rest of this is opinion, but I think the current clinical evidence does support that there's some role for benefit of probiotics in C. difficile. The reason I can't recommend this, though, is it's one thing to say that there might be a benefit here, that it looks like there is a benefit. It's another thing to make a very specific falsifiable scientific hypothesis, another thing to make a very specific claim about here's the strain and dose that a patient should take for a benefit, and we aren't at that point yet. In the interest of time, I'll stand there.
Thank you, Christian. We're going to, like we did for the last panel, we're going to do questions for all the speakers after. Our next speaker is Colleen Kelly from Brown University, who's going to talk to us about clinical evidence on FMT for C. diff.
Thank you very much. Thank you for the invitation to speak today. So I was asked to summarize the evidence that we have from randomized controlled trials for C. diff infection. I want to just sort of start off with my early experience with what we called fecal bacterial therapy at the time. And in my first year, I treated 2 patients just like this, a 61-year-old woman who had had 6 intensive care unit admissions over a 12-month period. During some of those, almost like lost her colon, almost went to surgery, almost died. Each one was very dramatic. The second was a young girl who was 19 who got a dose of clindamycin getting for some dental work. And this was during her first year of college. She then developed recurrent C. difficile episodes. She had to quit the soccer team at her college and take a semester off of school. And both of these patients were treated with repeated courses of vancomycin, metronidazole, rifaximin, probiotics, and to no avail. And both resolved their C. diff infection with a single FMT. And in fact, in the first 2 years of fecal bacterial therapy at our practice, 24 of the 26 initial patients I treated did not develop a further C. diff occurrence after that first FMT. And by 2011, we weren't calling it fecal bacterial therapy anymore, this terminology FMT, and as Vince Young spoke this morning, we're transferring these entire communities of microorganisms from one person to another to increase the diversity and repopulate some of those beneficial anaerobes. And at that time, when I was having my early experience with FMT, others also were seeing that this was working. And this is the results of a paper published by Zane Kassam around that time that just demonstrated that we were seeing really high cure rates in open label clinical trials and case reports, close to 90% overall, with some evidence that it may be more efficacious when given from below than from above. But the real game changer was in 2013 when this Dutch group published the first randomized controlled trial in the New England Journal of Medicine. It was a relatively small trial, 42 patients, who had at least one C. difficile recurrence. And they were randomized to one of 3 arms. either a short course of vancomycin followed by a bowel lavage, like a bowel prep, and infusion of 500 cc's of donor stool by a nasoduodenal tube. The other two groups either got a standard course of vancomycin, 14 days, with or without that bowel lavage. And that study was actually terminated at the interim analysis because FMT was so effective. 81% resolved their C. diff after a single FMT, and then the couple that needed to get retreated, it was up to close to 94%, compared to 20 to 30% in the vancomycin groups. And others have also looked in a randomized way at FMT versus the standard of care, which is vancomycin taper. Camerota and colleagues in 2015 reported on a similarly sized patient population with recurrent C. diff. Their intervention was, again, a short course of vancomycin, followed by FMT delivered by colonoscopy. If they saw pseudomembranes, which are indicative of more severe disease, then they would repeat and do potentially more than one FMT. But most patients in the trial only got a single FMT. This was compared to a group that just got a standard course of 10 days of vancomycin and then a pulse taper dosing over the subsequent 3 weeks. That study was actually also stopped early for superior efficacy of FMT. You can see the numbers there. Conversely, Susie Hoda and colleagues in Canada more recently published a study of patients with recurrent C. diff who were treated with either the standard course of vancomycin followed by a taper, or instead of tapering the vancomycin, they were given a single FMT by enema. And in that study, that was also stopped early, but for futility, in that that single enema did not appear effective at resolving the recurrent C. diff cycle in those patients. So I was the PI for this clinical trial, which was published in 2016, and we did our best to find a placebo, which in this case was autologous FMT. So patients would submit their own stool to us, and then I picked a card, and they either got a fresh donor FMT by colonoscopy, or they were reinfused with their own stool. It's important to know that they were all treated with at least a standard course of vancomycin, 10 to 14 days, and symptoms had resolved prior to getting that FMT. Vancomycin was stopped 3 days prior to the procedure, and they were infused with either their own or a donor stool. Overall, 91% of patients resolved with FMT versus 63% who got the placebo. And you can tell there were some differences between sites. And if you just let me, I'm going to leave that till later. I promise you I will address it. But I do want to say there were no SAEs in the FMT related group, no related SAEs related to FMT. And I do want to point out that we were limited by the IND. This trial did not include patients over age 75 or patients who were immunocompromised. Others have compared FMT to FMT, comparing different delivery modalities. So Youngster and Libby Hohen published in Clinical Infectious Disease in 2014, looking at recurrent C. diff patients. They were treated with 41 grams of stool that was frozen and thawed and administered either by nasogastric tube or by colonoscopy, and there really weren't differences between those groups in efficacy. And more recently, Dina Kao in Canada randomized patients who had had at least 3 episodes of recurrent C. diff to frozen capsules, which were then thawed and administered, or the same amount of stool, 100 grams administered by colonoscopy. They actually found them to be equally effective, 96% with the first dose, either by capsule or colonoscopy, though the capsules were rated as cheaper and preferable to patients overall. Other groups have compared dosage formulations. Fresh FMT was compared to frozen in a large trial by Christine Lee, also conducted in Canada. This was published in JAMA a couple of years ago. They had over 200 patients who had had at least one recurrence. So I do want to point out that this study, patients were kind of interrupted on much earlier in the C. diff cycle. It was after a single recurrence versus 3 or more. And many in the study had only had a single recurrence, in fact, 92% of those patients. Their overall efficacy with a single enema was 62%, and it didn't appear to differ whether they got stool that had been frozen or that was fresh and administered that way. But you can see to get up to those 90-some% numbers, you needed 3 to 5 enema FMTs. Another group from Baylor publishes this past year looking at fresh versus frozen versus lyophilized formulations of FMT. This was about 50 grams of stool, so it was the similar dose, but the preparation method differed, and it really fresh 100% resolved, 83% after getting previously frozen stool, and 78% after lyophilized, and the differences were not significant. Because most of these studies are small and really underpowered to detect meaningful differences there. So this, as you know, we heard a little bit of, we're supposed to not necessarily trust systematic reviews and meta-analyses, but Paul Moietti did a good job with this one, published last year, looking at summarizing the 5 big randomized control trials for FMT. And those included 284 patients. And I want to point out the number needed to treat was 3. That's huge. There was significant heterogeneity across these studies because of different modes of delivery and doses. But despite that, and looking through all these with grade criteria, it was determined with moderate quality of evidence, to be effective. And importantly, in all those patients, there were no FMT-related severe adverse events. And this is something I've also seen in my own practice. I'm up to nearly 300 FMTs at this point, 10 years in, and I have to date not seen a definite FMT-related complication. So they are certainly, I'm sure they occur, but they are rare. And since 2013, American Gastro Society guidelines and European guidelines have promoted FMT after patients have failed standard treatments with pulsed and tapered vancomycin. And more recently, the IDSA guidelines, which were published last year, also support using FMT for patients with multiply recurrent C. diff, used with strong recommendation, despite the moderate quality of evidence. So, here's something that works. We know it works. Handing the ball to industry, we're all as clinicians looking for something easier than putting fresh stool in people. But the results so far from the industry-funded trials in this population have been disappointing. Seres Health in 2016 reported in their capsule study, and I do want to, there's a little caveat that Seres product was not FMT per se. It was derived from human stool, though it was ethanol treated to kill off vegetative forms, and it was basically Clostridial spores. But there was no significant differences in those who received the placebo and those that received the Seres capsules. Rebiotics presented in abstract form and also more recently published results of their phase 2 trial comparing placebo to a single FMT or 2 FMTs. And interestingly, 2 doses of FMT was not more effective than placebo, though a single dose of FMT was. However, their a priori endpoint was resolution with 2 FMT enemas, and therefore their study was also not significant. So lessons learned from all of this. Delivery methods certainly matters. Single-dose enemas are less effective, and we see this from a couple RCTs now. Lee's study, Susie Hoda in Canada, and then the Rebiotics results, very similar in efficacy to that single-dose FMT. Fortunately, freezing doesn't impact efficacy, so we don't have to worry about keeping fresh stool around. And also, fortunately, capsules and colonoscopic FMT appear equally effective, so we don't necessarily have to instrument these patients and put them through the procedural risks. And why are we having, these difficulties? And I think one of the things are the diagnostic challenges around C. diff. So about 10 years ago, everyone went to the PCR because it's more sensitive, and we weren't going to miss any cases. The problem is we pick up a lot of colonized people, and colonization rates are high, up to 15% of healthy adults. I think that's a little high. I think it's closer to like 3%, but this was just from some series. Hospital inpatients, up to 29%, and residents of long-term care facilities, up to 50% of these people are going to test positive for C. diff, the organism, without actually having C. difficile infection being sick. The other thing that we see after C. diff is post-infectious IBS that occurs in close to 25% of people, where they may go on for a period of time to have loose stools and diarrhea on and off and some bloating and discomfort, and that may, in the setting of colonization, be mistaken for a recurrence and called that in a clinical trial and treated as such. So don't rely on PCR for diagnosis in these studies, and enroll from highly experienced FMT centers because we're seeing this all the time. In our center, we published 25% of patients referred to me. It was subsequent, like 100 people consecutively referred, and a quarter of them actually did not have recurrent CDI. I didn't need to give them another treatment for their C. diff, and I found all kinds of things. And that's just the list of things that I found. I found undiagnosed Crohn's disease, celiac disease, lactose intolerant, 3 cases of factitious diarrhea, people who just like to come to the hospital and get attention. One of the things that we found, interestingly, that there was an inverse relationship between age and these alternative diagnoses. The younger people in these trials are less likely to actually have real true C. diff compared to older patients. And I think John's going to talk more next a little bit about the series data, and I think that they did see more efficacy in the older groups in their paper. But importantly, I think people were cured, and I think that that's kind of what I think happened with a lot of the patients at the New York site in our study. Some of them had been on continuous courses of vancomycin for a very long time, waiting to see Dr. Brandt. He did not necessarily stop that vancomycin. He said, okay, I'll enroll you in this study, and then we'll stop the vanco 3 days before and give you an FMT. But I think that a lot of them were probably already cured. One had been on a continuous course of vancomycin for 148 weeks. That was an outlier, though. So I think keeping these things in mind with your study design and how long patients should be treated with vancomycin prior to being enrolled in an FMT trial is important. So to summarize here, I think FMT works for C. diff. We just don't know exactly how well yet, but I'm certain that it works. It also appears to be very, very safe. And we need to really take into consideration these things when we're designing clinical trials. Who are the most appropriate patients to enroll? At what point in the cycle of recurrence? Should it be after a first recurrence or a second or a third? Should we be looking at FMT for patients with severe complicated C. diff who are potentially going to lose their colon or die? Or even as treatment for primary C. diff? And there's been a couple of papers recently suggesting that maybe instead of an initial course of vancomycin or Flagyl, giving a dose of FMT. And then what should be the best endpoints and for how long after, should we be looking at 8 weeks, 12 weeks, diarrhea-free, of course, like the PCR versus the enzyme immunoassay. So all of these things really should be important to those of you who are in the audience who are looking to design a pill for us to use. So thank you very much.
Thanks, Colleen. So we'll move on to our final speaker in the session now. John Onnes is going to come talk to us. John's from Seres Therapeutics. He's going to talk about CMC considerations for microbiome-based products.
Thank you very much, Paul. So now for something completely different, as they say. So the benefit of going late in the afternoon is that a lot of your intro slides have already been covered by people in various forms, and so you can kind of go through them. So the microbiome, as an interesting subject for a pharmaceutical development, is a fairly recent sort of evolution. It's paralleling, in my mind, a lot of what went on for stem cells about 20, 25 years ago, where people first view them as tools to understand disease, next as targets to manipulate, and then only later to become therapies. And you can see that in sort of the applications that people have started to develop. So microbes as tools, obviously, as Vince Young pointed out, people want to understand how their drugs are metabolized, but then also to try to maybe suss out exactly which compounds bacteria are secreting to create new drugs. This is an approach that's kind of favored by the larger, more conservative players in industry. Microbes as targets, I think everybody would like to have surgical strike kind of antibiotics that only get the pathogen of interest and don't have the collateral damage of the broad spectrum antibiotics that we currently have. And then there's a fair amount of research in prebiotics. If you look for interventional studies in clinicaltrials.gov, you'll find almost 300 studies on prebiotics attempting to manipulate levels of microbiome components. It's not obvious to me, I think, that there's a miracle food that you can eat that's going to cure you of disease, but there may be certainly concepts like synbiotics that were talked about just a second ago that could be valid. What we're here to talk about, of course, is microbes as therapies, where we're trying to not so much do antibiotic-like maneuvers of loss of function, but really have gain of function, or in some cases modulation of function, for example, for immune system, by replacing or altering the microbiome. I don't know that I need to really belabor the different types of microbial therapies. Clearly, we've got two different sets of people here, the traditional probiotics and then the newer area of gut commensals. I think the traditional probiotics, these are basically dietarily acquired organisms. You get them with dairy products, fermented foods, and such, by and large, for the strains. I'll disagree with Dan Merenstein in that every time I talk, I'll update the clinicaltrials.gov search for interventional studies, and it keeps growing and growing and growing. It's over 1,100 studies, over 110 in the past year that I've found. So I think there's a robust amount of research on it, but I think the results by and large have been seen to be modest for various reasons that we've heard this morning and this afternoon. I find it interesting, Bob Durkin didn't have a kind of an equivalent sort of metric, but since their European equivalent, the European Food Safety Authority, put in place a rule that said basically you can't make health claims unless you submit a scientific dossier and you prove your claim. They've reviewed over 300 of these things, and they've only approved one, and that was for a fairly obvious secretion of cobalamin, which is known to occur by bacteria. So there's not a heck of a lot of evidence. I also don't need to probably talk about safety so much as to, because we've talked about that a bit. I don't know how many of you caught Bob's subliminal drawing of a line, though, where he said there are something like 500 inspections and 7,000 production facilities. Work that out. It's about one inspection every 14 years. Would it surprise you if things get a little sloppy in the interim? I think not. Gut organisms, as we've heard, have gotten a lot of interest since the Human Microbiome Project came along, and the confluence of the C. difficile epidemic and the advent of FMT as a potential curative for that. Clearly, FMT it's a good initial stab. It's doing great things for a lot of people. I think the efficacy and safety, as Colleen just described, is still a bit ambiguous and could be further refined. And of course, for any of these products that are made on gut commensals, I think it's as yet TBD that they actually, they put the proof in the pudding too for safety and efficacy. But I think it goes without saying that where we would all like to go is to get to designed microbiome therapeutics, which would be either single strains or consortia of strains of purified organisms for the GI tract. In some instances, such as our colleagues here from Synlogic, they might be genetically engineered for heterologous gene expression. I don't think I need to go to this slide very much either, because Vince Young described how basically the microbiome works as an ecology, how it has steady states unless they're disrupted by certain events such as pathogen infections or broad-spectrum antibiotic use. I think the interesting thing that came out of the Human Microbiome Project is that whereas if you look at the strains of microbiological diversity, you see that everybody in this room would have a vastly different microbiome. But if you look at the gene content as a functional diversity, it's fairly consistent. And so that gave companies like Seres hope that you could actually potentially develop drugs that don't have to be, say, tailored to individual microbiomes, that you can really simply try to design things that have the proper function and replace that function. As I mentioned in the last slide, so whereas traditional probiotics tend to have very short half-lives, they wash out pretty much as soon as you stop dosing them. On the other hand, the gut commensals tend to stay persistent, and that's been seen in trials of fecal transplantation and other trials as well. And here, basically, the idea is you take a disrupted disease ecology and you're going to replace it and stabilize to some normal ecology. Per this cartoon here, where you'll have microbes from your product that will engraft, and then they'll be augmented by other microbes that come along, and you get rid of your disease microbes such as C. difficile. This upper right panel here is data of engraftment from a trial that Seres did in ulcerative colitis with a product called SER-287, which is a spore composition. And basically what you can see is that over the dosing period, depending on the regimen we gave, whether it's a weekly or daily dose, you get engraftment that starts to plateau out about day 10 and through the end of dosing. And so you can create a persistent change. The interesting thing is after you stop dosing, a month later you still have a persistence of the microbes. So they seem to have engrafted longer term. And that engraftment appears to change the structure of the microbiome. This is a principal components analysis plot that simply shows subjects who went into remission for ulcerative colitis versus those who didn't. You can see that you've got a distinct difference in the structure of the microbiome. So these are the kinds of things we're trying to do at Ceres, is to develop drugs in that vein that are going to be commensal microbes, consortia of them, to alter disease. And our paradigm really is to use proof of concept consortia, probe consortia like FMT or other natural consortia, basically take the results from studies of those interventions, which are really the gold standard, rather than using observational studies, try to find organisms that seem to have impact, that are present in your drug and are associated with success of your trial, identify the metabolites that are associated with those organisms or those changes, and then try to map those to pathways that are expressed by the organisms, and then devise novel consortia that you can use to develop into drugs, right? That's probably the novel part. More conventional is doing the screening for your drug candidates in vitro and in vivo, and really pulling from large strain libraries to construct those candidates. And then the next novel bit is the manufacturing, which is what I'll talk about from here on. So there's several unique features to manufacturing consortia of gut commensal microbes, right? These are not your grandfather's industrial microorganisms. They're not Chinese hamster ovary cells. They're not E. coli, recombinants. They're not Saccharomyces. Most of these have never been in any kind of a GMP production. They're generally strict anaerobes, quite often not aerotolerant, so you have to keep them isolated from oxygen. And many of them are spore formers, which is a unique feature. So when we're making consortia of bugs, we have to deal with a multiplicity of organisms in the product. Making all of them, as you heard from Sheila, you need to basically be able to count them all. You need to make sure that you've got their culture behaviors down. You need to preserve them all, make sure that they all survive your formulation and they get to the site of the gut where you want to deliver them, and then you have to be able to count them. And then last but not least, you need to be able to manufacture them in a GMP fashion. Right? So just going through those, the first bit is to actually be able to grow gut microbes. And just like the slides that you'll see with throwaways of 10 times as many microbes as human cells and so forth, you'll also hear throwaway statements like 99% of the human gut microbes are uncultivatable or haven't been cultivated. Well, there it is for lack of trying, basically. Coming out of the Human Microbiome Project, there was a list of most wanted organisms. Ceres has about 75% of those most wanted organisms in our strain libraries. The problem from a CMC production perspective is that quite often they are isolated in things that you wouldn't normally take into production, things like brain heart infusion augers, rumen fluid media, blood augers, and so forth. So the trick for us CMC guys and gals is to take that strain of interest and be able to grow it in GMP acceptable media and do that in an efficient fashion. So we have a multi-stage screen that we use to basically get away from complex and ill-defined and perhaps undesirable components and get to something that's much better defined in an optimized process. And the trick is to have set up screening paradigms that make use of high-throughput robotics, that make use of bioinformatics and omics technologies in order to be able to do this with a modicum of manpower application so you don't burn yourself out to death. I'll also note that you not only need to be able to adapt things and grow them in culture, but you also probably need to be able to optimize phenotypes. So in Ceres' case, we're interested in a lot of firmicutes to date, and so we're interested specifically in sporulation. And optimizing that sporulation, especially in the GMP media, can be a complex endeavor. Nevertheless, we've managed to have pretty good success at doing this and getting to productivities in our fermentations that are acceptable for future use. The next thing I mentioned is formulation and delivery. Similar problems here as you've got for the fermentation. You need to be able to preserve a range of phenotypes, right? And here, basically, your formulations and chemistry and processing has to be acceptable for gram negatives, for gram positives, a range of different types of organisms, cocci and bacillus and so forth. And so you need similar sorts of platform screening methodologies, which I'm not going to go through here. But suffice to say, we can take some very sensitive Clostridial strains and do a lot better than what you can find for, say, commercial buffers. So this upper right panel simply shows losses of titer through freezing, drying, 1 week, and 4 weeks at accelerated temperatures. We can substantially knock that down with pretty straightforward optimization. And then lastly, before that, I'll just note that I think it's axiomatic that once you get away from spore phenotype organisms, you're going to have to go to dried state. It will, other than perhaps some products that could be frozen as liquids, such as FMT, your ideal product is going to be an oral capsule. You want to be able to put that on a shelf, right? And so you're going to be dealing with dried powders. And those can be challenging to handle because now you have to prevent basically aerosolization of the powders. You want to prevent exposure of powders to moisture, to oxygen, and so forth. So that can be tricky to handle. And I'll just opine that it'll be a miracle if people get actually room temperature stable microbiome therapeutics in general. My guess is that most of them will be refrigerated cold chain products, excepting the spore products. Lastly, delivery. Of course, I think it would not be lost on anybody here that, yes, as Colleen mentioned, people do prefer to take capsules rather than have enemas, with maybe rare exceptions. But you have to address bioavailability and get your bugs past gastric acid and bile acids. And again, basically there are multiple technologies, capsule types and coatings for capsules or tablets that allow you to preserve the bacteria in the face of acid exposure to the extremes. So it can be done. Perhaps one of the more interesting aspects of CMC for microbial therapies is the quality control aspects. And here the challenge is to devise basically all of the elements of CIS-PQ to really thoroughly control your product. For safety, you can read in the Live Biologics Products Guidance, there's some motherhood and apple pie there. Yes, you should know your bug sequence. You should have it characterized for antibiotic resistance and so forth. You want to understand whether it's got prophage. An interesting feature is toxins. For a lot of these gut microbes, you may not have a reference genome, or you may have a poorly matched toxin gene. And so really, you may need to screen functionally, phenotypically, rather than by genetics to understand toxin expression. Identity, that's pretty straightforward. Strength, initially, of course, you can use colony-forming unit assays for species detection. Potency for activity, though, is an interesting concept, right? Even if you have a single microbe drug, it doesn't take very much thought to realize that basically even a single microbe has a secretome of hundreds, if not thousands, of compounds, right? So unlike a perhaps more precise single molecule type biologic where you're trying to hit one pathway and activate it, you're going to be doing polypharmacy. And in some diseases, you may actually need polypharmacy to have an effect. And so devising these potency assays will be interesting. The other thing that's really unique is for gut commensals is that USP 6162 are not generally useful. You'll get product breakthrough on these. And so you have to devise ways of suppressing that product breakthrough or enumerating it as being product or non-product. Lastly, I'll just finish up by saying GMP manufacture of commensal organisms is also a specialized endeavor and complex. I particularly like this phrase that's taken from the FDA's 2006 guidance on manufacturing of spore formers. Basically, manufacturers are encouraged to identify alternatives if they can, right? If I'm not putting spores in my plant, I've got a problem, unfortunately. So I have to deal with that, as would probably most people who are going to make products from gut commensals. And so you really have to make sure that you've got unique facility designs that have appropriate classifications, that have appropriate pressure gradients, so that you can both keep bugs you don't want out, keep your bugs in. You need to supplement that with contained product operations. Try to minimize the use of reusable equipment so you have a minimal chance of cross-contamination, and then use extensive decontamination procedures to make sure that you have addressed concerns of cross-contamination. And then last but not least, you also want to basically make sure that your environmental testing that is more or less well established for traditional biologics will actually address the microbes that you're producing, so that you can detect if there was something left from a prior campaign. Right? And then for consortia, basically we have to deal with multi-strain product considerations and being able to operate in a rapid fashion. Right? If we had to produce things serially, making a master bank, a working bank, and a drug substance, and repeat that every time for, say, 15, 20 strains, you've got a campaign that's well over half a year or more, right? So getting your procedural and temporal segregations down and having appropriate decontamination to deal with that is key to having elegant manufacturing. So thank you for your attention, and thanks to patients, to collaborators, and to all the internal team at Seres.
So now we'll have the three speakers from this session come up. We'll do about 10 minutes of questions for them, and then after that we'll go on to invite the three speakers from earlier up, and we'll do the panel discussion. Anyone have any questions? All very clear.
Sam Forey, NIST. I wanted to ask a clarifying question for the manufacturing controls about what kinds of evidence you are able to present to the FDA, to regulators, to demonstrate the validity of the analytical methods that you use, to demonstrate that your control processes, you have to provide those measurements and supporting validation measurements. What kind of measurements are you able to show to validate the protocols?
Oh, there we go. Yeah, I don't think it's any different than any other biologic. There's perhaps a slightly different spectrum in the sense of you got a lot more microbiological assays, obviously, right? And so, for example, on say bioburden testing, you're going to need to do a lot more extensive work to show that you're detecting your product, that you can pick out contaminants, right? There are a fair number of sequence-based assays too, which is probably the more novel thing, I think, for biologics production. And having validated sequencing and for that matter, databases to go along with that sequencing. You can produce sequence, but then how do you interpret it? How do you know what it is a whole other kettle of fish, right? And how you validate that's a different story.
Yeah. Sort of a follow-up to that based on sequencing. So you have an organism or a consortium of organisms, and we're very good at doing genome sequencing. And one of the things is purity, right? We talk about purity and also derivatives. Common thing in pharmacology is, oh, let's just throw a different methyl group and we'll change the things. At what point do we decide that, oh, how many single nucleotide variants do we have before we actually have to revalidate this as a brand new or derivative drug, quote unquote? And the reason I'm sort of asking it is, can we really work this under the existing rules that we have for drugs right now, in your opinion?
Well, so, I mean, historically, or currently, I guess, when you go to license a biologic, say a monoclonal antibody, you're expected to sequence the cassette, which is going to be 3,000 base pairs or something like that, show that you don't have mutations or characterize them, whatever, understand their loci of insertion. That's pretty tractable and understood. I would agree that it's, once you're dealing with a 5 megabase bacterial genome, how do you look at SNPs? How do you look at indels and so forth? I think, the key thing for all of this is you need to show stability from your initial materials and in your master bank to your final product, and then clinically demonstrate that the stuff works, right?
Yeah, I just wondering, just a point of clarification, Colleen. So now in your practice, are you using standardized preparations, or are you using related or household contacts for donors? Because it's changed a little bit. So what's your current practice right
Button. There we go. At this point, I'm using almost exclusively stool from Open Biome, and it's just a matter of, it's the easiest thing to do, and it really, these patients are really eager to just get everything done with. But if a patient requests that I use a related donor, I give them that option, and we go looking for one. And I do explain that kind of might take a little longer. There's no guarantee that the donor's insurance is going to pay for all of that laboratory stuff. And I don't cut any corners, even if they've been married for 50 years. They go through all of the HIV testing and everything else. So most of them opt for the Open Biome.
Thank you. My name is Isabel Walls. I work for USDA. In your talk, you mentioned, I think it was Lactobacillus reuteri, and it needs glycerol as a substrate for making Reuterin. And so I'm wondering, when you do the clinical trials, and I guess anybody, do you consider the substrate? Do you consider, is it what the people are eating? And if so, do you control what people are eating? Assuming they're in hospital, they're already sick, you should know what they're eating. Is that the substrate, and do you control for that when you do clinical trials?
Yeah, I think you should control for that, and so you either do that in a, as you mentioned, a controlled population, like an inpatient setting, where you know exactly what they're eating or what they're being given at least, or you co-formulate it. And the term synbiotic has been used a couple of times by some other speakers and audience members, and I think we'll hear a little bit more about a talk where a synbiotic was very successful after co-formulation in preventing neonatal sepsis later today, but yeah, I think it's so universal that you expect it to be in anyone who's got a normal diet, or you co-formulate it. I think would be the way to go.
I think we would fall more in the Stephen Friedman camp, that it's kind of futile to control what people eat through the course of their disease.
Even when they're in hospital?
That I would have to defer to my clinical colleague, Shelley Truxas. So I would refer you to her over there.
Yeah, I would just say, as a clinician in the hospital, you can control what you order for the patient to eat, but what they actually eat is completely different.
If no one else is going to ask a question, I can ask one. So we had a talk on use of probiotics for prevention of C. diff and on use of FMT for C. diff. So as clinicians, you have those options. What do you do? I think it seems like you are almost exclusively using FMT, or maybe it depends on the state where you're at, but in practice, are you using probiotics versus FMT?
So this actually comes up quite a bit, because all of these patients, once they've gotten over C. diff, there's kind of like a PTSD. So anytime they're ever going to need another antibiotic again, or going to have a surgery or anything, they're calling me and asking me what they should do. And I actually, if they're not immunocompromised, I do tell them to take a probiotic along with, and then for a period of time, like about a month afterwards. And does it work? Maybe. If it's going to really break the bank and they can't afford it, I tell them, there's not great evidence that it's going to do anything, but I think it really empowers them. They feel like they're doing something, and I think that's meaningful in some way. There are people who recommend giving antibiotics along with like an anti-C. diff antibiotic, like vancomycin or metronidazole, along with whatever antibiotic they're taking for their UTI or their pneumonia. I don't do that, just knowing what I know about C. diff. It's caused by dysbiosis. Just throwing another antibiotic into the mix never seemed like such a good idea, but that's definitely recommended by some other people.
Yeah, I would echo those same responses, and just add that most of the time I don't have to make a recommendation about these things. The patients are telling me what probiotics they're already taking for their C. diff. part of this is, my filter as an infectious diseases physician, I'm not seeing these patients until they're on their third, fourth, sometimes fifth episode or more of C. difficile anyway. And so by that time, they've already gone on the internet. One of the first things they found is probiotics for C. difficile, and they're just picking things. And right now my practice is I don't stop them. And I say, I don't really have much evidence either way to tell you what to do. I can tell you the evidence does show that there's some signal that there might be some benefit here. But specifically the probiotic that you're choosing to take, I have no guidance to give you on that specifically. I have been using kefir a lot more. So, not a probiotic according to the strictest definition, an undefined consortia. It's just a yogurt drink. It doesn't taste particularly good in my opinion, but my patients like it and they drink it when they have C. diff. And we have some uncontrolled data, again, Johann Bachen's case series suggesting that there may be some efficacy there. So I certainly don't stop them, but I take kind of a more balanced approach of I don't even have to bring it up. And part of this also is I practice at the University of Michigan in Ann Arbor. We draw from wide catchment, but a lot of our patients are, educated. Some of them are coming in with notes and printouts from web pages that they've researched. So it's a different crowd, but usually I don't have to bring it up in clinic.
Any other questions? If not, I'll invite we have one more.
Weisel, what is the evidence that we're talking about live microbiome-based products, but that killed FMTs? Do they have any efficacy? Have they been tried?
Well, I will say that there have been studies looking at fresh and frozen, as we've heard, and the frozen preparations, there will still be spores, spore fraction in that stool, so it's not killed per se, but it's certainly reduced in terms of the vegetative content there. But then also people have, recently there was a case series, I don't remember which group it was, but it was about 5 patients, I want to say, that were successfully treated with FMT. These were fecal filtrates. And there were submicron filters where they actually tried to do cultures afterwards. They weren't able to culture any bacteria. So certainly there could have been viral particles and other microbes in there, but not bacteria. And those patients were all cured. Now that's just a case series, again, uncontrolled data, a series of 5. But I don't know that we've established completely that microbes themselves are the necessary component of stool when it comes to the therapeutic effect.
Just to add to that, when Seres was trying to develop our C. diff drugs, we wanted to understand whether it was the bacteria or not. And so we did do animal studies where we took the material, the spore fraction, 0.2 micron filtrates, 300 kilodalton ultrafiltrates, 2 kilodalton ultrafiltrates, and basically you saw elimination of the activity once you take the bacteria out.
Okay, so with that, I'll invite the 3 speakers from earlier up onto the stage. 2? We only have 2? Oh, we had one had to leave. Okay. 2 speakers from earlier. And my co-moderator, Susie, is going to come back and we'll have a discussion of all of these topics.
I just will say that unfortunately Dr. New had to leave us. He had a plane to catch, and with all of the plane issues going on, didn't want him to potentially miss his flight. So unfortunately, he won't be joining our panel, but we have 5 panel members. Do you want me to start? Yeah, go ahead. All right, so we had talked about having a number of questions for our panel members. But we would like to encourage folks from the audience. Clearly there was a good discussion that was going on this morning already. We have 3 slides worth of questions. This one is the most packed. First one talking pretty much about the microbiome, the second one really talking about organisms in general, and then the third really about logistics, which is kind of the areas where I think we've been headed this morning. So while you're coming up with your questions to ask the group, and actually, I would encourage if the group has questions for each other to think about that. But what we had thought up front in terms of just talking about the microbiome under all of the different circumstances that we've heard this morning is how do we characterize the pathophysiology of all these different illnesses with respect to the microbiome? Do we need to personalize therapy based on an individual's microbiome? How important is the strain selection in the treatment of a given indication? And for products with bacterial consortia, how important is strain synergy? And we'll get a little bit into the strains in the second slide as well. And then how do we differentiate treatment from prevention? And then how can current associative data be used to support clinical decision and or to advance the development of new products? So we'll throw all of those out there. You know, it's kind of, you can pick a question you would like to start with. You can ask one of your co-panelists a question, and I would like to encourage the audience to come up and have a conversation. We're probably going to stick with this for about 15 minutes or so and then kind of go on to the next. But
I'm happy to tackle a little bit, less on the microbiome as it relates to gastroenteritis, but more on the pathogens, which I think is really important. So the advances now in diagnostic technology has been great. And so there are multi-analyte syndromic panels that are available on the market. They have their own challenges on the clinical side. But from a research side, they do enable good characterization of the infectious agent because not all diarrhea is the same. We don't always even find the pathogen. And I think the comment was a lot of C. difficile infection referred for fecal transplants aren't even C. diff, and they've got other diseases. So I think that really helps us know what we're talking about, and that we are able to separate apples from oranges and be able to figure out probiotic or agent disease, meaning which pathogen is actually causing the symptoms, is really, really important, I think, in terms of where research should be at this point in terms of gastroenteritis, and just kind of broadly treating all of them the same is probably not the way to go. And some of the studies that we did, we're actually now looking at that. We have that data. We're just getting into deeper analytics on that. Then the other piece is also to characterize the disruption in the microbiome and then the healing from the acute episodes as well. So we've been collecting stool down 5 days down the road and 28 days down the road in these kids after their randomization to probiotics or not probiotics. So we can start looking at the impact on that as well.
A little bit just again to talk a little bit about the IND issues. And I was really happy to hear the discussion that was earlier that talked about how with dietary supplements and foods that we really don't need to have INDs for structure function endpoints and studies, which I think is a huge declaration to come out of this. And I know it's part of the guidance document. It was just good to have that reaffirmed, and that use of endpoints that are focused on reduction of risk of disease also probably don't need an IND necessarily, and that's great. I guess the one other component that I wanted to talk about in terms of the insistence that human studies and probiotics be conducted under INDs has to do with the guidances and the FDA stances that have occurred in the past that I think are, cast the net quite broadly in terms of how they view drug endpoints. And now, granted, there's a definition, treatment, cure, prevention of disease. But I do think that there have been some judgments that FDA has made. For example, the example of antibiotic-associated diarrhea was brought up. So right now, in FDA's mind or interpretation, any substance that's used to prevent side effects of antibiotics would in itself be considered a drug application. And my point is that that's actually a judgment call on FDA's part. And I think to the extent that studies can be conducted in reasonably healthy people that are safe studies on endpoints that you may be able to consider structure function, you may be able to consider disease, that to the extent that they're safe and it can pave the way for innovation in the food and dietary supplement category, my appeal is just to see if the FDA would be willing to just consider those things a little bit more broadly so that we don't have such a narrow view of what a structure function claim is versus a very broad view of what is encompassed.
I'm not a clinician treating patients, but I guess I'm a bit mystified. I have to say, coming from a person who's worked in the pharmaceutical industry for now 28 some years, about the seeming confusion. And I guess my question back to you would be, what do you want to do with the information? And it seems like what you want to do is you want to make a claim about a treatment or cure of a disease. And if that's what you do, it's the old drag racers, run what you brung, put up or shut up. Do the rigorous trials under IND, prove efficacy, prove safety, and show that your product's under control, and get a license.
No, and I completely agree with you that there's conducting a trial not under the IND rubric does not compromise safety or appropriate design of clinical trials or product definition. All of those things are assumed.
I think, doing studies in healthy people who are seeking to, have supported organs or better this is different than doing it in somebody who's diseased. I think that's...
No, and that's a fair distinction.
And there you need to take care, and you should have a lot more controls that really call for IND filing.
And I'm not objecting to that. When you said what's the goal is to provide dietary support for people who need it, either healthy people who are at risk of developing something, or someone who may be considered generally healthy, a child going in for an antibiotic or an ear infection, that may be able to use dietary support to be able to prevent the development of some kind of side effect, or worse, where you might get some kind of pathogen emergence because of the antibiotic treatment. And if a dietary approach that doesn't require getting a prescription and something that's generally available as long as it's safe, and you want to look at it from the research point of view, and you control the study properly, you make sure the safety is there and the manufacturing is appropriate, I don't think that it serves anybody to force that into the drug rubric when the intent is never to market a drug. The intent is to market dietary support.
But we could probably continue this over a lot of beers, but you still seem to be going back to, you're talking about at-risk populations. So if there's an at-risk...
I'm saying either prevention and healthy, or there are at-risk, but we currently allow foods to address at-risk people. I mean, an at-risk person who with lactose intolerance has to eat lactose-reduced foods. They're at risk for developing symptoms from consuming too much lactose. A person with high cholesterol is considered to be at risk, but that's a general population targeted group where you can use foods to address that. And my point is a child taking an antibiotic or an ear infection is at risk of developing some kind of intestinal potential problems. And I'm not saying there isn't room for drugs. There obviously are. I'm just asking for there to be more room for dietary support for conditions like that. But you are absolutely right. You have to have the same, you have to have good control of the study. It has to be properly designed, properly powered, all of those things. I'm not talking about study quality here. I'm talking about forcing it under the drug rubric, where there really is an intent.
So can I just add into this mix? I would love to hear a further discussion of one of the questions that's up there is about the strain selection, and what are you using to be able to do the studies that you want to? No, because you want to do studies, but I'm just curious as to how you do the strain selection associated with that.
There's many different ways to go about doing strain selection. I think we heard some preclinical type studies that have been conducted already today. To me, that's the science behind it. You develop your hypothesis based on whatever preclinical data you have. But to me, that's not really germane to the regulatory conversation, because you have to determine safety of whatever intervention you're going to define, or dietary intervention you're defining. And I'm not sure why it's important to tease apart the exact rationale for a particular strain to be chosen.
Safety is part of your strain.
Sure.
Right?
Oh, no, of course, yeah. I mean, I'm sorry if I didn't make that clear. Obviously, you have to choose strains that are safe, yes.
Can I ask you a question? Because what are you proposing to develop? Is it a food with probiotics in it, like accepted probiotics that we know is going to be Lactobacillus GG, or it's going to be this, or is it going to be a food with some other strains that don't have that long history that we have with probiotics, like strains from like someone's gut or something like that? What are you exactly talking about doing?
Well, I think what I was talking about is more kind of the old school stuff in terms of strains that we know quite a bit about the safety. We've got good history of safe use. They're on the European QPS list, where you know what's going on with them for the most part in terms of a safety assessment. But I think the broader question could be relevant to next generation probiotics, where you say, well, then if you find some Calibacterium or something that looks interesting, would that be an appropriate addition to a food? But that I don't think is such a difficult question. You just have to go through the proper safety evaluations, and you have to submit that RAS, affirmation or notice and get a ruling on it.
We have a
Ernestine,
question
do you
from
want
our
to address one of the
overflow room.
One minute.
I want to address the question, do we need to personalize the therapy based on individuals' microbiome? I get that question a lot. It's been in the news a lot after the 2 cell studies. And I think it's a sort of a red herring question. I think the answer is clearly yes, we would love to do that. But to pretend we're anywhere close to that in medicine is really not to understand what we're doing in medicine. We do that, and people can disagree with me, for a few cancers, and that's almost it. You know, people have been talking about for 20 years, you're going to get a genetic test, and then I'm going to tell you if you should take a beta blocker or an ACE inhibitor. But we're not close to that yet. So for a few cancers, we do it. Maybe sometimes for IBD, I don't know, we look, maybe not even. But it's very few things we personalize treatment for. And to pretend that you need to do that for probiotics, I think is inaccurate. Now, I think it'd be great if we could do it, but we're probably 50, 60 years away from even being close to that. So I think I think it's important to address because I get that question all the time. And often it's people like the Cell authors who are selling something in personalized medicine and then publish something that says you need personalized medicine, which is a little suspicious.
LD30, go ahead.
Hi, this is Joelle Woolston. I'm from a company called Intralytics in Baltimore, Maryland. And I was hoping the panel could address the question, how do we differentiate treatment from prevention? The question is specific to...
Yeah, I have the same question that Mary Ellen had earlier. Maybe I'm unable to ask it eloquently. We are not against saying that it has to be a characterized strain or the CMC has to be wonderful. It should be 100% safe. But then, if it is, I can give an example of vitamin D. So if you give it in really high dose, of course it has to be on a prescription, but you're still selling it over the counter. People can take it a small amount. Imagine something like that it hasn't reached that stage of development. Are you going to stop vitamin D trials and research and demand that all vitamin D research should be done under an IND, or would you still let vitamin B or other vitamins be sold OTC and at the same time concurrently develop high-dose vitamin D as a drug for a particular medical ailment?
I think very different situation. I mean, you're using a vitamin as a comparison here. I just want to, this is also off topic, the panel is meant for discussion with our panelists and our speakers, not for direct questions to the moderators and to the FDA. So let's go ahead and address the questions.
I have a question for the actual panel. Are you guys ready? I represent an organization that works for advocacy and education for people and caregivers of people with C. diff. So I feel like the FDA has drug its feet on determining what a fecal transplant is or what that is as a product. Is it like blood? Is it its own thing? Is it a drug? And maybe it's potentially all of those things. But what I would be interested to hear from each of you as possible, and I think probiotics would fall into this too, is like, would determining a designation of these things, what would be the pros and cons of that? Like, would there be the pro of like, this is its category, we understand it, we can move ahead with it? And would the con be this will limit us in the way that has kind of been addressed in talking about some of the probiotics, where it's like, well, this isn't a drug, it's a probiotic, but if you want to use it this way, it is a drug. Because as a layperson, this all sounds really arbitrary. Like, it doesn't come across as driven so much by science as by arbitrary rules that we could get into how those rules get established, but it doesn't feel very sciencey. It feels very lobbying-y. So, just curious what you think the pros and cons are.
I'll just start with that. When I obtained the IND to do my clinical trial, I had to go through, at that point, still the pre-IND process, and I didn't really understand what I was doing. I was kind of going for, I really looked at fecal transplant as a transfusion or as almost like you would look at an organ transplant. And, you would screen a donor, make sure they don't have any underlying diseases or any communicable problems. But I was told that no, it's a drug and it's a biologic for this reason. And my understanding was that because it's excreted, and things that are excreted can't be in the transfusion paradigm. Maybe I'm, correct me if I'm wrong. But I think a lot of us have been thinking about this and thinking that you're right, we're kind of trying to kind of jam sometimes like a square peg into a round hole, making some of these whole stool, very complex FMTs, we're talking about like multiple microorganisms and how they might be interacting with each other, coming from these fresh stool from donors and what we can learn from that, and trying to say that that's a drug because it's very difficult to have it be identical batch for batch and all the things that you have to do to have a drug. I know in the audience here, and whether, I was part of a working group at University of Maryland that Diane Hoffman put together to kind of talk about going forward and kind of regulatory aspects of gut microbiome therapeutics. And really the outcome of that was published last year in Nature. I think it was very interesting to look at maybe whole stool FMT done from just like a patient of mine who wants to use his wife's stool to treat his C. diff. That's the practice of medicine. The FDA doesn't need to be involved. It's me. It's my hospital that gives me privileges. It's my state medical board that makes sure that I'm doing things appropriately. And then as you kind of move up the ladder and things become either more characterized or you go to the level of like Open Biome that has a stool bank, obviously they're doing a great job, but you can't trust that anyone couldn't open up a stool bank and just start shipping stool all over the place. There needs to be some oversight, and that might make more sense in the kind of tissue transplant, almost transfusion paradigm. And then as you get into things that are more and more characterized that you're looking to encapsulate and sell as a drug, then maybe that would make more sense for like the IND and the typical drug pathway. So again, not my idea. It came out of a huge working group, but that was really, kind of put together as maybe one solution to all of this.
Sorry, if I can just also talk about probiotics a little bit, and maybe I think the reason, even we're talking about probiotics actually in a way we never talk about antibiotics. We don't talk about antibiotics are good for. We talk about an antibiotic, a dose, a regimen, a duration, and an indication. And we really talk about it that way. And actually, I think, once again, I'm willing to get things thrown at me, I think the probiotic industry, the way it's marketed, it doesn't make sense to most clinicians that all 700 probiotics available at Whole Foods today are all good for everything. And if we are not, I'll say I think they should be studied well, rigorously, and regulated, and they should be regulated like a drug. And then you might be able to get at some of Dan's comments of why aren't we using them? Because I think the way the industry has set itself up, it doesn't conform to the way most clinicians like to think of a probiotic is good for this in this patient for this long at this dose. And I think as long as we talk about probiotics like they're one thing, there's how many? 10 million trillion in our body, and the number of brands out there. If we talk about it like that as to clinicians, I think we glaze over because it just doesn't register with us the way talking to me about this drug for this treatment in this patient does. And I think that I actually would encourage regulating it more. I hate to say this, but the more we regulate it, the more people will use evidence-based therapies and feel comfortable in them.
Oh, so no throwing stones. I was just going to elaborate on one other reason why I don't envy the role of regulators when it comes to fecal transplant, because, on the one hand, don't do this, but if you were to Google fecal transplant and go to YouTube videos, you will see that people are doing this stuff. And sometimes it's very sophisticated, sometimes turkey basters and things are involved. And the other thing that I didn't mention is, in addition to taking probiotics, a lot of the patients that I see have tried fecal transplant on their own. I don't know if you've encountered this, but it's not super common, but it's getting more common now than it used to be. I think to a large degree, because this is kind of a limbo zone and it's not clear, and patients are desperate. They do get desperate. That there is enough regulation of this that it's not widespreadly, it's not available at your local community physician for a lot of patients. So that's why they're coming to places like universities to receive this treatment. So on the one hand, there is unregulated use happening. On the other hand, although I think it is safe in the short run, as from the data that Colleen showed us, that in the long run we don't know. And I tell patients this, that I'm treating your C. difficile right now, but I don't even know how to answer the question of whether 10 years down the line, did I give you diabetes? Did I give you a higher risk of obesity or cancer? And those are, I think, that's one of the roles of really trying to characterize this and establish, better precautions and actually be able to answer some of these questions that regulation can have.
Two things. One, I agree with most of that, although I would say that, we don't know that for lots of things. We're just learning that statins now, 1 in 100 people get diabetes from a statin. We didn't know that for years. So we have everyone on a statin, right? So you need to follow these long term to follow it. I would say when you get back to over-regulating probiotics, that the way it's set up now and the way they're defining an IND is I can't actually go to the supermarket, I can't go to Whole Foods and pull a product and try to study it. I've talked to the FDA about that. Unless the company wants to work with me and help me get the IND. So regulating it more, I think, would cause more problems, actually.
And I would concur completely, as I couldn't get an IND for one of the drugs we wanted to study. So how you get an IND is a different process. Let's not go there. But conceptually, I think doing studies properly and doing them, in controlled manners and under proper regulations is the way to go. How that's regulated, I'm not even going to touch on that. I didn't go there.
And this really gets into some of the questions we have. I switched to the second slide here. On this slide, talking about how do we ensure that the product is what the product's supposed to be, the high quality and reliability in terms of consistent manufacture and purity. And then we can get into some questions about single versus combination. But again, I think we've already discussed that's already I would like to hear at some point from those who are conducting controlled clinical trials for FMT products to weigh in on some of these questions.
I think one of the themes that I've heard in all of your talks this morning, whether it's NEC, C. diff, antibiotic-associated diarrhea, and I think also the paper that was published at the Weizmann Institute last week started to, I think, open up the question as far as why are we seeing efficacy in one subpopulation, and potentially damage in another. And I feel like we've amassed a substantial amount of data, but we keep just trying to do studies to understand efficacy without looking at some sort of diagnostic tool or like really digging into responder, non-responder dynamics. So I guess my question is would a tool like this be helpful? And obviously it would have to be condition specific to some degree and maybe gender or population specific. But do we have, I guess the questions are, do we have enough information in the field right now to start creating some of these tools like they did in the Cell paper where they created an algorithm based off of the experiment they ran and then, validated the algorithm. So can we start moving in that direction as a way to kind of overcome some of the challenges that we're seeing from a personalization perspective and help prove efficacy in a better way and maybe help you guys out a little more?
Well, yeah, I'm not the microbial ecologist on the panel, but I will say that I think we're not at that stage yet. And I think we might have the tools that it takes to get there. As has been brought up, there's the whole idea of anatomy versus physiology. It was on Dr. Young's slide, and I think, John, you talked about it, about how the structure of the community is one thing, but you can have lots of different structures that functionally perform the same functions and look the same. And so we've gotten very good, I think, with next generation sequencing at looking at structure, at looking at 16S, but we're not quite there yet when it comes to bridging those various disparate structures and figuring out what the functional phenotype is from a different set of structures just yet. Maybe once we get there, I think we'll be able to make better progress at individualizing therapeutics, but not quite yet.
Yeah, I guess I would just add to that by, again, going back to what I was trying to say about the sort of the bedside to bench to bedside paradigm, you need to do the interventional controlled trials. You need to be taking specimens, whether it's stool specimens, tissue specimens, and then you have to use all of your powers of analytics to try to figure out what's going on and why things work in order to really build a true understanding. That's what we're trying to do at Ceres, and I would assume a lot of other people are as well.
As I was mentioning, we want to dive deeper into our negative studies because there may be populations in there, whether it be pathogen or response, that will actually tease out. Maybe there are certain responders in here and who those are. Then down the road, you can decide whether you use that therapeutically or not. But I think 100% actually negative trials are almost more important to find out why it didn't work.
All respect to our hosts, I would like us for just a moment not to talk about regulation and remove that. And have a philosophical argument, because we have this tension here. I hope it doesn't take 50, 60 years before we can do precision medicine, okay? But there's a tension between doing science, and someone already said science has a bad word, you know, has this other meaning about being sciencey, et cetera. But how do we balance the time it takes to do studies, both preclinical and clinical, and amongst the clinicians, the desire to do something for our patients in the here and now? And what have you guys used to try to kind of, there are two opposing things. One takes longer time, one you have a patient sick in front of you. What have you been doing to try to kind of balance that sort of tension outside of regulation? Okay, forget about whether or not you get in trouble doing it. How do you balance it?
So to me, that gets to where you do the question was asked, number needed to treat versus number needed to harm. And I wish Dr. New was here, because that to me is the question when it comes to the NEC issue. Because as long as you can get a safe product, which there's no one going to disagree with that, you need a safe product that's not contaminated, that's studied. It seems like that's the kind of thing where you look at it and you say the number needed to treat versus number needed to harm, and you need to try experimental before the evidence is there. And I would agree the evidence is not there for all the things. But that to me is a clear-cut thing, and I think it's same with FMT, but, you know, and lots of other indications.
The other thing that's going on a bit more in Europe than it is in North America, I think, are registry-based trials. So I think there are certain ways of capturing a lot of data, and actually I'm starting to plan a clinical trial of a diagnostic device, but I'm going to embed it into care. So it's going to be embedded into care. It's not going to be a therapeutic option, and then collecting the right data to answer the questions at large scales. So I think registry-based trials are probably the thing to look forward to, of being able to capture all the patients who are getting FMTs or C. diff at an institution or healthcare networks.
We
and
actually
I think
have an FMT registry. I'm glad you brought it up. That was a good plug. No, so there is a national FMT registry that's NIH funded, and that's kind of a joint collaboration between the GI and ID societies, and we're hoping to get 4,000 patients. The problem is one of the difficult things is, you know, it's very expensive to follow patients for up to 10 years, and like, how are we going to retain them? How logistically is this going to work? But we're working really hard on kind of reaching out to the patients through apps and emails and things like this. But our hope is that being able to follow and get that real world efficacy data will help answer some of these questions, and have a biobank even tied to that, so that if something does come up, we can like reach to that stool and say, okay, this person developed this unusual condition. Was it something from the donor?
And there were some patient advocacy groups here, and actually they were asking how you can help. I actually think getting patients to be willing to participate in trials and to actually be willing to have their data collected, integrated into registries is crucial. And that's one of the biggest barriers to research, period, is declining to participate in clinical trials, and then even declining to have data used in anonymous de-identified registries. And so patients should advocate for this if that's what, I can't tell you what to advocate for. I think it'd be an important thing to consider advocating for.
So let me just chime in 1 second. I'm going to move to the last slide for us, just so we have, just so we have all of the slides up there. And this one gets to some of the logistics that we've been talking about in terms of how do we address the lack of equipoise by some healthcare providers, and the ability to conduct clinical trials, some questions about funding needs, and then how to take advantage of some of the networks and some of the registries that are out there. So I just wanted to so that's the end of our kind of planted questions, so to speak, but we just wanted that to be out there as you're continuing the discussion.
I'm Lee Jones. I'm with Rebiotics, and I have been involved in conducting clinical trials with human stool-derived drug products. So I just want to remind everybody kind of how this got started in the drug world. So early on, we connected with the FDA and asked about, Colleen was talking about, how it was categorized. The FDA said that because they're organisms and they're not human tissue and cells, that they don't fall under the human tissue and cell regulations, and therefore was a drug product involved in CBER. We've conducted multiple phase 2 trials, and I would just want to remind everybody that there hasn't been any kind of finalized, formalized, products that have been regulatory approved at this point in time, I think, anywhere. And so we're all in early stages looking at it, and I want to echo the fact that we do need these clinical trials, and I think it's a little bit disingenuous to put things up there without the context when you're trying to compare non-regulated to regulated studies. I think we're early, and that's my main message, is that it's hard to draw conclusions on anything when it's so early in the thing. So I just want to remind people that there is a process. People are going through that process, and I think at some point in time, more data will be available. It's not going to be be-all, end-all. I think there's still a lot more to discover.
Oh, I just wanted to comment on a previous comment about there's been a lot of discussion about number needed to treat and number needed to harm and the idea of harm. And I just wanted to remind or comment that I think a portion of harm that's underappreciated is when patients are doing something that they think is efficacious but isn't actually efficacious, because then that delays treatment of something that actually could have helped them. So I think that's important to keep in mind. And so we do want patients to be using things that when we as physicians make recommendations, we want to have some sense of trust behind those recommendations and what we say. And I think it erodes trust if we recommend something that it isn't necessarily a specific product, but it's more of a category of product with variable efficacy, some of which may help and which don't help, especially if it's a very serious condition and the patient is delaying or avoiding some other potentially efficacious, known proven efficacious therapy in lieu of something else. That would be my comment.
I would agree, although I would say the data from choosing wisely and stuff is that we overtreat people. So maybe staying away from the doctor for lots of things is pretty beneficial.
And it's just about when patients come to me, and most of them come to me after they've suffered, 2, 3, or more recurrences. And I do have availability and accessibility to some clinical trials. Some are open label. The other's the Finch study that's starting to, looking another capsule study. And I give them the choice. If they are a candidate for a trial, I give them the choice to be in it or not. I don't feel like I should, force someone to take a placebo for something. And I'm bound by my relationship with that patient, and what they want to do and what's best for them, and not, this greater good of, helping this company develop their drug. And there are definitely patients who aren't appropriate for FMT. I see them. There may be limited life expectancy, very frail, and I maintain those patients on just vancomycin. I said maybe for the rest of their life. That's, 6 months to a year. But I think that the position I'm in, I conducted that placebo-controlled trial, double-blind as best as I could, to not just convince the world, but to convince myself that I wasn't imagining that this worked as well as it did. And I can say there's never been anything that has surprised me more than FMT in some cases, where there were, people severe, complicated, septic in the intensive care unit, and after 2 or 3 doses of stool enemas, we're turning things around. And I think it's very hard to go backwards from that and to say, okay, well, now we need to kind of pull back, let's get some animal data, let's figure out what strain. And I'm not saying that's not important, but I don't think we should lose the momentum that we have in combating this epidemic.
A question in 1D06. You can ask your question.
Hi there, this is Richard Ethio from Longmont Health Solutions. Just a general point on the IND restriction for dietary supplements. Industry sometimes finds
My name is John Honig. I'm with the Crohn's and Colitis Foundation. We're a patient advocacy group and research funder. So I wanted to address the question, how to address funding needs, and also return to the last.
We're muted.
So all of you are focused on advancing a certain type of microbiome-based product through different, and use secure resources for that in different ways. And so we've discussed the need for a variety of things like really good CMC, like long-term registry studies with really good data collection, and RCTs, and all these things have a role. And we've also learned that there can be a variety of different regulatory pathways that a microbiome-based product can take in the direction or the drug direction. So given that we've seen one beautiful presentation about how a CMC process can be developed, and it seems like something that's very resource intensive, and that's presumably incentivized by the way in which the eventual product would be paid for and regulated. For those of you that are seeking resources for products that don't fall into that drug category, is there a way to address the funding needs? I mean, it seems woefully inadequate to address these challenges that you're talking about with the current mechanisms that we have for funding. So just wondering. The only thing, I mean, I go for federal funding generally, both in Canada and the U.S. And the one thing that I did notice, and I don't know if anyone knows more about it, but the NCAM has stopped funding RCTs of probiotic research. And I don't know how that's impacted people down here, but that was quite surprising to me, given that they would seem to be the natural institute to do it. And I don't know if that plays into, Dan, some of your noticing a reduction in clinical trials, but they have made it very clear that their RO1s generally are no longer eligible for RCTs of probiotics.
Yeah, they did fund my first two, and now they're funding the mechanism trial, but they don't fund RCTs for it. But I think that's for lots of supplements too. But it could be.
I'm going to make a comment about the regulatory environment because I think there's confusion here, at least among some of us in the way we talk about this, but we need regulation, and the more that's a confusion because probiotics, food in general, it's not only drugs that are highly regulated. There are processes. The difficulty is that we have a different interpretation of what that process entails and what the conclusions are. And I think it's important, especially for FDA industry clinicians, to have, this is a nice, but frankly, those issues of the regulatory issues, FMT, dietary supplements, etc., are not going to be addressed in this video. Well, I'm suggesting that maybe we should get together in a true working group to talk about how to interpret structure, function, etc.
I wanted to deviate from the questions on the slide. There's already momentum in that direction. And I've heard a couple of keywords: momentum, we've talked about confusion, regulation, industry, and I'm standing here from the IPA perspective, probiotic companies. What is the laundry list from a clinician's perspective industry needs to meet? Because we can talk about FMT versus probiotics, we can talk about efficacy, safety, we can talk about a whole bunch of things. But I mean, I'm a scientist who worked for probiotic companies. We have evidence-based products. We have at least multiple clinical trials behind products. I don't work for the company anymore. But what is it that industry needs to do to support the process, to keep the momentum going, to allow you to keep working with your patients? And I mean, that can be anything from safety and better studies, but can you give some specific things that we can work on? Not just better products, more evidence.
I can say that when I'm recommending, because patients always ask about probiotics, and I see a lot of IBS, and I try to, if I do recommend one, I will usually, for instance, Align or Bifidobacteria infantis, that they took some initiative and conducted a clinical trial and show that it worked. So I think if industry themselves, particularly probiotics, puts a little into these clinical trials, I think that's always helpful.
I mean, I really think, for some disease processes, maybe there isn't a need for more evidence. Dan talked obviously about antibiotic-associated diarrhea. I think for others, we are looking at phase 3 clinical trials, which generally they haven't funded very large. They haven't done them investigator-initiated. So they've been very directed by industry, conducted in that manner. And so I think really allowing for large phase 3 trials that will definitively answer questions, not going into the meta-analyses of small little sub-center, single-center studies where they control the data, but really total independence of data and access, and allowing investigators to choose the outcomes with patients that are important to them, directed and dedicated out of the disease process. That to me is really, I mean, that's what distinguishes. And then you can start talking about, the really, really large clinical trial of probiotic X for drug X, as opposed to the generic, hodgepodge of the meta-analyses.
I'm a microbiologist by training, and my basic understanding of the levels of evidence in medicine, we start off, of course, at the top with your meta-analyses and systematic reviews, but if it's a strong enough trial that's adequately powered, do we need all the phase 3 trials?
Well, there's pretty good evidence from most pharmacologic drug studies that after early days of excitement from early kind of smaller clinical trials that might be deemed phase 2, phase 3. When you get into the large, robust phase 3, 4 trials, the excitement kind of dwindles. And so I think that, in order to be able to even get to the large phase 3 to make, people don't adopt. So adaptation in medicine takes 18 to 20 years from the time a clinical trial is done. And sometimes during that 18-year window, there's contradictory evidence that emerges that diminishes the excitement of the original ones. And so there's a lot of smaller early ones that might be powered as a single center, even a multi-center trial that's powered. How good is your power? Don't forget 5% of studies will still be positive, but they're actually truly false positives. And so really, I think the rigor of those studies needs to increase to be able to support it. And so, yeah, I think kind of some of the small, just because a study is positive, let's put it this way too. How many probiotics are competing in the same field? So if we do 50 probiotic studies for prevention of asthma, okay, how many of them would be positive by chance alone? And then does that company then get to say, well, ours was positive, let's go for asthma. And then another one is done on prevention of atopic dermatitis, and there's also 50 companies doing studies of that right now. And the list goes on, whether it be autism or other concerns, IBS, IBD, prevention of. And so the number of studies are massive. So the number of false positive studies, I haven't done the math, but we need a statistician who can run the data of, like, I can't remember how many studies, 40, 20, there's 1,000 some odd this year. So 50 some odd positive studies alone this year. Should we adopt them?
No,
So,
fair point.
right. So I think we need to be careful when we say a positive study was done and we should adopt.
Can we go from the premise that, and I appreciate the word you said, robust. You said if we have a well-designed study with a sort of consortium of knowledge surrounding it, whether that's preclinical, in vivo, animal, in vitro, supporting with a couple of good studies that might not be phase 3 powered, in the numbers, but that make a logical clinical argument and that show efficacy, would that be enough? Because we also now start to intersect at the level of efficacy, regulations, the legal aspects, the manufacturing, the upscaling. There's all sorts of issues at play, but all of that aside, what is enough? And if we're talking about a treatment, for example, we take neck, we take any of the conditions we talked about today. I mean, these are patients are suffering here, and that's.
I guess the question is, what do you mean by enough? Enough for clinicians to adopt? Actually, generally, clinicians will adopt. I find when the pharmaceutical reps drop the samples off at their office, the patients adopt because they're given free probiotics and they come into the emergency on probiotics because their family doctor gave them to them. So I don't know what you really mean by adopt. I mean, this is a longer topic of what drives medical adoption and maybe deeper than we want to go today in the interest. I'm happy to chat about it some more afterwards from my personal perspective. But
fair enough.
So I will say that we are technically over our time, but there have been 2 people patiently waiting in line. So if you can ask quickly, that would be lovely. Thank you.
So my name is Richard Ethier. I'm from Lamont Health Solutions. And I wanted to ask Dr. Friedman a little bit about, he had mentioned that one product not allowed to get an IND in the United States. And just maybe talk about what exactly were the issues there, because as far as I know, there weren't any manufacturing problems, and maybe a little bit about the differences in Canada and the United States with the natural health products.
did Dr. Tompkins suggest you ask the question? So there were the issues where I worked very closely with Laumont and Dr. Tompkins on the submission of the IND, and they were fully supportive of us getting the IND. The issue was we couldn't meet the purity requirements by the, I believe it was the FDA at the time. This is going back about 6 years. And the product master file couldn't meet that purity requirements, and we decided based on the decision made by Laumont was the production costs were going to be exorbitantly high to achieve those requirements, and the decision was made not to pursue the IND anymore. I don't know if there's something else you want to add, Richard?
As it says up there, how to take advantage of pediatric and other trial networks. With NHPs in Canada, you're allowed to make disease claims and run clinical trials. Probiotics are one of the classes in NHPs. I think this could be a very positive network for the FDA to use.
Well, a good plug for Canadian research. So yes, we were able to get NHPD approval and Health Canada approval for the conduct using that product with the purity data that we had at the time. from Health Canada. So perhaps it is a little bit more of a little more liberal on the requirements, but I'm not going to go too deep into the politics. Let's stay out of there for now.
Hi, I'm Carolyn Edelstein. I'm Executive Director of Open Biome. We're a nonprofit stool bank in Boston. I wanted to begin by saying that we're absolutely echoing the panel in that, in the long run, the right option for C. diff patients is to have, a rigorously tested product that has been evaluated and approved by FDA, and that we share the aim of seeing enrollment into the trials that will get us to that point. And on the other side, I think so in the 5 years since we've started the service of providing fecal microbiota products under the current policy of enforcement discretion, we've sent out about 38,000 treatments to a network of 1,100 sites. And that's, we're very proud of that work. We're also pretty surprised by it. I think by now we would have expected maybe faster progress in the field. And so I think, my question is really, is there more that we can be doing? I think Dr. I think you were the speaker who made the point that what's so unusual about FMT is that patients can do this themselves at home. And that part of the tension of balancing access to material and wanting to make sure that there's access to something that's been rigorously prepared and is available through the medical system is running up against this challenge of enrolling these trials. And I think there's probably more that Open Biome can be doing. There's probably more that we all can be doing to navigate that tension, and I'd be curious to hear the panelists' thoughts.
I would just like to say I'm impressed that you sent out 38,000, and I'm 9 years in and I'm still studying yogurt. And I still need an IND to study yogurt. So I think that says a lot about where we stand in the U.S.
So like Colleen at my site at the University of Michigan, we also use your product for our patients, and we provided the option. So we have the Open Biome product available, and we have directed donor stool available as well. And basically, shortly after we introduced your product, essentially nobody wants to do this through directed donors. And before that, we had that was the only option. We had to have the patients go and find their own donor, find their own stool, and they were forced to ask their neighbor or their pastor if they didn't have a spouse that would qualify and things like that. And so the patients didn't want this either. It's not like we were saying, oh, we have this Open Biome product and this is the only way to go now. No, nobody wants to do this stuff on their own. And nobody wants to go use their spouse's stool or their relative's stool or their neighbor's stool. And I think that physicians appreciate the service you're providing because it helps our patients a lot, and it also helps us in that, it's not on us now to ensure the quality of the product, to do all the screening. Even when we were doing directed donor screening, we just, the American College of Gastroenterology, there's been some position papers about what do you screen for as an individual clinician, and it's an order of magnitude less than what you guys are able to do and what you're able to offer as a centralized repository. And so I think keep doing what you're doing, look for regulatory guidance, operate within regulatory guidance, but ultimately we want a good, safe, effective product. So,
all right. Thanks to all the speakers. We're going to have to call it there. We are behind schedule, so let's do a 10-minute break. We'll start back up at 3:05.
Strains based on their therapeutic potential. So the idea behind this last session was to look at the rational selection, and have speakers address the rational selection of strains. And we'll hear a number of reasons for that based on their immunomodulatory properties, resolution to CDI, resistance to colitis, and even behavior modification. So there is going to be some preclinical work and some early development work. So we've organized this, and thanks to Neil, one of our speakers, had a great suggestion. We've organized it to go from talking about bacterial consortia to a pivot on how to isolate individual strains, and then some examples. And we've already heard a little preview to our final speaker on a LBP to prevent sepsis. So with that, I'm going to introduce my first speaker. His name is Bernat Oli. He's from Vedanta Biosciences. He's the Chief Executive Officer. I met Bernat back in 2013, actually, whenever we had a similar meeting. And it was just after the recent the release of the 2012 guidance on LBPs. So with that, I'll introduce Bernat, who's going to talk to us about drugs based on rationally defined bacterial consortia. Thanks, Bernat.
Thanks, Ryan. So, since the audience is very sophisticated for the average microbiome discussion, I'll skip all the background, other than to say that when I use the term defined bacterial consortium, to be precise, what I mean is that we make a product based on multiple bacteria that are made starting from pure clonal cell banks, not from a source material from a fecal donor. The way our drugs work, I think, should also be very easy to understand to everybody in the audience. We give them in capsule form as a lyophilized powder. It's been freeze-dried. The capsule releases the bacteria after the stomach, and the bacteria can colonize the intestine. In our hands, the colonization is important, and if the bacteria are dead, and if they're given dead, they no longer work across a range of problems that we study. And then we also see that depending on the bacteria that we pick, we can see them elicit a range of immune responses via the mucosal surface of the intestine, including both immunoregulatory and immunostimulatory responses. With the co-founders of the company, we've done, over the last few years, a range of work to try to systematically understand and explore which groups of bacteria in the intestine stimulate which types of immune responses. So, for example, starting on the right of this slide, this is work that we've done with one of our co-founders, Dr. Kenya Honda, now at the University of Keio, where we've identified groups of organisms that are potent inducers of regulatory T cells, and we're exploring this biology in the context of IBD in partnership with J&J and also food allergies. And all the way to the left of this slide, you can see counterexamples where we found bacteria also from healthy individuals that have opposite properties. They have the ability to induce either Th1, Th17, or cytotoxic CD8 T cell responses. In the continuum of approaches that are being pursued in the field, and that slide is not meant to be comprehensive, my view is that there's a fundamental trade-off between ecosystem effects and specificity. What I think ecosystem approaches, like, for example, fecal transplantation, bring to the table that's really unique is the ability to do something that would be very difficult to do with other drug modalities, which is change the composition of the gut microbiota in a somewhat controllable manner. I'll say controllable in quotation marks. And of course, at the other end of the spectrum, if you go full reductionistic, you can gain in specificity that you don't have with a fecal approach. But in our hands, we've seen that come at the cost of losing the polypharmaceutic effects of microbial communities and also the ability to robustly change the composition of the gut microbiota. And what we asked is, can we find some intermediate stage where we can still retain the ability of a community of bacteria to change the composition of the gut, but do it in a controllable manner and with more specificity. And here's an example of some work we've done, for example, looking at bacteria that can stimulate regulatory T cell responses. In short, finding that while you can identify a number of bacteria in the human flora of subjects across the world that have the ability to induce regulatory T cells, it's only certain assemblies in consortia that can really saturate the phenotype in animal models. So I think this brings me to a question that I think this is a good forum to bring up, which is how do we think about the contribution of different components of a drug to the final activity. And a lot of the work that we've done in the field has colored my opinion on that, and I'll emphasize its opinion. We've done often top-down work where we start with the full fecal community. We see that community has the ability to change the phenotype, for example, Treg induction or Th17 induction, and then we'll scale back and find when do we lose that activity. And usually, like you can see, for example, in the middle panel, we can identify fractions that are equally active or sometimes even more active than a full fecal transplant at saturating a given phenotype. And then there's a tricky bridge to cross when you try to really bring down that activity to the absolute minimum number of bacteria. What we often found in our hands, and we've seen that when we've looked at different phenotypes, including Treg induction, Th17 induction, CD8 induction, is that as we go really low in membership or diversity of species in a composition, we'll see often the effects wash away. And so we think that suggests that there's an important role for ecological redundancy within a consortium unit to help give a product or a specific composition the best chance of success. The reason I thought I'd bring this up is, in other contexts, we've had the discussion of, is this a combination product? Can you draw a parallel with, say, a multi-component vaccine? And I see a fundamental difference in that. If you pick the example, for example, of a multi-component vaccine, every immunogen is there for a reason. You know that it's targeting a certain pathogen, so the justification is straightforward. But when you're trying to change an ecological community, the issue of redundancy or the aspect of redundancy comes in. And also, the specific contribution of a strain is actually going to change from patient to patient depending on their starting microbiome. So it's not really an inherent property of the component of the product. This is in a snapshot the process that we follow today to try to identify new compositions of bacteria that we define as consortia. We try to triangulate between human, in vivo, and in vitro data to arrive at preclinical packages that give us confidence that we're not just chasing a correlation, but there's actually some evidence of causation, but at the same time, that we're not over-relying on animal models and chasing a causation pattern that has no relevance to humans. So we interrogate human data sets from studies that we sponsor or collaborate with clinical academics across the world, where we try to identify if often in the context of using fecal transplantation for a range of conditions, there's a pattern or any pattern of engraftment of strains from a healthy donor that may correlate with the clinical response. And I'll emphasize correlate because that data by itself doesn't tell us anything about whether those given bacteria may be actually causing a phenotype. If that's the case, then we'll often go into animal models and do systematic experiments to remove and reintroduce a full microbiota and see if a phenotype gets abrogated and then reconstituted. And if we then have confidence that that's the case and that therefore we're not just chasing an association, then we'll ask, okay, which are the bacteria that have the properties that we may be interested in? So for that, step number 3, we've created a very large library of bacteria from humans across the world, somewhere between 60 and 80,000 isolates now, sequenced the genomes of a few thousands of them, and also generated high-throughput assays to understand and characterize their properties, and I'll show a little bit more later how we do that. That gives us hits, or in other words, bacteria that have a specific property that may be useful, and then we still have to figure out how to assemble them in consortia that are more potent than the individual strain. And for that, we use a combination of bioinformatic algorithms that we've been publishing with collaborators at UMass, and also go back to the human data and ask ourselves from all the potential combinations which ones are actually occurring as co-occurring networks in humans that have a clinical response. We then have in-house our own manufacturing facilities to do GMP production of bacterial consortia. As our colleague from Series noted before, these are complex products. They have multiple ingredients. They're anaerobes. They're difficult to grow. They may need some customization. So we found it best to basically do all this trial and error work in-house, and there's a lot of it. And then we've moved in one of those consortia into human testing now, and we're just about to announce the results in the next few weeks, I think. This should give you a sense of some of the actual activities from culture collection, strain screening, drug substance production, and drug product production. So starting up on the left, we do a lot of high-throughput picking of colonies from donor material, which lets us go from fecal material to actual pure strains, from which point on you never again have to go back to fecal material as your source. On the upper right, you can see some of the high-throughput screening that we do to test multiple different types of bacteria or combinations of bacteria against activity assays or other forms of characterization to understand what the bacteria do. On the lower left, you can see some of the unit operations in the drug substance production, so that's in the GMP suite, where we do the fermentation, separation, and lyophilization. And then on the right, you can see some of the activities which we have in a separate facility for the actual drug product manufacture, which involves solids handling. And as John mentioned before, there's some challenges associated with that, so we have it in a different facility. And that's where we produce the actual final product that's going to be bottled and sent to the clinical sites. We have a a range of different projects from infectious diseases, immune diseases, including EDF, IBD, food allergy, and cancer immunotherapy at different stages. I'll use the first as an example to walk you as a case study through the steps that we've used in our platform. So the target product profile of B303, which is a defined consortium that we're developing for C. diff. This is an LBP that is administered as an enteric capsule. It has 8 pure clonal strains of bacteria as its components. The dosing regimen is repeated oral once a day following standard care antibiotic. And the number of days we'll treat for is one of the outcomes of a phase 1 study that we're running now. In terms of PK, we believe this is going to be gut restricted and not absorbed, and also we expect abundant strain colonization lasting for a window of time longer than the time it takes for most recurrences in C. diff to occur. We think that one of the key differentiators from antibiotic approaches would be of an ideal target profile, the ability to reconstitute colonization resistance after an antibiotic, but also potentially to start helping address the transfer of antibiotic resistance. We started this work, or just to follow as a case study the framework I laid out before. We've had an ongoing collaboration with the University of Leiden, where we followed a group of subjects that are being treated with FMT for recurrent C. difficile at any number of recurrences, and look at pre and post FMT samples to understand if there's patterns of donor engraftment that correlate with clinical response. And to make a long story short, we do see that there's a range. Basically, what you see in this heat map is on the x-axis samples from individual, either healthy donors on the right, C. diff subjects during treatment, or see if there's a clinical response. And on the y-axis, you see the different genera of bacteria represented. And again, to make a long story short, you see that C. diff subjects have a group of bacteria up in the top left that are largely absent from healthy donors, and then largely gone after a successful clinical response to FMT. And also that healthy donors have groups of bacteria that are relatively abundant and largely missing from C. diff active infection subjects, which you see on the bottom of this chart, but then get re-engrafted after a successful clinical response. Basically, we've made sure that the species that we select for B303 are representatives of this genera that are associated with clinical response. There's, I think, plenty of evidence in the field, some of it actually generated by Vince Young's lab, showing that use of certain antibiotics that are associated with C. diff infection can result in very extensive elimination of Clostridium clusters 4 and 14A, which are two groups of abundant bacteria within the firmicutes, which we found to be associated with better clinical responses. Our hypothesis is that by reintroducing those groups, we can restore colonization resistance and then render the host less susceptible to a reinfection. Some of the basic characterization that we do with the strains, starting with safety, is laid out here. We've conducted tests to determine the extent to which antibiotic resistance and virulence is transferable from a product strain to the surrounding microbiota, and that included assessing for the presence of antibiotic-resistant genes, virulence factors, and phages near mobile elements. And we mapped out their location with respect to predicted mobile elements, and basically found that there were, for none of the strains, ARGs or phages near mobile elements or ARG phase associations. And also we've tested the clinical sensitivities of each of the bacterial strains to antibiotics, and found that each of the strain products, each of the strain substances are susceptible to multiple clinically relevant antibiotics. And I think a relevant point here is this is one of the advantages of working with a fine material. You can design and control and make sure that your product actually doesn't harbor patterns of resistance or virulence that could be problematic. But there's also a limit to that. In this case, we've been able to find multiple clinically relevant antibiotics that can knock out the whole consortium at once. But just to make an obvious point, the larger the consortium and the more diverse genetically, the more difficult it's going to be to find a group of clinically relevant antibiotics that work for all the consortia at the same time, as opposed to for individual strains individually. So this may be like a little detail, but I think it's important from a regulatory standpoint. We've done a range of models, both in vitro and in vivo, to characterize the potency of each of the individual strains. Can they directly kill C. difficile or not? And also tried them in animal models. Actually, the model that Vince developed is the one I'm showing here, and shown that we could match the activity of a fecal transplant in animals by using a defined consortium. And now to wrap it up, we're in the process of wrapping up a phase 1A study where we've studied healthy volunteers that were pretreated with vancomycin in a course that attempts to emulate the typical courses of C. diff subjects, and looked at safety, PK, and PD in normal healthy volunteers. And in here, I lay out the objectives of the studies. We're looking for safety, tolerability, and what we would like to see is that this consortium of organisms can rapidly and durably colonize the intestine. We'd like to see them stay behind after you've given the last dose. We want to see abundant colonization. We also want to see robust colonization. And by that, we mean that all 8 bacteria colonize all the people, not some bacteria colonize some people and not others. And this is my last slide, just to make a point of some of the techniques that we've developed to be able to measure pharmacokinetics in clinical studies. We have the benefit, in contrast with the fecal transplantation approach, of actually knowing exactly what strains we're putting in. We have whole genome sequences for each of them. So we've been able to create a panel of markers for each of the genomes. And then when we look at stool metagenome sequences from fecal samples from the actual study, we can look for both the depth as well as the proportion of markers that we detect, and basically feed that to a statistical distribution to have confidence that what we're detecting is exactly the strain that we gave, not a close relative that happened to be in the person before we dosed them or acquired by the person after we dosed them. We think that some of these tools are going to be a basic starting point to start understanding PK in the field. And to be clear, when I use the word PK, I'm not talking about administration, distribution, method administration. I'm talking about colonization. How quickly, abundantly, and durably are the microbes staying behind? This has to start with having a reliable technique to measure the microbes you gave, not something that was already there to begin with. So I'll wrap it up here. Thanks a lot for your time.
Okay, so we're on time. We're going to roll along. We're going to hold questions until the end. The next speaker is Elaine Petroff, who's an associate professor and clinician scientist, ID physician at Queen's University in Canada. And Elaine is going to talk to us about the development of a defined consortia for recurrent C. difficile infection.
So, I'd like to thank the organizers for inviting me here to speak today. And I am going to talk to you about development of defined consortia, treatment of recurrent C. diff. These are my disclosures. And I'm just going to start with this slide. So thanks to Vince, I can skip a lot of my early slides and zoom along to the end. But I just wanted to throw this up here. I often use this slide when I give these talks, and every time I use a slide, I have to go in and update it and add another disease on here. So pretty soon I'm going to run out of room at this rate. But having said that, even though there's been an explosion in this field, I think everybody would agree that really the strongest clinical evidence is probably for recurrent C. diff when it comes to microbiome therapy. And what we see with this is basically ecosystem collapse. And on this slide actually is one of Vince's earlier studies back in 2008. Actually, I believe it's been 10 years. But basically, he was one of his group showed that recurrent C. diff patients have lower microbial diversity. And in fact, he showed the graph from this study compared to controls, but also compared to first-time comers who develop C. diff and then recover. And so this really illustrated how this subpopulation of patients that get C. diff is different. And these are the patients that do not respond as well to vancomycin. And there's been several studies now that have corroborated what was shown in the New England Journal paper when they showed that about 30% of the patients respond to Vanco, and the other 2/3 or 70% don't, and that's been since corroborated with a bunch of subsequent studies. And so what do we do with these patients? This is sort of how the whole transplant programs took off, at least at our hospital. And really what we're trying to do here is ecosystem repair. So I won't go through this in a lot of detail. It's already been covered by several groups before. But basically, we're trying to take a healthy ecosystem and put it in to replace or replenish what is essentially a sick ecosystem. And so by healthy, we mean diversity of species that provide functional redundancy amongst the organisms, so there's some overlap in some functions between these organisms, and it provides resistance to disease, as opposed to a sick ecosystem where we're dealing with low species diversity and an imbalance, or dysbiosis is the other term that sometimes we hear, which leads to an impaired function and a susceptibility to disease. This is made worse by giving patients vancomycin because, yes, it does clear out the C. diff that's in there, but unfortunately it also torches the porous, so to speak, and it kills the innocent bystanders, which are kind of exacerbating the problem when we can't recover those organisms. And so what we're left with is a ravaged ecosystem that really can't get back up on its feet. So what are some of the options? We've kind of gone through all of these today, so I kind of probably go through some of these more quickly, but I wanted to sort of briefly touch on all three. There are options for ecosystem repair being probiotics, FMT, or defined consortia, which is the approach that we're taking. And so with probiotics, at least for the case of recurrent C. diff treatment, I know we've talked a lot about C. diff prevention with probiotics, but as far as it goes for treatment, really there's no evidence that this is going to work. And if you think about it, it kind of isn't that surprising because a single organism or a few species of Lactobacillus or Bifido are really not enough to improve diversity. And in fact, if you put into a system that has very little an overload of a particular organism, you can also exacerbate the dysbiosis and cause even more of an imbalance. And this has been touched on a little bit with previous speakers. And also, I did want to point this out. I didn't hear anyone mention this, but this Annals of Internal Medicine paper, I don't know if anyone saw this, but it was a paper that looked at prebiotics, probiotics, symbiotics, and adverse event reporting. And in fact, they're grossly underreported when you look at all these clinical trials. And so this, this situation of dysbiosis and imbalance and the adverse events that you get with probiotics, I think probably is happening a lot more than we realize. And then finally, I was hesitating to put this trial in, but I think I'm going to throw it in, put the elephant in the room. So there was this paper that came out, which we've all alluded to, that came out in Cell. And basically what they showed was that the impact of the microbiome by probiotics is probably not really what we think it is. And there may be interference as opposed to enhancement of recovery of the microbiome. And so I'm just going to very quickly show you a few figures from this paper. And I would strongly recommend that some of you pull it, because it's actually a beautiful study. I'm just going to show you the human data. Actually, they did this in mice. They did it in 2 separate strains of mice, and they showed basically the same thing. But you can see from the design here that they split them into 3 groups, and these are healthy volunteers, and they took them at baseline. So the gray baseline, that's their naive microbiota before they receive antibiotics. And they give them antibiotics, And then they either got fecal transplant, probiotics, or nothing. So the nothing group is the spontaneous recovery. And if you think about it, that's kind of what we always do with patients. If someone comes in with a UTI, we'll give them antibiotics and send them on their way. That's generally how we've done it in the past. So when they then looked in the follow-up period, out here past 3 weeks, and actually they followed them out to like 5 months. And what they found is that the probiotics group actually had fewer species than the spontaneous recovery group, which is kind of interesting. And the same was true for the bacterial load. And fecal transplant is in brown, you can see here. And then if they looked at the communities, so this is kind of a busy slide, but if you just focus on the unifrac distance from unweighted to baseline. So what that means is the further away from baseline, so is basically shows a disruption of the community. And so if you look at here, you can see that the probiotic group is further away from the baseline of the naive gut microbiota of these patients than either fecal transplant or spontaneous recovery. Another way to look at that is in the PCA plots. You can see that the probiotics and the antibiotics groups cluster together, and over here you have the spontaneous groups with the fecal transplant, and the gray is the naive. So they cluster together. And so what this indicates, and then, oh yeah, this is another really cool thing that they did. So they had all these, this is the probiotic species that they used in the study, and they took them out of the analysis, and this is the supplemental figure. And basically they saw the same thing. I suspect a reviewer probably asked them to remove those just to see if it was an artifact and see if the data still held true when they took them out, and actually they saw the same thing. And so what this indicates is that the the volunteers that got the probiotics did not recover their microbiota to the same degree as the patients that got nothing or the ones that got fecal transplant, indicating that maybe you're doing harm without even realizing. And again, Mary Ellen's going to get mad at me for saying that, but I think it's worth discussing. It just is a good illustration of how we think that we understand what's going on, but maybe we don't actually fully understand and recognize what we're doing to the microbiota. And so what I came out of this, or concluded, is that microbes work better in teams. So if you have a few probiotic species that are acting alone, they may not be as effective as an ecosystem, which is what FMT is like. It's more like an ecosystem. And so there we've got synergy, and they're all working together as a team. And so really FMT is sort of the ultimate probiotic ecosystem. I won't go through this. We all know that it's effective. This is just one of the studies that we did with Christine Lee back a couple of years ago. And I think FMT is great. We've been doing them since 2009 at our hospital, but they do come with their own set of challenges. And some of these may be Canada specific, but I'm going to mention them anyway. So the first one, which has always made me nervous as an ID doc, is the risk of transmitting something. I know this has not happened yet, knock on wood, thank goodness. And I'm not saying from this slide that Zika is being transmitted by stool. I'm not saying that someone's gotten it from a stool transplant. The reason I put this up here is a patient actually asked me this question. And I didn't actually know what to tell her because she came to me with this, which is this Zika, don't give blood, you might have Zika. And then she asked about stool. Can I get Zika from stool? Because she was pregnant. And I wasn't comfortable with her getting a stool transplant once she pointed this out to me. So, you know, and next week it'll be some other virus. Like, it just seems like there's always something that pops up. And so, even though nothing's happened, I can't actually advise advise my patients that nothing ever will. This is sort of like with the HIV system, HIV situation with blood back when HIV was new. So that is a risk that still makes me a little uncomfortable. The other thing is that our public health labs have become increasingly resistant to doing the screening tests, which has not been very helpful. And part of this, I know, is probably because the screening, compared to 10 years ago, has actually become a lot more comprehensive. So if you look at the recommendations from the AGA, for example, from several years ago compared to what's come out more recently with the IDSA, there's a difference there in terms of what they're now recommending that we screen for. And our labs say that these tests are not validated to be run on healthy formed stool. That's the excuse that they give us, and they kick them back. But it puts us in an awkward situation because then we don't know what to do with this donor and we have to call them back. And then that leads me to my next point, that donors, like maintaining a stable donor supply, has been a major challenge. And in fact, at our hospital, we don't have any donors right now. I'm having to send people elsewhere because we can't get enough donors. And this came out, this is a joke. It's a program in Canada called This Hour Has 22 Minutes. But it's actually kind of true. We almost have to do stool donor drives the same way that we do blood drives to try to get people to come out and donate. And then quality control, that's a whole other interesting, I don't have any answers for this. And this was really driven home when we did this study. So we looked at a stool transplant donor that we had who has been very good at donating, and all of his stools that he's donated have cured the patients that we've treated with his stool. But we sampled his stool a little over a year apart. I think it was 12 months or maybe it was even 18 months apart. And the composition, you can see, even though you don't necessarily see all the different species and strains and everything listed on the side there, you can tell just by looking that this is not the same mixture. But having said that, it was effective in both of those. And so coming up with a generic stool is not going to be an obvious solution. So we came up with this microbial ecosystem therapeutics idea, which is basically a cleaned up stool transplant. And so we're hoping that it'll be more reproducibly, more like an FMT, but just more reproducible and better characterized. And we're emphasizing diversity, ecological resilience, which I'll talk a little bit about in a sec, and safety. And so we're looking at human gut-derived commensals, so a little bit different from probiotics. And this is not really rocket science, actually. I just pulled this off the British Columbia website. This is some forest ecology thing. The same principles would apply for a jungle in Costa Rica, and essentially we've just adapted these same ecological principles to the work on the ecosystems in the gut. And so this is our approach. We take precious fecal samples, we do detailed anaerobic culture, and then remove pathogens. We characterize what bacteria are in there, and then we take what we have after we've done all of that, put it back into the bioreactor and test it. And if the community holds together, then we would administer that to a patient. And the goal is to come up basically with a cleaned up stool transplant. That's kind of what we're trying to do here. And so what's unique about this is that it's one ecosystem, one donor. So we don't mix and match strains from different people and mush them up all together and put them in together. These have all co-evolved in the same person, so we keep that ecological principle intact. And we take out what we think would be undesirable to have in there, such as viruses, candida, all of that comes out. The bacteria that we have are identified. We check them for antibiotic resistance. Those also come out. And then once we have what's left, that's what we then test and put it into a bioreactor and see if it holds together. And this is just an example of one of these bioreactors. You may have heard the term RoboGut. It's also been used to describe this. So basically, it's an in vitro system that simulates the environment of the distal human gut, like an artificial colon is another way to look at it. And so you have food that goes in, and then waste that comes out. There's a stir here to mimic peristalsis. You can adjust the rate that it flows through, the same way you can sort of mimic the GI transit time, and it's all controlled temperature, anaerobic conditions, and pH. And this is sort of what it looks like as a photograph, and if we pull away all the wiring, you can see in the back those large volume vessels back there. So we inoculate identically at the same time, and then one serves as a test vessel and the other serves as a control. And we can run these for weeks at a time. And the other advantage that this one has over the smaller bioreactors is we can control pH and some of these other parameters that are a little more tricky to control with the small volume ones. And so this is just an example of optimization, something that we would do with this. So this is actually a fail. So this is showing you, but we, as we all know, learn more from our failures than from our successes. And so here I'll just run you through this briefly. So here we have fecal transplant material. So it's donor stool that gets inoculated into the bioreactor, and we run that out. And then we can hit it with drugs, or we can change nutrients. We can manipulate the conditions here. You can see that we've administered clindamycin. And so as long as this percent similarity index is above 90%, we consider that the ecosystem is holding together pretty well. If, on the other hand, here we have the mixture after we've taken things out. And so then we inoculate that into the bioreactor. And in the case of this particular ecosystem, you can see that it collapses. So after we give Clinda, it does not recover. So this would be an example of how we can fine-tune these ecosystems and test them for resilience and robustness. And we can use different drugs to do this, as well as different nutrients. And then the other thing that we can do is compare in vitro and in vivo. So we did this study as well, where we took our mixture, and then on day 0, we inoculated it into a chemostat, or a bioreactor, and we also inoculated into a patient. And you can see here day 14, sample from the patient, and day 12. They don't look exactly the same, but they're starting to look similar to each other. So we think that this bioreactor represents a good surrogate for in vitro, in vivo work. These are some of the animal studies we've done. I'm not going to go through those, but those are just the references. So we've done C. diff, Salmonella, and DSS colitis. So this is the study that we did with the humans, and you can see here Lactobacillus and Bifido are in here, but they're not the main ones. They're part of the team. And then this is just data showing that the at 6 months period of time right here and here for these two patients, we have a composite mixture of the bacterial composition of the pretreatment native flora or microbiota from the patient and the Repupulate mixture, the microbial ecosystem therapeutic that we put in, showing that these do colonize. So next steps, we have a new ecosystem, new donor. We've actually expanded to more species, and this thing is a monster. It's like got a lot of different very interesting bacteria in it, and it's it's a clinical pilot study that's currently underway. So just as a summary, what we're doing with this stuff that we think makes it a little bit unique is the ecological principles that we're using to develop these mixtures, known composition, diversity, patient safety, and this came up earlier. Outcomes can be tracked, and now we can link them back to a specific bacterial composition because we know exactly what's in there, unlike what we were talking about with stool and having the stool registry. And whoops. Good. Oh, he's doing it. Anyway, my acknowledgments, both Canada, US, I just wanted to acknowledge all my collaborators.
Thanks, Elena. Appreciate it. Okay, so moving from defined consortia to finding a needle in a haystack, where our next speaker is Neil Serrana, a freshly minted assistant professor of pediatrics and molecular genetics and microbiology at Duke University. He braved the hurricane to come to us, and we were on call to give it a webinar presentation, but we're really happy to have Neil. Thanks.
Thanks very much, Ryan, for the invitation to come as well and to get me out of a rather wet Chapel Hill right now. There's been a lot of talk about how do we move forward in the field, and I think, as has been mentioned by many so far, there's an issue in the field of moving from associations to and correlations to causation. I showed this. This is a study that Dirk Evers and Rand Xavier did a number of years ago, where they looked at pediatric patients with new onset Crohn's disease and identified a large number of different genera and some bacterial families that were either more or less abundant in patients versus controls. And the question with these is always, where do you go from here? And you see all these associations, but how do you either define a consortia, or how do you define organisms that actually matter? And so it's this question of how do you go to causation that's been challenging. If you think about this in a different way, you can picture the microbiome as a haystack, where each individual piece of hay is a different microbe that's there. And I think all of the work that's been described so far today has highlighted the fact that there's something there. And the poster child for this is really FMT, particularly for Clostridium difficile infections. You know there's a needle in there, and there may be multiple needles. But how do you actually find that needle? And is there a better way than FMT to go about it? And so what many in the field have been doing are these microbiome-wide association studies to basically subset the haystack. And you go from a large haystack down to a smaller haystack. And we know that there's a needle in there too, and again, there may be multiple needles. I should say I wanted to update this picture as Ryan mentioned, I just moved from Boston to North Carolina, and I wanted to update this with pictures of my own haystacks. But the weather of the last few days didn't really allow for that. And so if we, instead of going to these smaller haystacks, essentially, can we just find the needle itself? And along with this, though, it sort of presupposes the idea that the needle is better than the haystack. Just to think about this, if you think about FMT, at least for Clostridium difficile, it's being tested for a large number of other indications. It has biological activity. As many have talked about, there's questions about whether it's reproducible or not, and there's I think a lot of the conversations in the Q&A sessions have highlighted some questions around the regulatory aspects of it, batch-to-batch variation, if you will. When you think about bacterial cocktails, many of these issues are resolved, and it also, from a company standpoint, also improves patent position. But when you think about single isolates, you get all of that and potentially more. And I don't mean these check marks to be completely black or white as they appear here, but sort of at least one man's opinion as to which one offers a little bit more benefit or not. And one of them being that a single isolate, or with a single isolate, it may be a little bit easier to define a mechanism underlying how this organism impacts disease overall. And if one can identify a mechanism, then that allows you, as people brought up in a different session, to perhaps do precision medicine with microbiome-oriented therapeutics. So if you know that the organism of interest acts through a certain mechanism, you can identify patients that have a defect in that pathway, and then target that patient population specifically. If there's only one organism being given, it, in theory, at least has lower potential for side effects than giving 8 or 10 or 20 different organisms at a time. And also, I think, it allows the possibility of defining specific molecules from that bacterium that can then be used in sort of a classic drug development process. And so if FMT is essentially the iPhone, if you will. Ultimately, we'll get to the iPhone 10X or 10S, which is the bacterial molecules themselves. But how do you choose these strains, really, is I think the issue that has come up sort of repeatedly over the sessions so far today. In a work that in Dennis Kasser's lab, published a year ago, they approached this question from a fairly reductionist point of view. So they took a diverse, phylogenetically diverse set of organisms, 53 highlighted by the stars around this cladogram, and then generated mice that were monocolonized with each of these, and then really did an absurd number of immune phenotypes for each of these monocolonized mice, performed correlations among all of these different immune phenotypes, and then created a dendrogram based off of those correlations. And what you get in the end, though, is you look at these are color-coded now by phylum in the squares and by genera in the circles. And even if I don't tell you what these genera are, because there are too many to really make it a meaningful key, what becomes apparent is that the taxonomy doesn't really correlate with the immune phenotype, either at the phylum level or the genus level. And for many of these species, multiple isolates of the same species were used in these experiments, and they gave different results. And so I think this highlights that not only one can't just infer because Lactobacillus is a commonly used probiotic that it will have the same activity as a different Lactobacillus species. And even if you say Lactobacillus reuteri, a different strain of the same species may have very different functionalities as well. So then it gets back to this question that how do you choose? How do you find that needle overall? And I think what we realize is that all of these microbiome-wide association studies share a lot in common with genome-wide association studies. They have a lot of the same strengths and some of the same weaknesses. But GWAS studies are really an outgrowth or an adjunct to what geneticists have been doing for decades, which are family pedigree analyses. And there, a geneticist will identify a patient that they think has a hereditary disease, look through the family pedigree, identify other family members that have the same disease, look through their genomes, and identify regions that are shared in those with disease, absent in those without. If you do this over enough family pedigrees, you can really hone in at the gene level. And so we reason, can we do something analogous to this for the microbiome? And so, as proof of concept, we use mice, which, as Vince pointed out, it makes it a little easier. Now the colors represent different microbiomes, and we can take mice with different microbiotas, put them in the same cage, take advantage of the fact that they're coprophagic, they eat each other's poop, and now we generate mice with a hybrid microbiota that is reflective of its parental microbiomes, much like a child has a genome reflective of both of its. And so with this, if the microbial effect on disease is dominant, we should be able to triangulate microbes that are associated with the phenotype of interest. And so as proof of concept, we had multiple genetically identical or at least related strains of mice with different microbiotas. In red, germ-free mice. In blue, a strain of mice with a mouse microbiota that we've been breeding inside of notobiotic isolators for about a decade. Ones with a human microbiota, again, bred in isolators for about a decade, and then just wild-type mice that we bought from a vendor. And what you can see, these are experiments done with DSS colitis and showing basically just survival. Number of mice that, in 2 cases, they all died. In a couple of cases, they virtually all lived. And so we can take this very stark phenotypic difference and now do microbiome-wide association studies. And if we focus just on these parental strains of comparing either the wild-type mice that we buy from vendors versus the notobiotic mice with the mouse microbiota, or the ones with the human microbiota versus the mouse microbiota, there's still 100 to 160 different taxa that are differentially abundant between these groups, which again leaves us with the question of what do we do next? How do we choose which organism to focus on? So we use this idea of microbial pedigrees. I'm not going to go through all the data, but we found that if you co-house these mice just for a day, that in both sets you get intermediate phenotypes. The mice that used to die now live a little longer, and the mice that used to survive now die quite a bit more. But again, this only gets us down to 60 to 90 different taxa that are differentially abundant. But we applied an additional criterion that geneticists would do with the family pedigree, which is look for things that are shared among all 4 comparisons. And when we applied that additional criterion, only one thing came out, which is the bacterial family Lactospiraceae, which was associated with survival from DSS colitis. And importantly, even though all of this was done in mice, our results mimicked what would have been shown in humans. Again, this is just going back to that same study by Dirk Evers and Renwick Xavier that found that Lactospiraceae were decreased in patients with Crohn's disease. So our mouse data at least has some relevance to the human cohorts as well. We went through, and much like using a scenario similar to what Bernat described for what Kenya Honda had done, several different examples, we defined a bacterial community, a bacterial cocktail, that enriched for Lactospiraceae, gave it back to our colitis-prone mice, demonstrated that would protect mice from disease. But then we went ahead and tried to pick single colonies and identified one species that fell within the family of Lactospiraceae, happens to be a new bacterial species that we're calling Clostridium immunis. As a control, we chose a different bacteria, Clostridium inoculum, gave both of them to our colitis-prone mice. Those that got the control organism still all died with the same kinetics. Those that got the Lactospiraceae isolate are now protected from disease. I should note that this is done with a single gavage of these organisms 1 week prior to challenge with DSS. As I am not a company, I have not done all of the dosing regimens that one might be able to do to sort of see if we can improve this from 60% survival to 100. But proof of concept is that we could identify organisms using this approach that down to a single species that is protected from these experiments in a causally related manner. And so what we were able to do is use this concept of microbial pedigrees or microphenotype triangulation to bioinformatically pinpoint a limited number of taxa that are associated with our phenotype. And by doing this, we increase the specificity of our results at a cost of sensitivity. So we may not be identifying everything, but the ones that we do identify through this approach are more specific to the phenotype of interest. Using a directed microbial culture techniques, are able to isolate the organism of interest, add them back to our mice to demonstrate causality. And Vince earlier this morning had mentioned Koch's postulates, and we've now sort of demonstrated Koch's postulates with a commensal organism. Even though the Koch had intended these to be for the identification of pathogens specifically, I think that these really need to start to be applied to study of commensal organisms as well to really add to the scientific rigor within this field as well. In data I'm not going to show, we've used this same approach to identify other organisms that are able to induce post-expression of antimicrobial peptides, again, in a causally related manner. So this isn't a one-off result, but we've been able to do this for at least two different phenotypes, and now applying it to several others as well. So in the big picture, though, even though we did this with mice, the approach itself is it can be applied to human cohorts as well. And so we can look to our patients, identify pedigrees that matter, to then identify taxa that are related in a causal manner to the phenotype of interest, use concepts of microbial pathogenesis that have been honed over the last century to identify the bacterial factors from these organisms that mediate the protection, and then go through a standard drug development process to develop those. I just want to end with the idea that I think really we've just scratched the surface overall of the roughly trillion or so bacterial species that live in the world. There are about 10,000 or so in the collective human microbiome. There's a very small, and we can quibble about this number, but clearly less than 100 or so different immunomodulatory bacterial species that have been identified, with only a couple, very limited number of bacterial molecules that have been identified to date. And so there's clearly work that needs to be done at all of these levels, as well as trying to understand how to translate this. With that, I will stop.
Okay, so we're going to transition to the next talk and actually hear about one of those molecules. Greg Bates is a Senior Vice President of Regulatory Affairs at Axial Biotherapeutics, officially my favorite biotech company name, Axial. And the title of his talk is Bacteroides fragilis, used in a mouse model of autism.
And thank you for inviting me. It's been a very interesting day today. I'm looking forward to more discussions as we move on. So I'm going to follow what Neil said by talking about maybe trying to identify that needle in the haystack. And I think we potentially may have identified one of the needles, but I think there's probably multiple needles in multiple haystacks, which with different needles being important in different diseases. But Axial Biotherapeutics, the company that I work for, is a reasonably new company, and we're looking at the gut-brain axis. So we're trying to determine the connection between the microbiome and neurologic disease. We're specifically focusing on neurologic diseases that have a gut component to them. So that's what that slide says. We're really trying to focus on that gut-brain connection to figure out how we can manipulate the microbiome to help treat neurological disorders that may have a causality in it. The work that we're doing at Axial is based on some of the groundbreaking work that was published by Sarkis Mazmanian at he has published quite a bit on the connection between the gut and the brain, and the connection between microbiome and neurological disease, and understanding what those connections are. He has published data in ASD, autism spectrum disorder. Today, we also have a program on Parkinson's disease that's also implicated as gut influence. We have 3 programs that are expected to be in the clinic within 12 months. Today, we don't have clinical data, so I'm going to be talking to you about the preclinical data that we have that lead us to believe that we're onto something. When we do get to the clinic, our initial clinical focus will be try to look at objective biomarkers, as well as a GI function, because autism does have a GI well, which is...
Can you please speak into the microphone, please?
Sorry. Which is linked in severity to the neurologic symptoms that you see as well. So our target, again, is to look at the microbiome and its effect on the neurological disease. And we're trying to target therapies that are focused at the gut rather than the traditional way of treating neurological disease by giving systemic therapies for obvious reasons. Hopefully improved safety, decreased systemic exposure, getting around PK/PD issues with systemic therapies as well. Our therapies are both live biotherapeutic products as well as small molecules that are based on some of the activities that the microbial organisms that we're targeting may have in the gut. Generally, our approach, and this is our approach for Parkinson's, not so much our approach for autism, which I'm going to talk about in a bit, is to transplant a diseased microbiome from a person with neurologic disease into a germ-free mouse to see if we can create disease. So, for instance, in Parkinson's disease, if you take the feces from a patient with Parkinson's and transplant it into an alpha-synuclein overexpressing mouse model, you can actually create the symptoms of Parkinson's in the mouse. So that allows us then to have a handle that we can grab onto to try to figure out what is it in that microbiome to cause these symptoms. With autism, I'll talk a little bit about the work that Sarkis did and that we've continued at Axial in the autism area. But let me first talk a little bit about autism itself. So autism, as many may know, is increasing in prevalence quite a bit. It's currently estimated that it affects about 1 in 59 children. This has increased substantially over the last 10 to 15 years. The CDC, when they come out with their reports every couple of years, it goes up every time. And this is more than just an increased diagnosis. It seems to be increasing in the population in general core behavioral deficits, really autism spectrum disorder is a spectrum, so it's a heterogeneous disease, but these cognitive deficits in children have certain things in common. And the core behaviors are impaired social interaction, impaired communication, and they have repetitive stereotyped behaviors. There's a number of comorbidities that go along with that, irritability, anxiety, and GI symptoms as well. There's currently no drugs approved for treating drugs or biologics approved for treating the core behaviors of autism. There's only 2 approved drugs right now. That's risperidone and aripiprazole, and they're approved for treating the irritability associated with ASD. ASD is a wide open field. People have done studies using all sorts of interventions, including FMTs, probiotics, you name it, with varying degrees of success. They know why one works and why the other one doesn't. Matter of fact, there have been some FMT studies where the Fragilis, which is the organism that we're using, has been in the FMTs, but very inconsistent results with that. Again, going back to autism, autism is also a disease that occurs much more commonly in boys than in girls, about 4 to 4.5 times more likely to be in a boy than in a girl. Certainly a reason to that. There's probably a genetic component underlying autism with environmental factors kicking off the disease, as there may be with Parkinson's as well. It can be diagnosed as early as age 2, and kids start showing symptoms very, very early in life. Importantly, there is a subgroup of subjects with autism who have abnormal GI function. Some have diarrhea, some have constipation, bloating, abdominal pain. It varies from child to child. But there is definitely a GI component to the disease. So when you look at kids that have ASD, you try to look at information that correlates the gut microbiome to ASD. First of all, you see that, again, there are a number of kids with ASD that do have GI component to their disease. If you look at the microbiome of these kids versus a neurotypical child, there are differences. There's lots of publications on what the differences might be, and many of them are different from one another. So there is no fingerprint microbiome of an ASD child. There has a tendency to be less diversity in kids with ASD, particularly in the bacteroides and the clostridial components. But there is no current fingerprint as to what in the microbiome is causing ASD. Many risk factors that occur in kids with ASD. Mothers that have infections when they're pregnant have a higher risk of having ASD kids. Antibiotic use has been associated with ASD, birth by C-section. A lot of the things that we hear associated with microbiome type diseases. The kids that do have GI symptoms also show alterations in their intestinal permeability. So if you do a LAMA test on a kid with intestinal permeability, with intestinal problems with autism, they frequently will have impaired intestinal permeability. So we started trying to think, okay, well, how can these things be connected? What is the connection between the gut? We can't identify a specific organism or even a specific microbial fingerprint that's associated with ASD. What could it, from the gut, be affecting the central nervous system? So people have looked at the metabolomics around ASD as well. And there have been a number of papers written about uremic toxins. Urinary paracresol has been published on. It's found to be elevated. And there's also literature out there on 4-ethylphenyl sulfate, or 4-EPS, which is a close analog to paracresol that is also increased as well. They're very closely related molecules, and we're measuring both paracresol and 4-ethylphenyl sulfate. So we looked ourselves at a cohort of ASD children who were part of the CHARGE database at UC and looked specifically at the metabolomics of those kids and identified that in a subset of about 33%, about a third of the kids have a significantly increased level of 4-EPS circulating. The microbiome sourced uremic toxin. So we've looked at a number of different cohorts now and have been able to reproduce this and replicate this in other cohorts of kids. And it does provide a potential stratification opportunity in doing clinical trials. Look at high 4-EPS children versus low 4-EPS children. The issue in autism is that there's a very limited amount of cross-sectional data in the autism population identifying how much one autistic child will differ from another, and what sets these subgroups of kids with that symptoms apart from others. So the treatment hypothesis that we're looking at Axial is the effect of the metabolites getting into the circulation and affecting the and the fact that these kids with gut problems have impaired intestinal permeability, have increased intestinal permeability. So Sarkis in his lab had been doing a lot of work with B. fragilis, with Bacteroides fragilis, and had shown that Bacteroides fragilis, as well as a number of other Bacteroides molecules, do have an effect at improving the intestinal barrier and decreasing intestinal permeability. So he started to investigate B. fragilis in mouse models of autism. So a little bit about Bacteroides fragilis. B. fragilis, again, is a compound that Sarkis had worked with before. There are other Bacteroides that also have an effect on the intestinal barrier. Beta iota micron also has an effect. B. fragilis was chosen because he had a specific strain of B. fragilis that he'd been working on for quite a while that was non-toxigenic. Again, B. fragilis is not B. fragilis. One strain is different from another. There are enterotoxic strains of B. fragilis that can actually cause disease. The specific strain that we're working on here is specifically non-toxigenic. It's not capable of producing an enterotoxin. It's a non-spore-forming gram-negative commensal. It's very prevalent in the adult population, 50% or more. Actually, it's been shown in some studies to be as high as 90+% in children, which decreases a bit as kids get older, down to the 50 to 70% in adults. We believe that the organism functions in part by a direct interaction with the intestinal epithelial cells. So hopefully, this organism will help to improve the intestinal barrier and decrease the exposure of the systemic organism from these toxins that may be usually cause the symptoms, or just release some of the symptoms causes. So B. fragilis, first of all, when you look in vitro, it does have the ability to repair epithelial cell barrier integrity. So I think I'm a little bit taller than the microphone. If you take a Keiko-2 monolayer and you disrupt it by exposure to cytokines, and you increase its exposure to B. fragilis in a dose-responsive manner, you see a repair of the integrity of that barrier. So this is an interesting finding in vitro. So in vivo, we see the same thing. So the rest of the data I'm going to show is from a model that's called the MIA model, maternal immune activation model. And this goes back to the clinical notice that pregnant women who get maternal infections have a higher risk of developing or having ASD children. So this model is basically taking a pregnant dam and injecting the mouse with poly-IC, which is a viral mimic, a double-stranded RNA viral mimic, and causes an immune activation in the mother. when the offspring are born, by 3 weeks of age, you start seeing leaky gut, and you will also start seeing symptoms of autism. So when you take these mice and expose them to E. fragilis, you see a improvement. Let me see if I can. So this is measured using FITCI dextran, which is a radiolabeled dextran which permeates across the intestinal wall. If you use DSS, you see a great increase in the permeability of the gut barrier. The S here, that is a wild type mouse. The P is the poly-IC, so that's the MIA offspring that have a naturally leaky gut compared to a non-treated wild type mouse. If you add B. fragilis to it, you see a substantial decrease in the intestinal permeability that you get with that model. And on the right, these are tight junction protein staining, and you can see again, if you add B. fragilis, you get a repair of the tight junctions. So then we look at the symptoms of autism that show up in these mice. And you can really examine in these mice what are correlates to the core behaviors that you see in children with ASD. So again, one is repetitive behaviors. So mice bury marbles if you put marbles in their cage. And mice that have been offspring of in this MIA model. So again, if you get here onto the left, these are the wild type mice. On the right, these are the mice that are MIA mice. So if you look at standard wild type mice, they bury about 30% or so of the marbles in their cage. You give them some B. fragilis, it's not really different. The MIA mice, their marble burying behavior goes up to approximately 45% or so of the marbles. They have an increase in this repetitive behavior. You give them B. fragilis, it brings them back down again to what the normal level was in the control mouse. This is a measure of anxiety and locomotion both. This is a open field exploration test where you put a mouse in a little box and you have a camera on him and you see what you measure what he does. A mouse that has greater anxiety will hang around the edges of the cage, around the edges of the box, whereas a mouse that has less anxiety will spend more time in the center, in the open area, less protected from the walls of the cage. So MIA mice spend much less time in the center of the cage. So if you look over here, these are the numbers of times that the mice enter the center of the cage and the amount of time that they spend in the center of the cage. And you can see again in the wild type mice, they're here. The MIA mice, they have has a significant decrease in the number of entries to the center and a significant decrease in the duration of time that they spend in the center. And if you give them B. fragilis, it puts them back to where they were before. They get a more normal phenotype. And that's not because of an effect on locomotion, because if you measure the distance traveled for these mice, it's the same for all of them. So this is really mice having less anxiety and going back into the middle of the cage again. Communicative behavior is another of the issues that one sees in kids with ASD. And again, this is also replicated in the mouse model. You get mice, when they're together, make ultrasonic vocalizations towards each other to communicate. In the MIA model, which is here on the right, if you look at untreated, they have a substantially lower number of ultrasonic calls, vocalizations, and the duration of those vocalizations goes down substantially. When you treat them with Bacillus fragilis, their number of calls goes back up to normal, and interestingly, the duration per call actually goes above the wild type. There's something that B. fragilis is doing here to increase and improve the communication as measured through ultrasonic vocalizations. And if you look at 4-EPS levels, the data here on the left is from the MIA model. You'll see that in wild-type mice, they have virtually non-measurable levels of 4-EPS. In the poly-IC treated offspring, you see a substantial increase. It's about a 46-fold increase, and this is the most dysregulated metabolic product of the gut that you see in these animals. When you treat them with Bacillus fragilis, their level of 4-EPS goes back down again. So the hypothesis was that perhaps 4-EPS could be one of the factors that's traveling from the gut to the central nervous system and causing these changes. So if you actually expose animals just to 4-EPS, you see impairment in communication, increased repetitive behaviors, and increased anxiety as well. And this happens whether you give them 4-EPS orally, you gavage them with 4-EPS for a period of time, or whether in this data here is from dicolonized animals that were dicolonized to specifically produce 4-EP in their gut. So 4-EP appears to have an effect. B. fragilis appears to have an effect on the intestine. We'll test this in the clinic next. We've had our pre-IND meeting already, and within the next, hopefully 12 months, we'll be in the clinic and see if we can test this hypothesis in humans. Thank you.
I think I need some B. fragilis. We're about 20 minutes over here, so we're going to try and get back on time. So with that, our next talk is from Pinaki Anagrihi, who's going to talk about, who is a pediatric infectious disease physician and professor and founding director of the Center for Global Health and Development. And he's going to talk to us about a very large study done to look at preventing sepsis using a strain of L. plantarum. With specific focus on the timing and the why he particularly chose this strain. So,
thank you for bringing me here. I don't know if I will ever see NIH and FDA under one roof and so many elite group of people listening to me. I don't know if there is anything left, because after hearing so many wonderful talks that span from bioinformatics, machine learning to NNP of 3, FMT8, I don't think there is a whole lot left for me to add. And I'm not going to talk to you about neonatal sepsis and in the developing world setting how bad it is. I get carried away. I spent half an hour telling you how bad it is. 1 million deaths and the morbidity. And even in this country, if you look at the NICU, sepsis continues to be a big problem. And it adds to, if you give them antibiotics, it adds to increased incidence of TEC. So it's a bad disease, and there is every reason to study and do something about it. As I go through, I will have to speak fast, and I will show you many pictures so that you can visualize what was done and try to summarize my work in 15 minutes. That took me about 25 years, give and take. And you can think if you're thinking about drug or biologic or supplement, how do you develop a new one? You just pick one, somebody tells you that it will work, or you learn about the pathogenesis. That's why you think about it, or I just do it for the fun of it. I don't know. Most of us, I think, we know about the disease a little bit. We know about the pathogenesis, and we try to address it when we develop a new drug. Quickly, I want you to think about the history of, because we are talking about microbiome, probiotics, about the history. And then I have to talk about necrotizing enterocolitis, although my topic is sepsis, because they are quite related to each other, and that is how the whole development took place. And then I will describe you the randomized clinical trial where we use a Lactobacillus plantarum strain along with fructooligosaccharides. And finally, hopefully, because I'm the last speaker, we'll be able to tell whether Seabird or Sipsan who wins. This picture, many of you who are familiar with the probiotics field probably know, but think about it. In 1982, he drank cholera during epidemic, and he showed that he could survive. I'm trying to make a point that it's about 100 years. And another gentleman, more or less, give and take, 100 years ago, did something good, for which we all remember him. And this is only 30 years ago, I think 35 years ago. Here, I'm giving you why necrotizing enterocolitis and sepsis, because all the studies that have been done, fortunately, they look at NEC and sepsis together, although the primary outcome could be one of the two. And the purpose of putting it together is in 1999, the first study came out, which was a kind of a soft pre-post study, but it told us that probiotics may work in preventing NEC. But half and half, some of them show some efficacy and half of them don't show anything. If you look at the more recent ones, it hasn't changed a whole lot. Even the last very, very recent papers, reviews that have been written, you can see only some work, and others don't, and sometimes there is even negative effects. This is in Omaha. There are 4 NICUs, and 3 of them have been using probiotics for 5, 6, 7 years, and each one of them is using a different type of probiotics. If you ask them, have you done any pre-post numbers, they don't try to address those. And NEC is a multifactorial disease. Everybody was thinking, even now they think that it's a triad of ischemia, bacteria, and dysregulated immunoinflammation, and no specific agent has been incriminated in this disease, though. It happens very quickly, and if you, as Dr. New was telling this morning, it's a bad disease. But this was in early 90s when I started looking at the disease, trying to find out what organism may be present, looking at just very tightly matched controls and NEC cases. And we didn't find anything. And we published that, okay, there is no specific agent. We were looking at bacteria, and by culture techniques, it was that we could find. But we did show that the colonization pattern may be important in causing or preventing necrotizing enterocolitis using simple KK2 cell culture model and Wheeling rabbit ileal loop models. Here on the left in the panel A, you see gram-negative E. coli. You throw them, some of them, the ones that come from NEC babies, attached in massive numbers. In the panel B, you have gram-positives, like Enterococcus faecium in this particular case. If you put them together, you don't find too many of E. coli attached there, but you find some gram-positives still there. Then we did some translocation model. Again, the same thing. We have small cups where you grow cells, and you put gram-negative, they translocate. If you put gram-positives along with it, they don't. And the transepithelial resistance goes down, and the protective phenomenon was quite visible, that gram-positives do something good there. So we wanted to do some animal modeling, and I don't know how many years I spent. I was a junior faculty, and we had some fellows looking at mice and rats and newborn mice, newborn rats, and none of them worked for this model. Then we went to rabbit ileal loop, which didn't work. Then we took brought some pregnant rabbits to the facility and looked at weanling rabbits. And I was I made sure that we don't compromise the vascular supply, and we made loops in weanling rabbits, and then we injected E. coli into the loops, or we injected E. coli and Enterococcus faecium or staph epi in combination the same amount, and they recovered overnight, and then we sacrificed them and looked at the pathology. The saline injected loops, normal histopathology. If you put E. coli, this is how it looks like. You don't have to be a pathologist. And if you put the exact same amount of E. coli along with some gram-positives, and you have some blunting, some submucosal edema, but no injury at all. So that told us that the gram-positives are really doing something good. So we could conclude that normal flora gram-negatives, E. coli, Klebsiella, they did not belong to any pathogenic groups, could produce disease. And bacterial attachment and translocation were key. And if we put some gram-positives, mostly coag-negative staph and enterococci, we can protect disease. And in these models, and in NEC patients, we always have sepsis, and the same thing happened in the rabbits. Whenever we had NEC, we will be able to culture the same organisms from the ear vein blood of the rabbits. So, can we give enterococci and staph epi to our babies? Obviously, no way, because those are pathogens for preemies in the NICU. And we thought, how about probiotics? Maybe we can. We just heard the term and which probiotics to use. And so many are there, and some of you who have gray hair might have heard about the story of Lactinex, which was a FDA-approved drug. Then it was taken off of the drug route, and it was put back on the OTC. So what to use? We had no clue which one to use. And nothing against any of the manufacturers, and I love them because of them we are here today. And the field has progressed to the extent. But you see what Align says: constipation, diarrhea, urgency, gas, bloating, and cultural, and all of them have different types of mixtures, and we had no clue as to which probiotic to use, and no wonder people called it snake oil sometimes, and I was interested that whatever bacteria we use, it should go and colonize and do its job, and none of the studies would ever talk about colonization, what happens, why did you take that particular strain, because it was available, we took it. And so without colonization, I was not comfortable doing any real studies. So these are all, by the way, funded by NIH, either Fogarty or NICHD, and with small funding from Fogarty, we did a study where, as usual, we took LGG because we thought it will colonize, and it didn't colonize at all. Then we took some sporogenes, which was called Bacillus coagulans, that also didn't colonize, and so in the mid-90s, if you know about, think about forest gum, that is exactly what was happening. We didn't really know. Were we really picking them up from the box of chocolates? I think that was happening. So I said, okay, go to hell, I don't care. We will screen we have a model, we have an in vitro, in vivo model. We will screen the strains before we think go beyond, go to the clinic. So we screened about 280 plus strains from the market, from here, from former Soviet Union, healthy stool, and all different sources. First focused on bifidobacteria, none of them did anything in our model, specifically in our model. Then we went to Acidophilus because that was known. I knew about Acidophilus. They didn't do much. Then came Lactobacillus plantarum, which showed something, and we had quite a few. I didn't even hear the term plantarum at that point in time. I said plant, and many of these were from baby stool. I said, why should the baby stool have plantarum? And finally we found that there were, and this is just the adherence picture I'm showing you, which was our first screening, and then same way they were taken through the animal modeling, and we found that there was one strain of plantarum and one strain of salivarius that did the job that we wanted them to do, i.e. stopping bacterial attachment and translocation and injury in the winding rabbit ileal loop. Because salivarius also translocated in our system, we didn't want to give it to babies, so we moved forward with Lactobacillus plantarum, and we had to do the typical toxicity studies, and instead of doing it in rodents, we went back to the newborn rabbits, day one. These are newborn day one rabbit pups, and fed them for a month, and then took it to the clinic where we did a first phase 1 type study in 33 infants. It was a 2-to-1 allocation where we gave Lactobacillus plantarum plus fructooligosaccharides, and it colonized really well. After giving one week of therapy, they got colonized for about 4 months. Then we did a slightly larger study, which can be called as a phase 2. This was also 2-to-1 allocation in about 280, which showed some impact as expected, but the sepsis, as you know, the incidence is pretty low if you look at the regular population. Then finally came the phase 3 trial after the success of phase 1 and phase 2, where we launched the field trial. It was a 1-to-1 allocation and a large trial that got published last year. And it was an individually randomized trial in the community setting in India. And we wanted to reduce sepsis by about 20%, because gram-negative sepsis was half and half. Gram-negative half was gram-positive, so we thought at least we will be able to reduce gram-negative sepsis. So for 80% power and 20% relative rate reduction, we wanted to enroll about 8,000 babies. DSMB stopped the study in about the middle when we had enrolled 4,600 babies. When they said, I have been on DSMBs, and only time we have stopped studies is when there is something wrong and we want to open the study or peak and do such things. And then we found, and we were happy to know that it was obviously due to efficacy. And this is the eastern part of India, one state, where we did the study in 2 different districts that are 200 miles apart. And this will take me 3 hours. I can make a movie how it was done. At EVO1 grant, 5 years, $5 million to set up labs, to all the infrastructure, the trained people. And then finally, we did the study with an R01 because we had already done the preparatory work prior to that. So it was individually randomized blocks of 4, and we gave Synbiotics starting day 2 of life. Day 2, 3, or 4. First day we didn't give because many babies die due to birth asphyxia, and they may have early onset sepsis, which we won't be able to really do much about. And then they were watched at home for 6 days. And all adverse events and serious adverse events were reported. Then we did BACTEC blood culture and microbiological identification and stored the samples. And we had to set up NICUs, and those have become bigger and better NICUs now, use cell phones. And at the bottom you see 300 workers, who each village has one lady who was trained, and then a 3-tier system to really monitor the study and bring the patients the moment they became sick. And a lot of focus group meetings, movies were made, and they had to be told that, okay, unless you bring the baby right away, the baby is going to die. And at the end, this is what we got. We screened 7,000 babies and enrolled 4,500. And there were quite a few ineligible. I will tell you why. And then, we had very few consent denial, but we had some that were, by the way, our inclusion criteria were 35 weeks or 2,000 grams. We didn't want to enroll really tiny babies because we were there for the first time, and we didn't know they will be dying due to all different causes, including birth asphyxia. So, and some of them we could not enroll because they were born at the hospital. They didn't come home by 3 days of life, and some of them had early onset sepsis. And after excluding those 2,500, and it was very tightly monitored by all different groups, apart from the investigators who came and said this is how a field worker would prepare the Synbiotic and squirt it into the baby's mouth. And the results coming to, when we look at death and sepsis, there was a drastic reduction, NNT of 27. I was always bragging about it. Now I won't, after I know that FMT has a NNT of 3. When you look at culture positive sepsis, it was also reduced in massive numbers, 27 versus 6. What happened, which we had no clue about, the reason this study was stopped in the middle is the respiratory tract infection. In this country, respiratory tract infection or pneumonia, those are different diseases. We know what they are. WHO, on the other hand, classifies neonatal sepsis only for the developing world, where they pool all of these conditions, including respiratory tract infection, because they can diagnose in the field, they will give them antibiotics. So that component, we had quite a few respiratory tract infection that also got reduced significantly, which really tipped the balance, and that's why the study was stopped in the middle. Some other infections were also reduced, including tonsillitis and local skin infection and diarrhea. But then if you combine all infections, not just sepsis, then the NNT was 18. And if you include diarrhea, it was about 13. Other morbidities we collected because it was a probiotic trial, but we didn't expect that they will have less wheezing or atopic disease in the first 2 months of life. And we could conclude that the Synbiotic preparation significantly, reduced sepsis, but it also had some effect where it reduced lower respiratory tract infections. So we now know that apart from blocking just the bacterial translocation, there are other immunomodulatory effects that are impacts that are going on. And why? I mean, this will it's a lot more work to be done, which we have started now, looking back about the timing when we gave the preparation. Is day 2, 3, or 4? Does it matter? Those of you who are familiar with the Peter Abbey's work on BCG and the non-specific stimulation of the immune system, one could say that it has nothing to do with the lactobacillus. You gave an antigen and you give it at the right time. So now we are looking at, does it make a difference? Because we have 4,000 babies, we are looking at day 2, 3 versus 4 and see which ones did better, if at all. And I won't go into the, this was from the very first study where we looked at it has been published for the microbiome. Again, there are tons of changes, and just so that you see how many, I mean, they're so diverse if you look at them. And now we are looking at bacteria host cell interaction, which all of us are very fond of. And if you look at this just simple attachment, the recent electron micro ultrastructural analysis that we are trying to do, they're not as simple as we think. They're not just coming and blocking attachment. And they secrete different factors at different time, after half an hour versus 1 hour, 3 hours, and 6 hours. These are the lactobacillus. And when they come very close to the cell surface, on the left-hand side you see the E. coli, on the right-hand side lactobacillus. And if you think about gene expression, we are talking about consortia, we are talking about thousands of species. This was a study we published some time ago, taking the same E. coli that I showed you. You put it on cultured cells, it expresses mRNA and 332 genes. Lactobacillus plantarum alone, 81. You combine them, it's only 86. Something is going on the transcriptome level here. So I will, coming back again, products are being sold with different, they can change the color and say, okay, if it is pink, it is for babies. If it is chewable, it's for junior. Cultural has everything. They add a little bit of, again, nothing against cultural. I mean, they're wonderful, it's the most studied organism. But you add a little bit of vitamin D, it is for growth. You add a little chamomile, and I'm not exaggerating that, okay, it will cool your baby down. So that is one aspect. The reason I'm telling that you can't, this is something which Joe New told about this morning, LGG. The pre-post study has now shown that it increases necrotizing enterocolitis and sepsis. So you can't take it for granted. It may be a wonderful probiotic for 500 diseases, may not be for this particular one. And I will end by saying that many, many years ago, all of you have heard about this, how artemisinin was developed. Think about the cinchona bark. They were eating it, and then 1820, and then chloroquine was there for 100 plus years. And then even now, if you go back to those areas, nivaquine is used not just as an antimalarial. It is just used for everything. Little bit fever, you want to feel good, take a pill. Also as a food supplement. So this is, I was telling you, it's only 30 years ago when it was licensed and Sanofi took it as a drug. So if you're wanting to use it, give it intravenously, give it for cerebral malaria, of course it has to be developed as a drug. But can we stop the people taking the ground cinchona bark, or somebody who is wanting to sell it in a capsule so that they will have less of a pain, or it will help in fever? Answer is probably no, they will do it no matter what. And this was Eli Mechnikov, one of my heroes, more than 100 years ago. Alexander Fleming, also about give and take 100 years, who you know about penicillin. Now, what we are trying to tell, think about the antibiotic thing, it took 100 years to do this. And in probiotics, it will take another 100 years. Now we are kind of waking up. We can't expect that, okay, we have a broad spectrum probiotic that's going to do cure all ailments for all diseases. So a lot more work to be done. Thank you.
Okay, so we're going to move right along. I'm going to invite all of our speakers for session 3 up to the table, and we're going to combine clarifying questions and the panel discussion, but we'll take questions first, certainly.
So please step up and ask questions.
I have a question for Axel. So the mouse model that you showed, you kept them showing groups where you added the fragilis. Do you have a control group where you added a different bacillus or some other, just to show that this specific to B fragilis?
No, in that particular experiment, that experiment was focused on B. frag. However, the laboratory has done that with some other organisms that haven't shown those effects. B. frag was specifically chosen for this series of experiments, though, because of its known effect on leaky gut. And we were looking really at 4-EPS, and is 4-EPS potentially what's causing the behavioral abnormalities?
At the end of the day, we'll wake each other up. So here's the deal. Thousands of bacteria in the gut, thousands of things we could potentially grow, thousands of different diseases we could potentially go through. How do we sort through that huge matrix so that we don't have to do the 1,000 by 1,000 experimental design? What have you guys used to try to sort things out? 2 minutes. No, sorry.
Sorry, thank you. A couple of things that we've tried, and I'm sure there could be a lot more. I think that leaning on some human data from interventional studies is useful because of all the potential combinations. There will be many that are just relevant to mice. So that can make the experimental space smaller. Something else that we've started experimenting with that I think will be useful at some point, and I don't know if the prime time is there yet, but we're working on it, is try to start using just good old mathematical modeling to predict how communities will behave when put together. And when I say mathematical modeling, today I mean just pure empirical modeling, fitting adjustable parameters to experimental data to then be able to predict how communities of bacteria will grow together. Hopefully at some point this goes into actual mechanistic modeling, being able to say, you know, from that genome, I expect these things to be expressed in this series of interactions. I don't think the field is anywhere close to that, but it will get there. Personally, I think we're at a very exciting time now where we're starting to transition from just phenomenology, microbiology, immunology, which is great, to actual having a few assembly rules to work from. And I think that when the field gets to the point where engineers and mathematicians can start coming in because there's enough information that you can actually model things, that's when we'll be able to really dramatically reduce the size of the experimental spaces that we can explore.
Yeah, I would have responded the same way. If we want to think about how the consortia is going to change modeling, mathematical modeling is the only way to do it. But even at the same time, we have to think, okay, fine, we know this is how the consortium is going to look like. But what will be the physiologic phenotypic change? For that, to me, it may sound naive, but we have to go back to humans. And if it is probiotics, that is why I was asking those questions. We can probably, because it is not a quote-unquote drug and a molecule, and if it is safe, we can do larger studies. So whether it is diet, whether it is exposure, whatever happens, we don't really care because these are randomized. And as long as they are large, all those variables will be taken care of. They will be distributed in half and half. So we call it not efficacy, effectiveness at that point. So I think there are not thousands of diseases. If you really look at the textbook, there are not that many. So we have to think about the pathogenesis and see if this has a microbiome has a role, and then go from there and use the best single one or a small. I am against consortia because once you give it, they are into a consortium. Even in the newborns, within a couple of days, they have dozens of bugs. So at least I know that I am giving one that colonizes, that stays in there. But whatever happens, the ultimate outcome is what we are interested in. and that's what we will check all the changes, physiologic and phenotypic changes.
I think the other approach is also starting from first principles on both ends, understanding mechanistically what's going on in the disease state and better classification of diseases, but then also on a organism by organism basis, figuring out what their phenotypic effects in the host are, and basically create this microbial toolbox where you know organism A has this effect on these different parameters, and then you can start to pick and choose for this disease that has these defects. We will need organisms A, C, and E. But for this other organism or the same disease in a different patient, you may mix and match from that toolbox that's already well defined.
So I think all these points are important, but I still believe in physiology, going back to Vince Young's comments earlier about understanding the physiology and how these bacteria are interacting and what they're doing. I think mathematical modeling is useful, but we still need to keep our eye on what happens physiologically when we put these organisms together. And I guess you had mentioned going after all these diseases. My feeling is do no harm, first and foremost, and baby steps. To me, recurrent C. diff is one of the easiest ones to go after first, and using that as sort of a learning experience and branching up from there is sort of what I personally would think would be the way to go, and always keeping in mind that organisms don't always do what you think they're going to do the same way. Teenagers don't always behave the way you think they're going to do. No matter what kind of mathematical modeling you do, they'll always come out and surprise you. And we've seen surprising things come out of these bioreactors when we put things together that we were not expecting at all. And then we could go and look in animal models to kind of dig into that a little more deeply. But yeah, I guess I would err on the side of go slow and do no harm. I mean, the other thing, I have a colleague, Erica Claude, who's a neonatologist, and we've had a lot of interesting conversations about NEC. And she pointed out to me something that I had never even thought about before, and that was that for prevention for NEC, if it's 1 in 10, then that means there's 9 out of 10 babies that would be getting probiotics that didn't actually need them, and for a preemie, bit different for neonates that are healthy born babies. But for preemies, she was questioning what this will do long term if we now set their set points with these new bacteria that we put in. Nobody knows what's going to happen. And so she's very hesitant to use probiotics. And you know, I never even thought of that before. I think we just have to be careful. Well, actually, so what she's promoting is decreased use of antibiotics and push breastfeeding, because breast milk has been one of the most protective elements, and really low birth weight seems to be the risk factor. So if you can get them to grow, gain weight, they don't have this same risk of NEC.
I would agree with what's been said. I mean, we're dealing with incredibly complex interactions and incredibly complex systems here, so we just need some levers really to get in there and really start understanding mechanistically what's going on. And one of the reasons, obviously, why B. fragilis was attractive is because we knew it was likely going to be safe, but it also has some of the features that we wanted to see and some of the effects in animal models that we think might be able to make a difference. B. fragilis is probably having an effect on other organisms, and it's probably, well, we know, it's changing microbiome makeup to a certain degree. It is turning it a little bit back to what it is in wild-type animals. So it's not B. fragile alone, and it's probably not 4-EPS alone. It's probably a whole slew of things that are stewing around in the soup that some might be getting in anyway because they penetrate through diffusion. Some maybe can only get through a leaky gut. I think we just have to start chipping away. and figuring out what these things do, and that'll lead us into other directions, maybe into other diseases.
I'll just add one point to this in terms of changing the microbiota permanently or doing something, some damage to it. There have been, in our studies, we looked at the microbiome for 6 months, and after fourth month it goes down to 0. By 6th month you don't see that particular strain in there. But others have done, including, I mean, not in neonates, but in infants, Isolari and others in Europe for the asthma allergy studies, that it doesn't stay permanently. It goes away. So you are not changing it for good. And in fact, we were asked by the Indian IRB, one crazy person came and said, you have to follow them for 18 months, 2 years, and finally, not 2 months, but 2 years. I said, what, 2 years? Our protocol has been approved for 2 months. We can't follow them for 2 years. Why? How do you know you have reduced sepsis? You have saved millions of babies. How can you prove to me that they're not going to grow horns in 2 years? And that is the real critical period. And what happens is you have all stunting everything and the GI dysfunction takes place. How can you, I said, if they have horns, they will be able to better fight against others. And but we had to follow for 2 years. We didn't do microbiome, but we had to follow and showed that there was nothing drastic. So long-term follow-up, longitudinal assessments, and with all the tools we have now, I think we should be fairly comfortable telling how we are changing and if the change is good or bad. And if something wrong happens, that happens when you're trying to discover something.
Deborah Topham with Knowledge Bank. Dr. Panigrahi, could you talk a little bit more about the dosing that you use for lactobacillus, along with what dosing you used for the FOS, and what kind of or type of FOS that you used?
Yeah, it was fructooligosaccharides, 150 milligram in each dose, and 10 to the power 1 billion organisms, Lactobacillus plantarum. So it was available as a lyophilized powder, which was mixed with 5% dextrosaline on site, and then it was put into the baby's mouth for 7 days. Depending on whether we started on day 2, we gave it for, all of them received about 7 doses.
That was 150 milligrams of FOS?
Yep.
And I guess at that point, are all of the babies in your group primarily breastfed, so they might have gotten the mammalian oligosaccharides along with the fructooligosaccharides?
Yep.
That would be the sugars, or is that
Yep.
right, Dr. Young, on that
Yep.
point?
And in fact, we had some very angry people writing to Nature that you are unethical. Breastfeeding is the only thing that really helps, that has reduced this and that. We said, no, you are unethical because you have been chanting about breastfeeding for 100 years. Nothing has happened. Infection has gone up. Although breastfeeding rate has gone up in developing countries, infection rates haven't gone down at all. It has gone up. So all the ones that we are showing, they're all exclusively breastfed. Unless breastfeeding was established, they were excluded. There were quite a few. So in spite of having breast milk, in spite of having oligosaccharides, maybe bifido, maybe whatever, there is not enough good bacteria that could protect them during that window.
Hey, Krishna Rao here with University of Michigan. So my question is, I guess, related to that, and maybe the other panelists can chime in too, which is, how did you decide to co-formulate with FOS to begin with? Was it based on preclinical data? Was it based on some idea of what substrate this would grow best on? And in general, how do you make those decisions? When do you arrive at the decision where you say, hey, this probiotic is not enough, we need to co-formulate with something?
Well, I can answer. That was the only thing I have done in my life without solid scientific evidence. If you ask me, give the same plantarum without fructooligosaccharides, will it colonize, will it work? The answer is we didn't do that. I think we were impatient because we had already spent 4 or 5 years doing 2-3 other clinical trials that we expected that they are going to colonize and have something. When they didn't work, we had one organism that was expected to colonize. We wanted to have it do it even better. And we had enough evidence that if you just give probiotics, that's not enough. You have to give something from outside. So it was just, we wanted to increase our chances of success, so we added another. Instead of FOS, will GOS be better? All that we don't know. We have to work on it.
So to keep things going on this, I put up a slide. We have a couple of slides. One focusing on models. So everybody talked about a model in one way or another. And, so in essence, we've talked about or we've heard about the complex relationship between host and microbe, and that complicates these models. So we have everything from a hostless fermentation model to humanized mouse models to the human model itself. So and the focus of the session is on strains. And so I think I've heard on multiple occasions that strains matter, and that just a simple L. plantarum is not out of the 20, it's not the same. So how do you leverage these models? And if I could go one by one, how have you leveraged these models? Is it one particular strain of beef raj? Are the strains falling apart? Are you in your RoboGut? Are you checking multiple strains of the same genus species? So maybe just kind of briefly go through and talk about why strains matter and how you've leveraged models.
Yeah, well, with regards to beef raj, definitely strain matters. There's no question about that. There are enterotoxic strains. We wanted to avoid that. The strain that we ended up using happened to be the strain that was in Sarkis's lab, but we have used other sources of beef raj that have not shown the same level of efficacy. So there seems to be some magic sauce in the particular strain that we're dealing with. We don't really know what that is. Again, we know what some of the metabolites are, but we don't know really whether, well, certainly not the only metabolites, and are those the only metabolites that are having an effect distantly in the CNS? We don't really know.
Yeah, so to probe a little bit more on the basis of this question, is that beef raj paired with that mouse model, or is that beef raj going to be something
that's
relevant to human?
Yeah. There are other mouse models. There are other animal models of ASD. There's a BTBR model, which is a inbred spontaneous model. There's the CatNap2 model, which is a genetic model. And we do see efficacy on the behaviors in all of them. But all the models are different. They all cause different aspects of the disease, and they may or may not replicate what's happening in humans. Not all of them have a gut component, so we can't really look at the effect on the GI abnormalities in any of them. But we do see consistency between those three models, the effect on the behavioral components, at least the behavioral components as they are shown in the phenotype of that particular model. So that much we know. But in answer to the last question, are humans still the most reliable model? I don't know if they're the most reliable model, but to me they're the most important model. So as soon as we can extrapolate safely beyond the mouse and get into humans, then I think the story starts over again. And then we'll start learning new things. And there's much that's unknown about ASD anyway. We'll certainly learn more in our exploration. Whether it results in an effective treatment, we hope it will. But we'll certainly learn.
I think so. My view is for matters of safety, PK/colonization, humans have worked best for us. And we haven't really relied on the results of the animal models for these two considerations for a number of reasons. I think for matters of causality, mechanism, and generally as a screening tool when you have to go through a number of different possibilities, humans for obvious reasons are not usable or appropriate. So for that we've relied on animals, and I think that for those uses they can be very helpful. So some of the models that have obvious limitations like germ-free models or antibiotic-treated animal models, mouse models of SPF background can actually be very useful to understand causality, to learn things about mechanism. So I think that depending on the use that you give them, even fermentation models can be useful if you're trying to explore simpler questions that don't really, where the immune system doesn't really play a very obvious role, you just want to understand microbial interactions, those may be sufficient too. So it really depends on what question you're asking.
I'm Lawrence Royce, and I'm wanting to know, there was one mention of lysates used, and I was wondering, has anyone done any work with killed species, and what kind of successes have you had? I know there's been quite a little bit of research that was done in the former Soviet Union using lysates, very successfully modulating the immune system, and with some very interesting results. I think one of the first discoveries in the Soviet Union was in 1976 with, I think, with Lactobacillus rhamnosus, that a lysate that had some interesting results. And I was wondering, does anyone else, who's done work in this area?
We've tried a lot of either bacterial lysates, heat-killed organisms, or a variety of endpoints. And I think it depends on the organism and the endpoint as to whether it shows an effect or not. So for some things it works, for other things it doesn't. I think figuring out the why and when can you predict one to work or not still needs to get resolved, partly because to define sort of what the molecules and what's the pathway of that interaction. Does it really require colonization or not? Does it require a certain threshold that we're not giving, a concentration that we're not giving through the lysate in dosing regimens and things of that sort? But I think that for certain aspects or certain phenotypes, either lysates and or killed organisms, at least in our hands, have been successful.
The only other thing I would say is, I think with regards to ASD and the work that we've done so far, I think the organism is important. As far as what would be in a lysate that might have an effect, well, we've certainly looked at the metabolomics and looked at the metabolites that are different, and there certainly could be situations where a metabolite might be beneficial. But then, one would want to have a continuous source of the metabolite. Otherwise, the metabolite itself might be a drug. Then I think we would want to know what component of that lysate is responsible for the effect that we want to see, and then really focus on that particular component.
Yeah, I would respond the same way. I mean, we have tried, but only with very specific bugs, only formalin-killed bacteria, whether they could do the same thing in our in vitro and animal models, and they didn't. That doesn't mean that the components, if we are now thinking that it's not the whole bug that is doing 100% of the thing, if it is immunomodulation, maybe the components would have done something for that immunomodulation piece, even if they didn't help in against bacterial adherence and translocation. It all, I guess, boils down to the physiology and what you are trying to study. I think future years we will see different components and how they really interact with each other or with host cell and the ultimate physiologic effect. Those will be done in future years.
Yeah, well, there is PSA that also came out of Sarkis's work, I believe, when he was at UCLA. And there's a company, I can't remember the name of it, Symbiotic or something like that, that's working on specifically PSA and its effect on the immune system.
So I'm going to interject, as moderator. I'm going to take my prerogative and ask kind of one last topical question. And it really has to do with the fact that clearly we've seen the historical use of probiotics, and now we see this upsurge in rationally selected and based on human commensal colonization and the causal association with diseases. So I've asked a series of questions here, but I'm going to skip through a little bit. And I think it came up, I didn't mean to, I was going to bring it up, but Elaine mentioned this recent study, and it has to do with the idea of what are our high-resolution assays, what are is colonization, and even in a human model, what is that telling us? And so we have some recent papers that have just come out, and they have suggested that there may be more to the story of just pass-through and detection in stool. And so I want to ask, essentially a question here. I mean, we've heard about high-resolution assays to detect the specific strains. That actual organism that you've given, Burnett talked about that. what is the role for actually looking within the intestine or within the communities to assess the efficacy of your strain? Because in the paper, one of the things that they did correlate is mucosal host transcriptional responses were correlated with mucosal colonization. And I think that's an interesting concept, and I'm wondering how each one of you take that idea and move forward with it, or not, whether or not colonization through stool detection is sufficient.
Well, I would fully agree with you, and I won't say that, especially if I'm thinking about the organism that I used. I think because I did in vitro experiments and animal experiments, I know that they had to be in contact with mucosal cells, and now we have the extra non-GI impact. And so if they wouldn't be there, they are not associated with the mucosa, I will be surprised if they're going to do their job. Doesn't mean that other probiotics, other components won't do it. So lacking that if I can take biopsy every 3 days from a baby, I'll be more than happy to do it. And many people have complained that, stool has nothing to do with it. They come, get in and get out, the real ones are inside, so you're not looking at it. But that's the best surrogate we currently have. And I'm sure, and that's why we use animal models, and that's why we have to have some other models to have some idea.
Oh, I totally agree with you, and actually, we really need better diagnostics than we currently have. That paper that you mentioned, it's where the rubber meets the road, right? The epithelial microbial interface, it totally makes sense that there's going to be stuff happening at that interface, and stool is kind of a crude measure. It's just kind of passing on through. So we're missing a lot of very useful information, but biopsying is difficult. As you know, we can't always put that into clinical trials, and it's complicated, ranging from colonoscopy time and the colonoscopy suite, being able to get the biopsies. But there's no question that we have to come up with better diagnostic tests. I mean, if you think about it, when we give antibiotics, we measure creatinine. Why aren't we measuring what it's doing to the microbiota as part of, we measure serum levels of aminoglycosides. We do all these things with antibiotics. We don't even measure that. There's a lot of diagnostics that need to be developed, and I think that whole area is being completely overlooked.
I guess I'll disagree a little bit in that I don't know that colonization itself is always required. To use the example from Bacteroides fragilis, either polysaccharide A or other bacterial sphingolipids, those products by themselves can still exert effects on the host and in disease models. So it's not clear that clearly you don't need bacterial attachment because there's no bacteria in those experiments. But then there's a question of how does those molecules interact with the host. And so there's still some host recognition of those molecules in some capacity. But whether or not you need bacterial colonization as a starting point, in your studies, that was sort of a prerequisite to move forward, is that the organisms had to colonize. But if one can identify the molecules themselves that then have an effect, you may be able to bypass that step of bacterial colonization itself.
Yeah, I would agree. Particularly with B. frag, I don't think we expect that it's going to colonize as quickly as we get it. We'll find out once we get into humans. But we aren't anticipating that it will.
And I still have to read the paper in detail. I just came the abstract. I looked at the summary from Cell, but I'd be skeptical about throwing away all what we've learned from fecal samples, because everything that this field knows is from fecal samples, and I think we've learned very useful things about what happens to immune phenotypes, for example, based on information that you can gather from stool, what happens to colonization resistance based on information that's in the stool, and then acted on these predictions to learn other things. So I'm sure that other types of data are useful, and when we can all have them with the tools available, then let's all have a party. But until then, and especially, if realistically, it's a tour de force to ask healthy individuals to go through a colonoscopy plus anesthesia plus whatever they had for no benefit for the healthy individuals to ultimately get this information. We're never going to get that. But we are going to get colonization from fecal samples if we ask for fecal samples and the patients are nice enough to give them to us. So I think there's a lot we can do with the information from fecal samples.
So I didn't mean to suggest that stool sampling is not worth doing. And in fact, in that Cell paper, they do sample stool in addition to doing the biopsies, but they have more ends of sampling from stool samples than they do biopsies. And certainly, I think that it's important, I guess, for certain diseases, though, like for recurrent C. diff, for example, I think it's a diversity issue. There's actually, I think, less of a host immune component here, maybe than say for IBD, for example. And so for conditions like IBD or ulcerative colitis, maybe we will need that additional information from biopsies. And I think a lot of it is going to depend on the disease entity that we're talking about.
Okay, with that, sorry, last question. Sorry if I forgot, please.
Hi, my name is Shenhua Li. I'm from Data Revive LLC. It's a regulatory consulting firm in Maryland. I have a very general question. So in some of your studies, you're using naturally existing streams as a drug to treat diseases. And if one day you found it's an efficacious drug and it's been proved, so is there IP protection on this drug and who owns the IP? That's my question.
Boy, that's a question for our IP attorney who's not here. Yeah, well, I think we believe that we'll have protections. Certainly we'll have the protections associated with the drug approval, with a biologic approval. But also method of use patents and things such as that, I think, will provide some level of protection, as well as the fact that it is a specific strain that we're talking about. And we believe that we'll be able to turn that into a therapeutic that's easy to take, a lyophilized preparation that's easy to take, and we'll have specific knowledge around the manufacturing method of it as well.
So the bottom line, I would say that naturally occurring strains are not patentable in general, but if you show that it works for sepsis, you can patent it. But if you show that it's working against cancer, you can patent it too. I mean, that's the general take-home I have learned in the last few years. But it will be really interesting when you find out if there is a component, like in our bug, we see that it does something, it secretes something. Then it again comes very close to the cell, intimately attaches. So then if we can find out what is that piece, what is that component that's doing the job, then that can be a drug, that can be a separate IP altogether. But until then, the live bugs, the whole bugs, I think it is yes and no. You can patent and you can trademark, you can do all different things, but it may not be as robust as having a chemical compound.
Yeah, and again, I should say if you're developing an organism that's not currently available as probiotic, and you get into a drug development process with it, and you file an IND and you start generating data, my understanding, and I don't know if the people from CFSAN are still here, but my understanding is that once you go down that pathway for something that hasn't been commercialized as a nutritional, you can't do that anymore. Or you can't start marketing it as a nutritional one once it's been shown to be a drug.
I was just curious about the strains that are not genetically modified, that are naturally existing. So if for example, the strains you found can be used to treat one disease and it's been approved, and then in another study it has been found to be effective for another disease, but yet you didn't patent it for that use, but can consumers just use it for another treatment without,
They'd have to source it. They'd have to get it from somewhere. And I would think if as part of somebody's clinical program they started seeing evidence that it might work in another condition, they'd jump on those patents right away to try to cover that from a method of use standpoint.
I think the other complicating piece there is what is the idea of a biosimilar in the field of probiotics? How far away from Bacteroides fragilis or any of the 8 strains that Vedanta has do you have to be? Is it a different B-Frag strain sufficiently far enough? Everyone has patents written to be incredibly broad, but until this goes to the courts and having courts adjudicate how narrowly those really have to be drawn, how many SNPs away from the genome sequence they submit do you need to be infringing on their IP? So you still be able to make some money off this, even when they do all the legwork.
All completely untested, as you said. That's the Wild West, and biosimilars are not as easy to get on the market as a generic is. You do have to do some clinical work.
Okay, so with that, we're gonna close session 3. Thank you very much, speakers. Very interesting and entertaining talks. I really appreciate it. And we're going to round the bases on this workshop, and I'm inviting Dr. Carolyn Deal, who opened us up today, to give some closing remarks.
Well, I know it's getting late, and I will just take 2 minutes. First of all, I want to thank all of our speakers today. Really appreciate them putting time and effort into their talks and contributing this. And then really thank all of you in the audience. You all were in this room. There is actually 2.5 overflow rooms in this building downstairs that were also full. So want to thank everyone who participated in all of the discussions. Because I think this has been something that we really wanted to hear a broad breadth of input on. I know my colleagues at CBER and CIFSAN did also. But then last of all, I really want to thank the organizing committee, which some of whom you've seen today, who were the moderators for this session, who put a lot of time and effort into coming up with this program. And so really want to thank all of them and all the colleagues from CBER and CIFSAN who participated and all the other NIAID participants. And so the last thing I wanted to leave with you in thinking about all of this is I think all of us have a lot of enthusiasm for the possibilities in the future for live microbiome-based products. I mean, this is a new, growing, and evolving area, and I think we're all learning. We're intrigued by a lot of the possibilities, but I think we also know that there are cautions that need to be considered, some of which have come out today. I think some of the things we've heard is there may be advantages to considerations for well-characterized products in terms of reproducibility of manufacturing, and more importantly, even to think about to ensure reliability of use. So there's one consideration as we move forward. I know there's been some debate about where there should be the lines between the regulatory considerations for probiotics and live biotherapeutics. And I think there's always some evolution and thought of that as how we go forward. But I think then one of the other things is those are the product issues, but there's also the clinical issues. Many of these are complex infections and diseases. They're not all well defined, and I think the case of necrotizing enterocolitis has certainly shown us that it's not always a well-defined infection. And I would even argue sometimes C. difficile infection is not also. And so it points to the need for clinical studies with well-defined clinical endpoints and also with well-defined diagnostics in those studies. So there, I think, is the other potential gap area that we see is some of the need for new and better refined diagnostics. All of these are needed to support regulatory decisions from our FDA colleagues in the future. But I think importantly for all of us in the public health and medical community, what we all really want, and most importantly, is to be able to have reliable regulatory decisions and to provide informative, useful, and reliable information to patients and to the providers. And so that's the thought I really want to leave you with, is all of this as it evolves over the next years, and as we get products that we can move into routine use, is that's the ultimate goal, is to be able to provide reliable, useful information, not only to the providers giving these products, but to the patients that receive them, and hopefully that we can improve them public health. So that's all the comments I wanted to make. I really, again, thank you all for coming, and I hope you don't drown on the way home, because the hurricane appears to have moved up here, leaving our North Carolina colleague ability to get home. So thank you all very much.
You didn't get it. We're still kicking. Take care.